The second chairperson, Professor Marc Talefo from University of Montpellier, France. Marc studied chemistry and earned his PhD from the University of Paul Sabatier, France. His research focuses on the discovery of breakthrough methodologies in the field of organic synthesis and homogeneous catalysis, bond formation by a copper iron manganese catalyzed annihilation of nucleophiles, transition metal free mediated coupling reaction, and related things. So, without wasting much of the time, I request them to please take care of the session and coordinate.
Hello.
So this is for the presentation presenters and as well as for the information of the chairperson. So you have got list of 14 people. They will be presenting their research papers for 4 minutes, and after each presentation, you may give your insights and words to those presenters, and then you're supposed to call the next one.
I have received a selected oral presentation list. Total number of participants 46. So when participant who will deliver his presentation, that only 5 minutes will be awarded. 4 minutes. 4 minutes. I request any participant completed within 4 minutes.
The first speaker is Aarti Saini.
See, you have to speak here. This one is pointer. You have to press it. Please
Good morning, all of you. I am Aarti, PhD scholar from Department of Chemistry, Central University of Rajasthan. Topic for my today's presentation is magnetic study of iron doped tin oxide materials obtained by thermal decomposition method. These are content of my presentation. So we will start with the scopes of iron doped tin oxide materials, which involve many applications in spintronic devices, magneto-optical devices, and these have good electrical conductivity, show paramagnetism, and have sensing property towards ethanol, carbon monoxide, hydrogen sulfide, methane, and acetone molecules. And different preparation methods involved in reported in different literatures involve reactive magnetron spurting technique, protic sol gel technique, and many others. And we have prepared hexameric organotin ferrocene carboxylate drum by already reported procedure and done its thermogravimetric analysis, which shows the compound shows 50% decomposition above 300 degrees Celsius, and after 450 degrees Celsius compound shows stable behavior. That's why we have chosen 600 degrees Celsius for temperature for decomposition of our compound. We have used 2-hour time for at 600 degrees Celsius for in muffle furnace for decomposition of the compound, and we have got dark brown colored solid which we have characterized by different techniques. First of all, we have done FTIR analysis, which shows stretching mode vibration at 593 centimeters reciprocal, which shows the presence of OSNO bond of SNO2. And powder XRD analysis were done, and Ritwell definement analysis shows that the 97% phase of the material contains iron and iron doped tin oxide material, and the crystal system was tetragonal and space group was P42 by MNM. And different planes, Miller planes are indexed in the figure. Structural model shows that in single unit cell there are 8 tin or there is 50% possibility of tin and iron occupation, and the tin iron atoms are present at the corner as well as body center of the unit cell. Every tin and iron atom is coordinated to 6 other oxygen atoms, and the bond distance and bond angles are given in the parameters are given in the table. Morphology study shows that SEM images shows film-like behavior, and agglomerated particles can be seen in TEM, and side pattern shows amorphous nature of the material as concentric circular rings are present. And HR-TEM shows different orientation of lattice fringes, which have interplanar distance of 0.332 nanometer, which is for 110 plane of SNO2 material. XPS analysis shows presence of different elements in the confirms the presence of 10 3D state, iron 2p and 3p states, and in oxygen 1s XPS, iron oxygen stretching frequency, XPS peak was clearly observed. And tin 3D XPS shows the presence of SN +4 oxidation state in iron doped state as well as in some oxidized form on the surface oxidized form of SNO2. And iron 3 state was confirmed by iron XPS, which shows peak at 713 electron volt and 726 electron volt, as well as satellite Fe3 satellite peak at 719 confirms the presence of Fe3 positive in the material. And along with SN3D peaks, SN3P peak was observed in the range of 716 electron volt. Mosberg studies confirm that Fe3 oxidation state is present and octahedral coordination is present. Zeeman splitting pattern was not observed. Magnetic analysis shows that ferromagnetic shows the material shows ferromagnetic behavior, and it shows 72 Kelvin spin reorientation temperature, above which the material shows paramagnetic behavior, and below that temperature it shows antiferromagnetic behavior. And MH graph of the material shows ferromagnetic behavior. The difference, there is space between, there is no non-zero space between this peak, so it shows ferromagnetic behavior. This is pictorial conclusion of my work, which shows preparation of SNO2 material, its powder XRD, and confirmation by other techniques also like XPS, which shows SN +4 oxidation state, iron +3 oxidation state. Iron oxidation state was also confirmed by Mosberg study, which shows +3 oxidation state as a single paramagnetic doublet was observed here. And SEM measure shows film-like material formation, and TEM shows that different lattice fringes are differently oriented in different directions. That was also confirmed by side images also, like different, there is no, there is formation of polycrystalline material, and the prepared material shows magnetic behavior, ferromagnetic behavior. This is acknowledgement. Thank you.
Your research entitled Thermal Decomposition Method.
Yes, sir.
Can you explain that the magnetic properties of conductance?
Magnetic
Magnetic
properties.
properties of conductance when you adopt stannic oxide. That the properties of the magnetic behavior. It means conductance. The properties it will be increases about this method. Can you explain?
Conducting studies we have not done. So we have done magnetic studies as we have done some.
Basically, magnetic is probably related to conductance, non-conductance, insulator.
Second, 12. Yes. So conductance, I think, need electrical studies. We have done magnetic studies. Here we have done zero field cooling and field cooled temperature dependent magnetization was recorded with the temperature around 390 up to 390 Kelvin temperature. The compound shows non-zero behavior which exhibits super paramagnetic behavior, and up to this temperature like 72 Kelvin. The material, paramagnetic behavior increases and antiferromagnetic behavior increases below 72 Kelvin temperature. So only magnetic properties we have studied, not conductance studies.
Thank you.
Thank you, sir.
Okay, next participant.
A very good morning to all. I, Surbhi Bhatt from Central University of Rajasthan, today here is to present a part of our lab work on the ruthenium pincer complex catalyzed an alkylation reaction of amine and alcohol, which is an efficient system for biological applicability. So starting with what are these pincer ligands? So pincer ligands, if we see, these are generally the tridentate ligand which occupy the adjacent binding site. As here we look on the structure, his X and L are the tridentate ligand which adopts the meridianal geometry. So these types of tridentate framework imports thermal stability to the system. If you look on to the different metal hairs, here L is represents the hardness and softness, metal binding, steric and electric control. Where it is Y determines the ring size and bite angle, it is also a chiral pocket, while this Z determines the electron density of a central atom, it is also an anchoring site. Where this part of the metal is a cavity for the metal binding with a tunable accessibility, and it's also a site for the ancestral ligand. So why we have chosen this ruthenium pincer complexes? Because it has some advantage. It has a strong electron transportability, low redox potential, high coordination affinity, high Lewis acidity, air and moisture intensity, variable oxidation states, and so on. So if you look onto the ruthenium pincer complex, it has a very wide range of applications in the hydrogenation reaction, transfer hydrogenation, alkylation reaction, cyclization reaction, dehydrogenation reactions, and dehydrogenative functionalization reactions. Keeping all in this mind, when we look onto literature, so these are some complexes of the ruthenium, which is very well known and is also applied in a very different organic transformation. Some have uses in the normal nitro to anion transformation, some have using transfer hydrogenation, and so on. So these all ruthenium pincer complexes, if we see, these are generally the aromatic systems. And there are very few reports on the aliphatic system, which is shown here in the organic letter in 2021, which uses an aliphatic system. So a plan is to design aliphatic tridentate systems in which this R can be alkyl or aryl. This PR can be allyl or aryl phosphines, and metal we have kept as ruthenium constant, which can tune both the catalytic and the biological application. So, for the synthesis of ligand in metal, for the synthesis of ligand, we have chosen this reported procedure where we have
started with bis 2-chloroethylamine. We reacted it with in situ generated nucleophile and we got this ligand L1 and L2 in 80%. Then we reacted this ligand with ruthenium precursor in toluene. After 3 hours, we got these 2 complexes of sulfur and selenium based in 84 to 86%. Then we move to its characterization. We have characterized both the complexes through proton NMR, 13C, 31P phosphorus, elemental analysis, IR spectroscopy, single crystal XRD, HRMS, UV visible spectroscopy, in which this ligand can be with a transition of pi to pi star, and this metal can be, the complexes are basically the metal to ligand transfer. Then we have also done the cyclic voltammetric studies in which we shown that in the sulfur-based ligand, the potential is around 0.6, while in 0.62 in the case of C2, we show that there is slightly better donor ability of C2 than C1. Then we have also characterized it through single crystal XRD. We have formed this crystal with the slow evaporation of DCM. This particular crystal crystallizes in the space group P1 bar, where its geometry is distorted octahedral and the crystal system is triclinic. After completing all the characterization part, we then move to the catalytic part, and we have chosen this orthophenyl diamine. We reacted it with benzyl alcohol with the appropriate catalyst-based solvent to form this 1,2-disubstituted benzimidazole product. So why we are focusing on this benzimidazole? Because this particular moiety has a very core importance in the pharmaceutical and biological application. So starting with the optimization reaction, we have first kept the solvent constant, which is toluene, and we just differed the base from potassium carbonate to potassium tertiary butoxide. And we came to know that in the potassium tertiary butoxide, we got the maximum yield of 96%. Then we kept the base constant potassium tertiary butoxide and just differed the solvent from the toluene to DCM, and we came to know that in the toluene, we got the maximum yield. After that, we then move to the second catalyst. We have applied the same reaction and it's got the decrease in yield. We show that our catalyst C2 is the better. And then we have done the same reaction with the C2 in the increase of catalyst loading, but it got the same yield. So after that, so in the entry 16 to 18, we just differ the time, the temperature, without the catalyst, without the base, and we just got a decrease in the yield. Completing this all, we then move to the substrate scope, and we have formed the 12 different substituents, keeping the best condition in hands, and we got these all in the 96%. This is a plausible mechanism for the synthesis of 1,2-disubstituted benzimidazole, in which we have taken this complex, reacting it with tertiary butoxide. There is a removal of the Cl, BiCl, addition of the alcohol, and we form this. After the hydride transfer, we got the aldehyde, which on reaction with orthophenyl diamine, through this process, it forms the 1,2-disubstituted benzimidazole product. So our catalyst is also converting the benzyl alcohol to benzimidazole. So to the curiosity, we have done the catalytic aerobic oxidation of the alcohol, in which we have chosen this different benzyl alcohol and converted it aldehyde in this 97%. So after completing all the catalytic application, we then move the biological application for the anti-cancer. So if we look on to the anti-cancer, after platinum, cisplatin and analogs, the ruthenium-based complexes are in a clinical trial. These are shown here. Previously in our group, ruthenium and copper complexes are explored in the different cancer cell lines. So continuing the same, I have started with just the preliminary of this biological application. So starting with the DNA binding through ethidium bromide displacement assay. So DNA, if we see, these are very non-fluorescent molecule, while this ethidium bromide is a weakly fluorescent molecule. But when this DNA and ethidium bromide intercalated with each other, this particular moiety is a highly fluorescent. On adding a ruthenium complex, there is a decrease in the fluorescent intensity due to displacement of ethidium bromide, which is shown here. Continuing the same, we have then used these two equations, Stern-Volmer and modified Stern-Volmer, and we found that it is in the range of 10⁶ and 10⁵ the quenching constant, which shows that our complexes are binding with DNA in a moderate mode. Similarly, after the DNA binding, we have started the protein binding studies, taking the BSA, bovine serum albumin, because it has a very similar structure with a human serum albumin. So BSA is itself intrinsic fluorescent due to these moieties, tryptophan, tyrosine, and phenylalanine. We have done the subsequent increasing of the C1 and C2, and there is a quenching. Similarly, we have solved it through Stern-Volmer and modified Stern-Volmer, and we got in the 10⁵ range of quenching constant, which shows that our compound is very much interacting with the albumin, and it is going in the body if in future with very ease. Similarly, we have done the microenvironment and conformation synchronous emission spectra for the Dell 60 nanometer and 50 nanometer to see the involvement of tryptophan and tyrosine residues in a molecule. This all work has recently been published in Wiley Chemistry and Asian Journal. The future scope of this will be that we can tune this steric and properties of ligand and design, and we can explore the different types of benzimidazole. We explore the different set of compounds. We study the mechanistic study through computational. We study the biological properties of this pincel complex. We explore these in various cancer cell line. We study the mechanism of these complexes inhibiting cancer cells. At last, I would like to acknowledge my supervisor, Dr. Anuj Kumar Sharma, Dr. Hemant Joshi for making me learn all the catalysis and synthesis part, Central University of Rajasthan for providing all the facilities, CSI and SARB for fund, my lab members and friends. And at last, I would like to thank University of Rajasthan to give me an opportunity to speak here. Thank you.
Thank you. You transform alcohol in aldehyde with ruthenium catalyst, right?
Yes, sir.
Do you know another type of catalyst able to do that? Another metal?
Other type of catalyst which involve this hydride type?
In the literature, I mean.
Yes, sir.
In the literature, do you know another type of catalyst with other type of metals, not the ruthenium, which is able to transform alcohol into aldehydes?
Yes, sir. They are iridium, nickel also.
Okay. Okay, it's okay. Thank you.
Thank you, sir.
In your research presentation, that the ruthenium, I think iron family, very cobalt nickel, ruthenium, rhodium, palladium, osmium, iridium, platinum. That you prepare the complex ruthenium with the 6-member complex. So what is the role of hapticity?
Sir.
Just this. Next.
Yes, sir. This one?
Next. Yes. Our lab work.
This one, sir?
Ruthenium direct link to 6-member ring.
Yes, sir.
So what do you know about the hapticity?
It is eta 6.
Nita 6. So what is the role Nita 6 complex in ruthenium for biological purpose?
Sir, basically, these moieties not a very advanced slope, but mainly this type of system, which is other than this aromatic type, that affect the most. If we replace this with hexamethylbenzene or benzene, so it is very slightly different in the biological properties. Thank you, sir.
Okay, next participant, OP3.
Thank you, ma'am. A very good morning to everyone. Myself, Sohan Singh from Dr. Joshi Research Group, Central University of Rajasthan. And I will talk in this session about the organochalcogen-based palladium pincer complexes for sterically favored selective CH activation reaction. So overall, I will discuss about the organometallic chemistry, and specifically, I will focus on the pincer type metal complexes. As my senior Surbi, we have already discussed about the pincer type ligand, but I would like to add some additional point that pincer metal complexes basically tridentate ligand coordinate metal complexes in which ligand bind to the metal center in the meridional fashion, and due to the meridional fashion, the electronic transition gets better, which results in the very good stability. And apart from it, a typical pincer metal complexes have hemi-label arms due to which the reactivity is generated. So we can alter various things to design a new pincer type ligand and its metal complexes like metal center, ligand, the atom directly connected to the metal center, the type of ring, substituent on ring, and the spacers in the arms. We can introduce the electronic effect on the directly connected atom to the metal center. So we can control many things in the pincer metal complexes like steric effect, correlative bite angle ring size, And we can also introduce the hemalability in the this type of complex. So on the basis of metal center and types of directly connected atom, if typical, we can give a name to the pincer metal complex like SES, PCP, NNO. So this type of, sorry. So these are the atom directly connected to the metal center. So the initial complexes were based on the phosphine, and their ligand was thermally stable but not air and moisture stable. So to remove this hurdle, we introduced the organochalcogens in the connecting atom. And the yield was also issue when we use the base metals. So to remove this issue also, we used noble metals like palladium, ruthenium, rhodium, etc. Organochalcogen based palladium pincer complexes are widely explored in various fields of sciences, and due to their some unique features like high thermal stability, high reactivity, excellent yield. When we talk about the catalysis, least catalytic yielding, good recovery, they show very good tolerance for the various functional group, catalytic versatility, mild reaction condition, atom economy, and moisture insensitivity. So due to these beneficial properties, these complexes are highly explored in various fields of science, like catalysis. Most of the work is done in the catalysis, but these catalysts were also explored in material science, bioorganic chemistry, as the previous discussion was there, supramolecular chemistry, biological and pharmaceutical sciences. In this slide, some of representative examples of organochalcogen-based palladium pincer complexes are given, in which palladium is present in different oxidation states. So taking idea from the literature, we have designed NCSC-based palladium pincer complexes, and for which we used reduced diphenyl diselenide, in the dry DCM to get the pH SeCH2Cl, which further reacted with the 3-hydroxybenzyl in the alkaline condition to give the ligand precursor. And this ligand precursor reacted with two different amine, tertiary butylamine and admethylamine to give the corresponding to different ligand. Where if R is tertiary butyl, then it stands for the L1, and if we use the admethylamine, then ligand is L2. These both two ligands reacted with the further palladium precursor in acetonitrile to give two different complexes. These novel synthesized ligands and catalysts were characterized by various analytical and spectroscopic technique like HRMS, FTIR, UV visible spectroscopy. The point which I want to get in the attention that when ligand became in the coordination, these two anisotropic protons become distereotopic and give two different signals in the proton NMR, which confirm the coordination of ligand to metal center. So in this number, we can see that this CH2 proton, these two SeCH2 proton, these are anisotropic in the ligand, but when it binded to the metal center becomes distereotopic and give two different doublet. Fortunately, we get crystals of C2 in which R is admethyl in the DCM and hexane. By the crystal structure, it is clear that palladium has distorted scorpionar geometry with +2 oxidation state, and the all bond length palladium, selenium, palladium nitrogen, palladium carbon, palladium chloride were in range which is reported in earlier. In this crystal structure, we can see that the space near the metal center is confined, so substrate needs specific orientation to approach this metal center, and due to which the regioselectivity is generated. So this type of catalysts are widely explored in the regioselective activation reaction. So we have also tried a type of reaction which is the activation of 3-phenyl thiophene ring. In the 3-phenyl thiophene ring, there are three types of very important and reactive site, and in the earlier reports in which palladium is used, C5 position is activated with ruthenium.
Please conclude. Conclude, please.
Yes, sir. With the ruthenium ortho position activated, but in this our work we have activated C2 position of the thiophene. Further, to get the best reaction condition, we screen the reaction condition, and we find that C1 complex, tertiary butyl based complex with the 5% in the DMF solvent with the silver acetate, we get the better yield. The substrate scope is under process, And the DFT study is also in under process. So after that, we will give the plausible mechanism. Thank you, sir. And finally, I would like to thank my supervisor, my lab mates, funding agencies, and of course, the Rajasthan University to give this opportunity. Thank you, sir.
Thank you. Thank you. Maybe some words, some additional words concerning the mechanism, because you have still as a student, but what are the fundamental steps of your position if you are not sure?
Yes, sir. Sir, we are under process, but the main thing is, which is in my mind, that we will give to for the plausible mechanism, that palladium forms a intermediate in which this bond and this bond both are activated, which is as my topic is that hysterically favored, because this is the hysterically favored CH intermediate will form, and this bond will break. That's why we are activating this side, that this palladium and this carbon bond will be stable. So we are now under the DFT calculation. So as the my title that I already mentioned that it is the hysterically favored. Here I have already mentioned the hysterically favored selective CH equation. So we are based on the intermediate. Okay, sir.
Can you explain about the supramolecules? Because
yes, sir. The palladium complexes are generally used to form the supramolecular moieties by using the we can say the crown ether, etc.
how you can explain about supramolecules? Because supramolecules aspect related
Yes, sir. Yes, sir. Supramolecules generally have the
lock and key system.
Yes, sir.
You know about lock and key system, gas and Hall system.
No, sir. So we are not about the studying about supramolecular chemistry, but supramolecular chemistry generally magnetic bonds exist.
Supramolecules they are depends in three aspects:
Yes,
lock and key, gas and host, and systematically large molecules formed.
Yes, sir.
They may be hydrogen bonding. They may be van der Waals interactions. So your study, which is related to, I think, inclusion type compounds.
Yes, sir.
Inclusion type compounds are stereotype compounds are complex type compounds.
Yes, sir. But generally, we in our scope, generally we study about the supramolecular moieties which have mechanical bond like rotaxane and etc. types molecules. Okay, sir. Thank you, sir. Thank you.
Next, OP4.
Good morning, one and all. Myself, Biplab Bora, PhD scholar from Central University of Gujarat. Here I am going to present one of my work entitled Visible Light Induced Organophotocatalytic and Singlet Oxygen Initiated Domino Construction of 1,4-dihydropyridines, quaternary centered, CT functionalized spiroindolines, and C11 functionalized spiroindenoquinoxaline pyridines. First of all, why we have selected these compounds like 1,4-dihydropyrimidines, spiroxindols, and spiroindenoquinoxalines? Actually, according to literature reports, almost 60% of small organic drug molecules approved by FDA contain heterocyclic compounds as a key moiety in their structure. Here, among various nitrogen-containing heterocyclic compounds like 1,4-dihydropyridine and spiroxindols and quinoxaline molecules are different types of heterocyclic compounds having wide range of biological applications, and they also appear in natural products. Due to this importance, we have designed 3 different types of target molecules like 1,4-dihydropyridines, these spiroxindols, and spiroindenoquinoxalines embedded pyridine molecules. If we look at some background reports, Hansetz in 1882 first reports the synthesis of this 1,4-dihydropyridines, and this method was today considered as one of the best method in the synthesis of this 1,4-dihydropyridines. And after that, so many reports are developed for the synthesis of these 1,4-dihydropyridine type compounds. But development of one particular method for synthesis of three different types of molecule is highly challenging. Here, for the first time, we have designed visible light induced, singlet oxygen initiated, and single electron transfer process for domino construction of these three different types of molecules. And this is our overall work. And to achieve this success, first of all, we have optimized the reaction conditions. And for optimizing the reaction conditions, we have chosen aldehyde, this active methylene compound, DMAD, and aniline as the model reactions. And the reaction was carried out in absence and presence of different types of photocatalyst here. And this is the different types of photocatalyst. And after optimizing the reaction conditions, we have found that Rose Bengal was Rose Bengal was found as the best catalyst for these reactions. And again, we have optimized the different solvent system required for these reactions, and we found that water ethanol is the best solvent for these reactions. And again, we optimized the different atmosphere for these reactions, and we found that blue LED as the visible light source in presence of molecular oxygens or molecular oxygen provide the best yield of the products. And however, the reaction in absence of light failed to yield any product. And after optimizing the reaction conditions, we have synthesized different types of 1,4-dihydropyridine molecules, and in all the cases, we have obtained good to excellent yield of the products. And the structure of the product was confirmed from proton NMR, carbon 13 NMR, and HRMS analysis FTIR also. And after synthesize these compounds, we again synthesize different types of 1,4-dihydropyridine compounds where fluorine was attached to the 4 positions of these compounds. Actually, in recent year, the installation of fluorine either as a single molecule or as a substituted molecule has emerged as a key fascinating area in medicinal chemistry. That's why we are trying to synthesize some fluorine containing 1,4-dihydropyridine molecules under our visible light initiated approach. And after this, we have again replaced this aldehyde by isapine-like compounds. And in this case, the corresponding spiroxine compounds were obtained in very good to excellent yields. Then again, the structure of the product was confirmed in proton NMR, carbon 13, fluorine NMR in HRMS analysis. And for the first time, we have designed and developed high-order multi-component reactions for the synthesis of these fused quinoxalines and these spiral 1,4-dihydropyridine molecules from executing the reaction between neohyde green orthophenylene diamine and this active methylene compound DMAD and aniline by using this visible light induced Rose Bengal photocatalysis. And to validate the synthetic capacity of we have carried out different scale-up experiment, and up to 10 mole per millimole, we have synthesized this product in also in quantitative yield. And to propose the mechanism of our reactions, we have carried out different types of control experiments. Initially, the reaction between isapine active methylene compound, DMAD, and aniline, in presence of this radical scavenger like tempo, prohibited the reaction, and we obtain only trace amount of product due to the formation of this radical tempo radical product, and it was confirmed by HRMS analysis. And again, we have examined the involvement of the superoxide radical anion by using this parabenzoquinone in the model reactions, and in these reactions no effect was observed. Therefore, we proposed that no superoxide radical anion was involved in our work. And again, for identifying whether singlet oxygen is involved or not, we again used DevCo as a singlet oxygen scavenger, and in this case, we failed to isolate any product. Therefore, we suggested that the singlet oxygen was involved in our work. And again, the involvement of this radical pathway was confirmed by another radical scavenger like BHT. And again, for identifying the key intermediates involved in our reactions, we again carried out different types of control experiments. And after this, we proposed the mechanism which involves two mainly catalytic cycles. So the first one is energy transfer, where rose Bengal upon irradiate with visible light undergoes this excitation state, and this excited state rose Bengal transfer its energy to this molecular oxygen and thereby forming the singlet oxygen, and this singlet oxygen then abstracts a proton from this active methylene compound, as this was confirmed by HRMS analysis. And in the second catalytic cycle, rose Bengal was involved as an is in undergo singlet state, and through intra-system causing it underwent its triplet states, and then it involves in reductive quenching cycles and thereby forming this intermediate. And the final reaction between these two, intermediate 3 and intermediate 8, and followed by 6 exotic cyclization, produced our desired product. And also the sustainability of our work was determined by using various types of green chemistry metrics like atom economy, carbon efficiency, and atom efficiency, and all the calculated values are in agreement with the ideal value. And finally, we have developed the first rose Bengal photocatalyzed 1-pot domino as well as phase approach for the synthesis of 1,4-dihydropyridine, C2 functionalized spiroxyindols, and C11 functionalized novel spiroindenoquindoxylin pyridines in aqueous medium. And also, we have proved the installation of varied fluorine-containing coordination-centered compounds. And this work was published recently in Organic and Biomolecular Chemistry, a journal published by Royal Society of Chemistry. And I would like to acknowledge Central University of Gujarat for giving the infrastructure to carry out my work. And I also like to thank UGC India for the fellowship. And I would like to thank Central University of Tamil Nadu. And I would also like to thank all the FCASI 2023 team for giving me the opportunity to present my work. And I am also thankful to my supervisor, Dr. L. Raju Sohan sir, and my dean, Prakash Siza, and my faculty, Dr. Isoria Vegari, sir. Thank you.
Thank you. To confirm, to validate your mechanism, did you perform the DFT calculation, or are you going to?
No, actually, we are processing this DFT calculation and also crystal preparation and biological activity of these compounds.
Okay.
We are under progress.
Okay.
Just the experimental section we just published.
I didn't see as blank experiment. Did you perform some test or for the reaction of mechanistic test in the dark?
No, sir.
Okay.
You have different types of spiro complexes synthesized, different types of spiro complexes.
Yes.
And you have studied that in singlet condition, and you also studied in triplet condition also, singlet and triplet. So what is the better result observed in singlet condition or triplet condition?
Singlets.
Singlet condition.
Yes.
What is the basic reason in behind? What is the basic reason in behind?
Due to the because of the high singlet oxygen quantum yield of rose Bengal. That is particularly most important thing because we observed these excellent results. Number one thing is the high intersystem crossing quantum yield of rose Bengal, and second one is high singlet oxygen quantum yield of rose Bengal. That's it. Thank you, sir.
Good morning, everyone. This is Anvasita from BITS Pilani campus. And my topic for today's presentation is glycopyrosalamine derived copper metallopolymer synthesis, characterization, efficient absorbent for metal ion, and catalyst for 1-pot synthesis of hydropyrozoles. So actually our group is working with carbohydrates because generally our research will continue in two ways: whether you choose a problem and design your research accordingly, or whether you can design your molecule and find various applications. So our group is working in second category. So as carbohydrates is most renewable source in the earth crust, but still very small amount research is done because of its a large number of chiral centers, more number of OH group, due to which it has a multiple binding site for metal complexes. So this is the main problem when we are working with the carbohydrates, but it can be solved when solved by using this method. For example, it can be we can use the modified sugar by protection of out of 5 OH group, 1 or 2 OH group can be protected, and then we can modify it in accordingly, like NH2 or other functional group can be added. So previously, our group has designed this ligand, monodentate ligand, monotopic and tridentate ligand, and they have synthesized this copper complex. And as you can see in the structure, a dinuclear complex is formed. So we have some idea that, okay, if we design a ditopic ligand, like which has 2 binding sites, we can maybe a polymer or a morph-like structure is obtained. But in our case, we find that unfortunately, we are not able to get the morph, but we get the polymeric structure. So this is the method for synthesis. We started with this this benzene ring. Then after formylation, we have synthesized this ligand by condensation of this shift-based reaction, and this ditopic ligand is formed. And it was confirmed by HNMR, mass, and carbon-13 NMR. And after reacting with copper acetate for 3 hours in ethanol, we found that this is forming a polymeric structure, amorphous polymer structure, which is confirmed by XRD. So this is still we are working on it to find out what is the coordination number of this copper by using EXAPS analysis. So this is the same image, and in same image, like you can see, there is a continuous growth of the molecule. We do not find any small dot or any other. This is a continuous growth, and all copper, carbon, oxygen, and nitrogen are arranged homogeneously. So this is the thermal analysis. In thermal analysis as well, when we did this DTA, here this type of current is representing the glassy temperature, and thus glassy temperature is generally obtained when the molecule is polymeric in nature. And this one part is because of the removal of the coordination molecule. As we already know, I already told that we are not reached up to the what is the coordination number of this copper. So we are still looking into it. So, but this part is probably because of the removal of the coordinated water or any solvent molecule. Further, we did this because it is a polymer, and we did this BET analysis, and BET analysis, as you can see, this is a 4th type of adsorption curve. And 4th type of adsorption curve is for if the interaction between absorbent and absorber is weak, then we got this 4th type of adsorption curve. And for this type of curve, generally people go with the Lagrangian isotherm, but for this type of curve, BGH plot is better. And by using BGH plot, we find that there are 2 types of holes or pores are present. One is around 60 nanometer and second one is around 94 nanometer. And average pore diameter is 162 nanometer. So because the molecule is slightly porous, so we planned that can we study some adsorption studies. So at first we go for the metal ion adsorption. For this, we have prepared a synthetic solution of metal ion, and then we have stirred it for half an hour at RT, and then we filter this because polymer is not soluble. So after that, we did this ICP OES examination, and we found this result that our polymer is good for adsorption of chromium and barium. So we further move to the we optimize the condition for the absorption of chromium and barium. So these are some conditions where at first we find that what is the concentration better for the absorption. As you can see, we have tried with 5 to 300 ppm solution, and our polymer is working best for chromium. It is 200 ppm, and for barium it is 150 ppm. Then as for removal, as you can see, this is the percentage removal from water. It is decreasing with the increasing concentration. because maybe because the pore size has occupied by the metal ion, that's why it is decreasing. And then we study the pH. We started the studying pH studies. As for barium, the 7 pH is found to be the best for the absorption of barium. And at 10 ppm, the chromium ion was almost absorbed on the surface of the polymer. But after that, it is decreased, maybe because of OH ion is more, and it was removing the metal ion which is absorbed on the surface. Then we see the effect of the doses, that how much amount of our polymer is required. And for this, 1 gram per liter to 4 gram per liter, and 2 gram per liter was found to be the best condition. And after that, we have taken the contact time. In contact time, as you can see, within 20 minutes, the all metal ions, the absorption reach at its maximum. And this is the characterization before and after absorption. As you can see in this IR spectra, that this blue one is for the copper complex, and after that, it is decreased.
Please conclude.
Okay, sir. Because here, coagulation water or OH molecule is removed, so absorbed. And this is PXRD, this is amorphous, but after absorption, some peak is there because of the absorption atoms. This is XPS studies. In XPS study, we find that binding energy for copper is there, but after absorption, we also find this barium and for chromium. And this is, we are thinking that maybe this is the proposed way to absorption. Then we apply this for one-pot synthesis as well. So this is the mechanism for RT room temperature within 5 to 10 minutes. We have find this optimized condition, and under optimized condition, we have designed this 11 11 substrates. And this is the NMR report. This is for NH, and this is for this CH. And this is for NMR spectroscopy. And mechanism is under process. And finally, I would like to acknowledge my Professor Ajay Kumar Shah, for their continued guidance, Rajasthan University and FKC for giving me this opportunity to present my work here, faculty members, group members, research scholars, and BITS Filani for fellowship. Thank you.
My question is related your metallopolymer.
Yes, sir.
Synthesis characterization efficient adsorbent for metal ion. In case you selected 3 metals: copper, barium, and chromium.
Actually, we have selected
Copper, it has a binding energy particular.
Yes, sir.
And other barium and chromium, you have selected for adsorbent nature.
Sir, I'm sorry, but I was not getting your question.
My question is that you have selected 3 metals.
No, sir. Actually, I'm not selected 3 metals. We have selected zinc, arsenic, cadmium, lead, and this barium and chromium. But our polymers work best with chromium and barium.
Chromium and barium.
Yes, sir.
Okay. And copper?
Copper we did not, because we synthesized copper already. So it is not reasonable to use copper, I think.
Because your graphical presentation, there are in three types of metals.
Sir, one is metallopolymer. In metallopolymer, we have taken the copper already in the metal complexes. So that's why. Thank you, sir.
Thank you, participants. I just would like to request you all, please stick to the time. We are running short of time. And I also request the judges to please sum up. Let them speak for 4 minutes and give them 1 minute for your discussion, not more than that.
Sir, this is OP7. OP7. No, she will come.
A hearty good morning to all of you. Standing in front of you is Aditi Soni from MNIT Jaipur. giving my oral presentation entitled as Synthesis of Novel Ferrosinyl Compounds and Their Application in Optical Sensing. The work has been performed under the guidance of Dr. Rajkumar Joshi, MNIT, Jaipur. So after revealing the title, you all must be thinking that why our group is so focused on novel ferrosinyl compounds to synthesize them. The reason behind that is ferrocene is a very peculiar molecule because of its reactivity, which is similar to that of benzene. So ferrocene and its derivative have grown in different fields of chemistry such as organometallic chemistry, catalysis, biological applications, etc. However, literature rarely covers the reactivity of the this beta-chloroferrosinyl acrylaldehyde. Since it has a potent synthetic tool and can achieve various achievements in the future, we focus to synthesize the various derivatives of these compounds. So in continuation to this, we have synthesized the various dienols via the Heck reaction and various derivatives of vinyl ethers as well. These all compounds that we have synthesized are the novel ferrocene substituted vinyl ethers and dienols with high yield products and high tolerance towards the functional group. These methodologies accompany with the mild reaction and employ inert-free and solvent-free environment. Further, the application also that we find out that copper and metal ion sensing was performed further. So in going detail, this slide presents the NHC PD2 full pincer catalyzed Mizuruki Heck type cross coupling of vinyl chlorides and alkenes. As we know that Heck coupling is a renowned reaction, so we carried out the reaction of beta-chloroferrosinyl acrylaldehyde with the help of full pincer NHC PD2 catalyst. This catalyst is the smallest contribution to the scientific world from our lab only. So we derived different derivatives of styrenes and acrylates. It should be noted, I want to note you that our reaction was also very successful for the synthesis of the few internal alkenes, that is cyclohexadiene and cycloheptatriene as well, which is very difficult to couple with the traditional methods. Further, a facile synthesis of ferrocene functionalized vinyl ethers were performed, nitrile and aldehyde derivatives with the various derivatives of alcohols like aliphatic, aromatic, heteroaromatic, and diols. So here we have derived the ferrocene melanin nitrile derivatives. After keenly observing all these derivatives, you can see here that these derivatives have prolonged conjugation and have several binding sites. So we focused on this derivative after the CV studies and UV visible spectroscopy studies. Further, we performed the metal ion sensing with this moiety. You can clearly see that it gives a significant color from yellow to colorless. That is, it is significant only for copper 2 ion. It is 365 nanometer band was exhibited at the high energy band because of pi to pi star and L to pi star transition, and 521 nanometer band low energy band because of MSCT transition. Further, upon the addition of copper plus 2 ion, we can see the hypsochromic shift here. That is a shift, peak has been shifted to 336 nanometer and the low energy band has been disappeared. Further, we have carried out the interference studies as well. Upon the addition of the other competitive ions, no any color change was observed. That is, we can say that our compound is specific to the copper ion only. Further, we performed the BH plot to know the association constant, which comes out to be 1.95 and 10 to the power 5 molar inverse. And the smartphone-based sensing was performed. To provide the alternative pathway for the UV visible spectrometry, we have done the smartphone use. Because here we have performed RGB color app we have taken in the iPhone 7s, and the calibration curve was plotted between RG ratio and the copper ion ratio. And the LOD value that comes out to be is 2.16 to 10 to the power minus 5 molar. Further, to provide the analytical possibility, we have done the TLC silica strip test. We have soaked the TLC silica strip in the 3 different concentration of the copper ion. That is, you can see here B, C, and D. A is our standard, and with B and C, D, different concentration. There are different colors we can see here, which shows that our compound.
Please conclude.
Yes, sir. We can see that our compound is cost effective and portable for copper and naked ion sensing. So at summary, I can say that we have developed a methodology with a variety of primary alcohols and alkenes. Reaction conditions are mild, and selective optical sensing of copper ion was shown with the smartphone sensor as well, and having a lower limit of detection of copper +2 ion. I want to acknowledge my guide, Dr. Rajkumar Joshi, for his guidance, encouragement, motivation, and constant supervision. University of Rajasthan for providing a chance to present my research work in the FCASI 2023. I would like to extend my gratitude to CSIR UGC. I express my gratitude to MRC and MIT for providing the characterization facility, and I give my special thanks to my lab mates. Thank you.
Thank you.
Yes, sir.
Could you do the same type of chemistry starting from CP star or CP prime ferrocene with the same branch? Do you see what I mean? CP star?
CP star cyclopentadiene.
It means with a substituent of the cyclopentadiene, methyl substituents are starting compound. That could eventually modulate the optical sensing. You see what I mean? You start from your starting reagent. You are ferrocene, okay, with a branch. You could start from the same, but with methyl substituents on the cyclopentadiene. It's called CP star. Okay. No.
No, sir.
You don't say, okay.
We have done the ferrocene derivatives.
Okay.
Mainly focused on that only.
Okay. Just another quick question. These compounds you prepared, these ferrocene derivatives, could be also interesting in catalysis, in homogeneous catalysis.
I'm sorry, so can you?
The molecules you prepared could be also interesting in homogeneous catalysis, in organic synthesis, I mean.
These compounds that we have prepared can have wide application in the catalysis, in the biological application that we are carrying forward also within our laboratory only.
Thank you.
Okay, thank you.
You prepare ferrocyanide complexes. I think ferrocene, there are two types, staggered and eclipse. So which type of ferrocene have you synthesized with different derivatives?
So I think ferrocene is most stable in its staggered form.
Staggered form.
Yes,
okay.
sir. So these compounds are there in staggered form.
Okay.
Yes, sir.
No problem. Instead of taking the ferrocene, we have also done this reaction with phenyl. So similarly, CP cyclopentadiene can be taken. CP star can be taken, like we can do it in this way. It's a good suggestion to develop varieties of ligands, basically CP star based ligands. Yeah, it works. And that can be taken as a R group. Simply, it is ferrocene is working as a R group over here. So we can do it that this way. Thank you.
OP6 is there? No.
Okay, 8 months.
Good morning to all of you. Myself, Akanksha Yadav. I'm doing my research under supervision of Professor Nilima Gupta. And my topic is deactive investigation of 1,3 dipolar cycloadditions of thiazolium lilacs. So fused as a heterocyclic compounds have great importance in organic and medicinal chemistry. And we have some examples for by which we can synthesize the fused as a heterocyclic compounds by using isoquinoline, pyridine, imidazole, and thiazole. So for the synthesis, we use 1,3-dipolar cycloaddition reactions, which is a fissile route for the beachhead as a heterocyclic compounds. And these reactions have high stereoselectivity and regioselectivity. So for our interest is more in thiazolium lids, which have already literature reported. So first, in the presence of copper, by using nitroalkene and thiazole ylids, the 3+2 cycloaddition takes place and the cycloadate form. By using the leaving group, that is this one, 3+2 cycloaddition also takes place in acetylene derivative, and we got the in literature it is reported this cycloadate. We have more focused on the 3+2 cycloaddition reactions with the simple thiazolium lids. For example, in the third literature, it is reported that the other than 3+2 cycloadditions, hydroxy group also form, which is further the rearrangement in the particular reaction form. By using transition metal and transition metal series, 5,5 annihilation can also take place in the reaction. So by keeping the literature in mind, we have designed our route for the synthesis, and that is by the simple thiazolium lids. We have tried to do 3+2 cycloaddition on acetylene derivative. But the result were not as we expected. By wearing the substrate scope like different kind of R1 and the acetylene derivative, we were trying to synthesize the derivative 4, which was not obtained. And the reaction followed the different paths, that is this one. The one equivalent with the acetylene derivative, the 3+2 cycloaddition takes place, but this was not stable. And by the auto-aromatization, the product 6 formed. Sorry, not for the auto-aromatization, the 6 product formed due to the rearrangement. The ring is not so stable, so that it will react with the second equivalent of acetylene derivative, and we got the 7 and 8 geometrical isomers. We have proved it experimentally also by the 7 and 8 NMR spectra by varying the coupling constant. The main focus when we found the geometrical isomers is the coupling constant. So we observed the different coupling constant for the cis and trans. Now, what we got experimentally, we try to prove it computationally also. So the computational study, we use the Gaussian 16 software by using B3LYP 631 plus G star basis set. Reaction mechanism and ring opening reactions are studied, and the intermediate and transition states are optimized and analyzed. So this is the computational study that we have done for our calculations. This is our YLID, which is available in 2 isomers. This 1A is less stable and 1B is more stable. So the 1A, which is less stable, is reacting with the acetylene derivative, and by the transition state TS2, it is forming the cycloadate 3. This is the same cycloadate. The 2 hydrogen atoms are on the same plane. So it is more prone to give the auto-aromatization and giving the product number 4. On the other hand, when we are talking about the 1B, that is more stable as compared to 1A, it is reacting with the 2, that is ethyl propionate, and by the transition state TS3, it is forming the 5 that has 2 hydrogens which are not in the same plane. So the main difference between 5 and 3 is that these have syn hydrogens and these are anti-hydrogens. These syn hydrogens are more prone to give the aromatization, but the aromatization is not possible in this case. So the rings again opens up, the giving the moiety 6. That 6 moieties again reacting with the 2, that is 1 more equivalent of the ethyl propionate, and giving the product 7 and 8. These are the geometrical isomers. So what we have concluded is that we have 2 isomers by DFT calculations, and they are not interconvertible because the energy gap is significant. So the less stable YLID, that is 1A, is giving the product number 4, and the more stable YLID, which is the 1B, giving the 2 geometrical isomers 7 and 8 that we have concluded experimentally also. This is the last slide. Thank you.
Thank you. I didn't remember at which temperature you proceed your reaction.
Sorry, sir.
I didn't see at which temperature you are working to prepare because it's important for the kinetic.
Sir, our main title was on computational study, DFT investigations. So I didn't mention that, but we did.
Yes, to prepare the.
120 degrees Celsius. 120 degrees Celsius, sir.
Okay.
What is the basic reason that your stereo, as a particular stereochemistry, that this 718 compound is stable? 118 complex is stable. What is the basic reason?
For the stability of 7 and 8. Sir, the 7 and 8 is forming from the 6th, and 6th is anionic form. So that is not stable. So the sulfur anionic form is giving the nucleophilic addition reaction on the 2, that is ethyl propylate. So it giving the 7 and 8 product. That is geometrical isomer cis and trans. I'm getting that one. Thank you.
Just I
Yes.
complete my sorry.
Yes.
And working at a different temperature below or above 100 degrees C, did you observe the same ratio of product?
So the reaction did not proceed at temperature less than 120.
Okay.
And after 120 degrees Celsius, the polymer is obtained. Like we are using the dipolarophile, that is 2, that is polymerized itself in the higher temperature. So the reaction did not take place at high temperature also.
Thank you.
Thank you.
OP7. Seven. 6. Okay, OP6. Sir, OP6.
मैम इसको स्लाइड कैसे करना?
ये पॉइंटर है,
ओके
ठीक है, ये आगे पीछे करना है.
ओके. Hello. Good morning, everyone. I am Deepak Sharma, research scholar from MNIT Jaipur. Today, going to present our presentation on title is Iron Copper Bimetallic Catalytic System for amide synthesis via aminocarbonylation using MoCO6 as a surrogate of CO. Firstly, we start introduction of method of amidation. There are many methods for including conventional method and catalytic method. However, these methods have following drawbacks like stoichiometric byproducts, hazardous reagents, and a limited substrate scope. And these all methods are more than one step reaction. And recently, amidation was achieved by aminocarbonylation with the help of catalyst. The advantage of this method is use a large substrate scope, a greener solvent, mild condition, and as well as a one-step process. So, I want to grab your attention towards iron selenium carbonyl cluster having following significance like easily synthesized on large scale, highly stable, and unreactive towards air and moisture. So till now, with the help of this cluster, only 3 reports are published showing nitro reduction, CN coupling, as well as direct amidation. So in our group, we are exploring the catalytic activity of this cluster. So further, we try to carry out the aminocarbonylation with phenylboronic acid and nitrobenzene. After doing a literature survey, 2 research groups are working at this methodology for aminocarbonylation reaction. They use palladium and nickel PPS3 catalytic system. Since these previous protocols have required expensive metal or ligand or base as well. So in order to rectify the previous literature, we developed the iron selenium carbonyl cluster catalyzed aminocarbonylation. The advantage of our protocol is cheap and substantiable iron cluster, inert and base-free condition, and large substrate scope and synthesize a drug molecule. So this is the 1H NMR and carbon 13 NMR of representative compound, and the 165.8 ppm is the characteristic peak of carbonyl. So after doing optimization, we develop a library of compound with substitution phenylboronic acid and substituted nitrobenzene. So delightfully, we were also synthesize two antipyretic drug molecules, phenacetine and paracetamol, using our methodology, in a good yield. So after doing a literature survey, he proposed a mechanism, and firstly, transmetallation of phenylboronic acid with copper iodide give a phenyl copper complex A. This phenyl copper complex A converted into acyl complex after coordination and insertion of CO by MoCO6. And then in the presence, at the same time, in the presence of water, nitroarene is reduced by iron selenium carbonyl cluster and MoCO6 to an aromatic amines. This aromatic amines is a nucleophilic attack on acyl copper intermediate to release a desired product. So this is a conclusion of work. We developed a first time bimetallic system, iron and copper for aminocarbonyl reaction, and the highlighted point of work is the in situ reduction of nitrobenzene and synthesize aliphatic, aromatic, and cyclic and heterocyclic amides, and reaction work under base or inert free condition, and we synthesize a large substrate scope. So Akmal, thank you, our supervisor and lab mates, MNIT Jaipur, CSR UGC, MRC, MNIT, and University of Rajasthan. Thank you and thank you so much.
Thank you.
Yes, sir.
Why do you use one equivalent of manganese? Because you have 6 equivalent of 6, right?
Yes, sir.
Why do you use one equivalent of molybdenum? You have 6 equivalents of carbonyl, you need only 1.
1 equivalent of MoCO6?
Yes. You have 6 equivalent of carbonyl. Okay.
It's the source of CO, sir.
Did you test with 0.5 equivalent of carbonyl compound, for example, this carbonyl compound?
Sorry, sir.
You need, for example, 1 carbon monoxide for each reaction, for each catalytic cycle, right?
Yeah.
But you had 1 equivalent of your carbonyl precursor. And in this 1 equivalent, you have 6 equivalent of carbon monoxide.
Okay.
So you don't need formally to use 1 equivalent of this compound. You can use less. Did you test with less, for example, 0.5 equivalent?
Low yield. Yeah.
You don't need 1 equivalent. Maybe you need, I don't know.
Yes, sir. When we test a 0.5 equivalent of MoCO6, the yield of the reduce of the product.
Okay.
Yeah.
Did you test with iron pentacarbonyl, for example? With iron pentacarbonyl, 5 carbonyl. Did you test?
No, sir. I'm not getting the product. You can CO5.
Okay. For the mechanism, it's a proposition.
Yeah.
I think it should be checked because I am not sure of the first steps. Okay. Thank you.
Thank you so much, sir.
In your research title, iron deoxystate and copper.
Yeah.
Bimetallic catalytic.
Yeah.
Where is the complex? Where is the complex you have synthesized?
Complex?
Yes.
We can use cluster.
Carbonyl?
Carbonyl cluster, yes, sir.
Where is the complex? Iron 0- copper bimetallic catalytic system for amide synthesis via amino
Yes, sir.
using.
This is this one.
Where is copper?
So copper iodide.
Copper iodide.
Yeah. In your this one, see, this is work. Copper iodide and iron cluster different. That's why we tell bimetallic system.
The copper is a metal,
Yes, sir.
it should be indicated here.
Yeah.
It should be.
Yeah. Thank you.
Okay, I think OP9.
So good morning to everyone. So today my presentation is about nickel and palladium catalyzed denitrogative cross-coupling reaction of benzotriazole. And particularly I'm going to show you how to prepare auto-substituted benzotriazole. So we all know about triazoles and its application in various annihilation reaction, especially in the presence of metal complexes. A catalyst is well established. So what we would like to do is that cross-coupling reactions, which is costly study, and we choose benzotriazole because it forms a metallocycle intermediate and it easily can be activated by using a first order metal as a complex as a catalyst. So this is metallocycle, a well-known intermediate and could be useful for various annihilation and cross-coupling reaction. So thinking into the strategies, so we started this, and we all know that triazoles, when upon undergoing oxidative addition, it releases only nitrogen molecule as a sole byproduct. So in terms of green, is also much better compared to the traditional ortho-halo or pseudo-halide compounds. So why we choose benzotriazole is also questions, because we found some moiety, the core moiety presence in some pharmaceuticals and natural products, and that's the reason we choose benzotriazole to study. So we did many reactions. The one type of reaction is associative reactions, where we use nickel cord as a catalyst, tributylphosphine as a ligand, where we formed autoarylated and alkylated benzamides in good yields. The reaction also works well with N-arylated benzotriazole as well as N-alkylated benzotriazole. We also extended the scope of the reaction with palladium, because until then only nickel was used as a complex for such kind of benzotriazole reaction. Palladium was not employed. And we also know palladium is useful for various types of cross-coupling reaction. And that's the reason first time we came up with the palladium acetate and Zen-ox combination to form this metallocycle and study this R-related product. These two reactions actually complements each other. If you see the yields of alkyl withdrawing bromonic acids, it actually works, and some functional groups such as aldehydes and also the pairing type moiety successfully worked in this reaction. But one disadvantage is that limitation is that the reaction works only with N-aryl, not with N-alkyl. So this is the common mechanism that I can propose for both nickel and palladium, where it proceeds via a metallocycle intermediate and followed by transmethylation and reductive elimination can give the corresponding product. Next, we also intend to study the alkylation reaction, particularly Sonogashira type reactions, where we found some interesting observations. We observed three types of products. One is isolindenone, isobenzopyramone, and orthoalkylated benzamide. So when we use the aromatic thermal alkyne as the coupling partner in the presence of palladium acetate and xanthine combination, copper cyanide as a catalyst, we got isolindenone, isobenzopyramone. We can selectively get this product by fine-tuning the solvents. Whereas when you go from alkyl thermal alkyne, so we observed orthoalkylated products in good yield. So we believe that whether it is an indenone or isobenzopyramone, the alkylated benzamide could be the intermediate. So this is the mechanism we proposed as per the traditional known way. And we thought if R is an activated one, like with aromatic thermal alkyne, we may get this cyclized product. If it is an alkyl group, we won't get. So in order to confirm whether the reaction proceeds via such kind of intermediate, so we did a lot of mechanistic studies just also to understand so role of copper in this reaction. So we tested the reaction in the absence of palladium sunbox or in the presence of presence of copper cyanide, where you can see we get the cyclized product but not at high yield. When we put these two combinations together, we obtain the corresponding products in good yields. So these are some substrate scopes where I demonstrated with aromatic thermal alkyne in the isoindenone synthesis, and this is with aliphatic thermal alkynes to our alkylated product.
And these are the scope with, for isobenzofuran. So these works are done by my PhD student, Dr. Hari Balakrishnan, under this PhD topics. So I thank SRM University, Andhra Pradesh, and as well as DST, Government of India, for the financial support. Thank you so much.
Thank you. It was a little bit quick, but when you use your co-catalytic system, not co-catalytic, when you use palladium and the copper, there is an effect of a solvent from DMSO to DMF or something like that.
Yes, solvent.
Can you explain that?
Yeah. So actually, the exact reasons we also don't know, but we feel that could be the solvent polarity or as well as the solubility of the copper salts, and also the only reason. Because generally this kind of cyclizations happens with the silver salt type, but this is the first time where in situ we can prepare this kind of reaction.
And if I remember the first slide, there is a nickel with a palladium tributylphosphine, right? Nickel, sorry, with tributylphosphine, if I remember.
Yes.
Well, is tributyl donating phosphine necessary or not?
It cannot be, because we found the reaction conditions with tributylphosphine ligand, but Professor Cheng also came up with the triphenylphosphine under a different system. So our systems combinations with this also works, with the triphenylphosphine also works.
Okay. Thank you.
Thank you so much.
यहाँ पे करो लेकिन भी कम करो
So good morning to all of you. First of all, I would like to thank the organizing committee of University of Rajasthan, and a special thanks to Dr. Satpal Singh for giving me the opportunity to present my work. So I will be giving a short oral presentation on diastereoselective allylation in the total synthesis of paragonic acids. So if you ask what is paragonic acid, this has a common core structure which has a common carboxylic acid group, and these are differentiated depending on various alkyl group and whether there is a methyl or methylene group attached to the carbon number 4. So these are isolated from the moss and lichens, and these have a excellent biological activity. So these are few representative structures of the paragonic acids which are differentiated depending on the methyl or methylene group attached to the carbon number 4. And also these are subdivided depending on whether the carbon number 2 and 3 have a syn geometry or an anti-geometry. So in my work, I'm going to focus on this murolic acid and protolecithrinic acid, which we have synthesized. So coming to the retrosynthetic approach, the murolic acid, which we have already synthesized in our lab, the first synthesis was done in 17 steps. And the protolecithrinic acid, whose synthesis was, few reports are there, in 8 to 12 steps they have completed. So we have developed a divergent synthetic approach so that we prepared these two of the molecules through this common intermediate 14 through cross metathesis, and this intermediate 14 could be accessed from compound number 13, which is the allyl bromolactone, through the key reaction Barbier type allylation followed by translactonization, and then the compound 3 could be accessed from this commercially available cheap compound D-mannitol. So coming to the forward synthesis, we have taken D-mannitol and we have done the acetonide protection, and then we have done the oxidative cleavage and followed by the Bailey-Shulman reaction with the methyl acrylate. It will give you the alpha beta unsaturated acid, and then the cyclization is done in presence of HBr to give this allyl bromolactone 13. Then the compound 13 is treated with the aldehyde B, which could be prepared from this undecenol, which is commercially available. Then we have done the key reaction, which is the Barbier type allylation, and then we got the compound number 16. Then compound number 16 is converted to 14 through the translactonization, and this 14 is the common intermediate through which we have synthesized both the molecules. So by taking this intermediate 14,
we have done the cross metathesis with the pentene. So we got the compound 18. Then to selectively reduce this particular double bond, we have tried various conditions, but in all the conditions, I was getting either starting material recovered or the complete reduced product. So we thought to mask the exocyclic double bond followed by H2PD reduction of this long chain alkyl and DBO elimination to give the exocyclic, the required alkene compound 20. Then we have done the Jones oxidation to give the desired first natural product, total-echesternic acid. Then using the same intermediate 14, we have done the other natural product synthesis also. So we have taken this alcohol for the cross metathesis, which we have prepared individually using this chiral epoxide through the Grignard reaction. Then we got the compound 23. Then following the same pathway, that is the masking of this exocyclic double bond followed by reduction and unmasking to in presence of the DBU, we got the compound 25. There is only one step left, so that I will be converting this diol to acid to complete the synthesis of the myrrolic acid. So in conclusion, we have developed a divergent synthesis of protolechesternic acid and the myrrolic acid. And the first synthesis I have completed in 11 steps with 3.2% overall yield. So at the end, I would like to thank my supervisor, Rodney Fernandez, for the constant motivation and support, and Department of Chemistry, IIT Bombay, and UGC for the fellowship, and the organizing committee of University of Rajasthan. Thank you.
Thank you. And for this last step, what are you going to do to get the myrrolic acid?
Sir,
What do you plan to do?
for the last step, I am thinking to do the oxidative cleavage of this diol. So I will get an aldehyde, which I will do the pinic oxidation for to convert it to acid.
Okay.
OP11.
So good morning all. My name is Mahesh, and I would like to present the work on computational studies on designing and development of non-fluorine acceptors for organic solar cell applications. So over the period, there are many conjugated polymers and small non-fluorine acceptors has designed for organic solar cell applications. And you can see the evolution has started from simple polythiophene backbone to complex donor acceptor type polymers. And the other hand, we have several types of acceptors which are based upon fullerenes and fullerene derivatives. And recently, the new class of non-fluorine acceptors has developed to use as electron acceptor materials. So the work we have done is design new non-fluorine acceptors which show high performance compared to the existing materials. So the architecture of the non-fluorine acceptors currently follow this architecture. Basically, the core has electron rich unit which is capped with a 2 electron deficient unit. The choice of these units control the absorption properties which complements the polymer units. So we have considered quite a number of variety of core units and end caps. And we have studied the all electronic structure properties of these molecules. And by considering all these probabilities, we got more than 100 molecules. And we fully studied the properties of these molecules. And we explored all sort of properties which are related to the organic solar cell performance. And the question is how to choose the performing materials among these materials. So the major problem in the case of organic solar cells phases is how to choose the right combination of donor and acceptor so that we can avoid the energy losses and the charge recombinations. So for that, we have come up with a simple selection protocols. These protocols are designed based upon act as a descriptors which can act as a performance related stuff. So we have considered these selection parameters, rather we can consider as a descriptors to choose the high performing materials. So over around 120 molecules, we can choose 4 potential molecules as the non-fluorine acceptors. So we have studied the model in organic interfaces where what would happen during the device operation. So the molecular orbitals that you are seeing is actually a states called charge transfer states, in which after the polymer absorbs the light, it will transfer the electrons through photo-induced electron transfer. So the hole will be on the polymer donor and electron will be on the acceptor. So these studies confirms that all these four molecules indeed form a charge transfer state, so that we can form a whole electron pair, which is called exciton, so which will dissociate and generate the charges. So in addition, what we have done, we have characterized the distance between the center of hole and center of the electron, so that we can see where the exciton can dissociate much faster. So since in this case we have a larger separation between hole and electron, the probability to dissociate them is very high. So we have developed a simple protocols to choose the right combination of donor and acceptors, and we have also studied many other molecular properties of these non-foreign acceptors, and these are some of the publications that we have obtained from all these characterization studies. So this is our beautiful complex. I would like to thank my PhD students who carried out the work, and the funding agencies for providing the necessary support to carry out the research work. And I would like to thank the conveners for providing the opportunity to talk our research work. Thank you.
Thank you. Did you compare this non-fullerene
acceptors
with, for example, tetratiafluorene? It's not really my but
and there are no reaction mechanisms here. Basically, since the lack of time, I didn't show the full results. We always compare with the experimental results to cross verify our results are really meaningful or not. And whatever the calculations that we have done indeed shows that these are much better than fullerene and fullerene derivatives. And synthetically, these are more easier to synthesize than the fullerene and fullerene derivatives. Yeah.
Okay. Have you synthesized organic solar cell?
No, we don't synthesize any. I mean, we don't do experimental work.
Solar cell?
Yeah.
So I want to know that what is the storage capacity of your cell?
No, it's not storage, sir. It's a energy conversion device. It absorbs the sunlight, and the sunlight will be converted as electricity.
Electricity. It is electric power which is generated by subsequent.
So it's an energy converter device. So it absorbs the sunlight and convert as electricity. That's it. It cannot store the electricity.
Okay. The performance of the time period.
Yeah. So the performance, that is how it's a figure of merit, which is expressed as a power conversion efficiency. The current power conversion efficiency is 18%. 18%, which is much lower than the inorganic based one. So that is why a lot of efforts are going on to improve the power conversion efficiency of these materials.
So the conversion efficiency which is related to power point of photocurrent.
No, it is the power conversion efficiency depends upon how well these molecules absorb the sunlight, and after the absorption life, it has to convert as a excitons, which is basically whole electron, and after conversion again they should not combine again, which is called recombination. It has to separate efficiently so that the whole electron act as a charges. Which is basically a current. So it has a lot of
basically current which is depend on the power point of photocurrent, power point of photopotential.
Yes, absorption, everything. Yeah.
Okay. Yeah. Good work.
Thank you.
This one, right?
Okay. Good morning to all. I am Sachin Sharma, under the supervision of Dr. Ishwar Srinivasan from Central University of Rajasthan. Today, I am going to discuss the topic, the synthetic route to fluorescent dihydropyrazoles and pyrazole from the MVH ketones. So the pyrrolizole and pyrrolizolines, we are very well known compounds as we know. So let's start with the Morita-Billy-Silman reaction. This reaction is a cis bond formation reaction between the activated alkene and the electrophilic carbonyl center in the presence of tertiary base. These reactions are 100% atom economic, milder reaction conditions, and gives a multiple transformation of ambient adducts. Some of the important transformation of these ambient adducts include oxygen compounds, nitrogen compounds, and miscellaneous type of compounds. Some of the important transformation in the respect of reaction includes the annulation reaction, allylic alkylation reaction, Friedel-Crafts reaction, and the most important which I am going to talk and focus is on the oxidation of the ambient adducts that we call as ambient ketone. Talking about the transformation of ambient ketone, ambient adduct can be easily transformed using IBX without any column chromatography to get the yield up to 100% of ambient ketones. These ambient ketones in the literature are not well discussed when we came into this area. These ambient ketone can undergo Diels-Alder reactions, can give 1,3-vicinal dicarbonyl compounds and all. The simplest way what we came with the idea to explore this ambient ketone was to have the pyrozoline compounds with the reaction of this phenyl hydrazine that can be cyclized by using a Michael reaction followed by the condensation. But in the midst of the study, what we see is a very uncertain results. Just using the base, simple base triethylamine, we get the benzohydrazides. And this paper was published in Holland in 2019. Further going ahead, we found another interesting result, and which was you can see here. Today, I am going to focus on the work which was left behind and we carried forward in this area, that is the synthesis of fluorescent dihydropyrazoles and pyrozoles. In the literature, we know this pyrozoles and pyrrolizolines are very common and can be easily synthesized from we can say the chalcones, and even number of methods are there. One of the methods we have attained and want to explore the ambient ketone. So we took this opportunity, and we see these compounds are highly of biological importance. So we went for the optimization studies by varying the base and the solvents, and we got the NP2 using acetonitrile, and with the 15 hours of time, we got the 83% yield that we take as the best optimal reaction condition for the substrate scope, where we in this for which we choose different phenyl hydrazines like parachloro, paramethyl, and aliphatic also, that is tertiary, where we use the different ambient ketones like halo substitutor, electron withdrawing, electron donating, meta substitutor, and heterolide. We've got good to excellent yield with the photo example up to 97% of yields. Further, these pyrrolizolines can be transformed with the two pyrozoles. In the reaction, we have seen the conversion, so we were interested to convert it into the pyrozoles. But normally it is not very favorable without substrate to get it converted into fully to pyrozoles. So we took the stoichiometry amount of PDC acetic acid, and we are able to convert our derivatives to the this pyrozoles, and we got with the different similar substituent, we have prepared 24 examples up to 99% yields, and without using any column chromatography, we are get to other pyrozoles. In this, the proposed mechanism behind this was using a phenyl hydrazine with the ambient ketone, and it is going to be with the Michael fashion to get the intermediate one. This intermediate one is getting interconverted to the hemiaminal form cycling, and with the loss of water molecule, we are getting our desired product 3A. These intermediate 1 and 2 were characterized through the NMS spectroscopy within the reaction, taking the aliquots of the reaction. So we further to get into the insights of this
steam, we have tried this gram scale reaction, and the gram scale reaction also was good, and we got the 79% yield. Further, as I mentioned that the compounds were fluorescent, we were interested to calculate its quantum yields, how fluorescent they are. So the compound having the tertiary group, they are non-fluorescent and they are not having any fluorescent properties. And the compound number 7 has has shown the best photophysical properties. Thank you. I would like to acknowledge my supervisor, Dr. Iswar Srinivasan, and Central University of Rajasthan for giving financial support, lab members, and to University of Rajasthan for organizing FCASI and giving me opportunity for the presentation. Thank you. That's all. Okay.
Sorry. Just Aripiprazole.
Sorry, it's okay. No need to sorry.
Aripiprazole, a lot of drugs have this motif, aripiprazole, and I say that there are a lot of aryl derivatives via the CC bond formations. So, did you try to synthesize drugs, well-known drugs, with your method?
But we are trying to do the biological studies of dihydropyrazoles, especially on the Alzheimer's diseases. So these are under the clinical trials, and surely we will get some positive results on the dihydropyrazoles.
Thank you.
Protein is there.
Good morning, everyone. I am J.S. Maru, faculty from Department of Chemistry, Gujarat University. Today, I would like to present the work on coumarin functionalized dimer with alkyl arms and investigations of liquid crystalline property and anticoagulant activity. Coumarin, a diverse pharmacophore, an active pharmacophore which is present in both synthetic and natural compounds. An excellent retrocyclic compound, it is known for its anticoagulant activities. Some marketed available compounds such as warfarin and dicumarol are used as an anticoagulant agent for the treatment of blood coagulation study. So, by considering the tremendous potential of coumarin derivative synthesis in anticoagulant activity field and significant liquid crystalline property, we have synthesized some coumarin-based analogs. So the synthesis of A1 to A6 mainly divided into two steps. First step is we prepared the dicumarol by reacting 2-oxo-4-hydroxy coumarin with formaldehyde. For the electrophilic transformations, dehydrolysis reactions was performed at 130 degrees centigrade for 3 hours. And in next step, various alkyl bromide were then treated with dicumarol to get the targeted compound A1 to A6. Okay, these are the structure of compound A1 to A6. All synthesized compounds were characterized through 1H-NMR, 13C-NMR, IR spectroscopy, and through mass spectrometry. So these are the spectral data of dicumarol compound 2, and these are the spectral data of compound number A3. And to check the liquid crystalline property, we have employed all the synthesized compound for liquid crystalline property. So all the synthesized compounds were displayed mesophase at various mesophase on cooling conditions. So these are the photographs of the various mesophase of synthesized compound A3, and these are the various mesophases for compound for at room temperature on cooling conditions. So all the compounds were displayed various mesophase on cooling condition at room temperature. XRD, DSC, TGM were employed to assess the thermal and liquid crystalline property of the synthesized compounds. So the conclusion based on the liquid crystalline property, compound with lower alkoxy side chain did not display liquid crystalline property, whereas compound with middle to higher alkoxy side chain displayed mimetic to smectic C-type liquid crystalline property. And additionally, we have performed the anticoagulant study to check the potential of the compound for the treatment of blood coagulating agent. So out of 6 compound, one of the compound A5 demonstrated promising activity, Barber agent, anticoagulant agent. Even 10 mg sample or 5 ml of blood are potent to inhibit the blood coagulation. So for the bioanalysis of the blood, we can replace the K3 EDTA with our sample. So the conclusion based on all the study, we have provided the protocol by which we can synthesize the various coumarin-based analogs with higher alkoxy side chain, resulting in room temperature liquid crystalline property may promising for industrial applications. And our work was recently recognized by the Journal of Molecular Structure. I would like to thank our HOD, Pranav Srivastav sir, and organizing committee for providing me a chance to speak, to present my work. Thank you very much.
Thank you. Do you have an explanation for the efficiency or, for example, E5 as anticoagulant and a relation with the structure of this compound? A relation between its structure and this efficiency as anticoagulant? Do you have an idea why?
Sir, pardon?
Do you have, if I remember, here, you have a E5, and you explain us that it's really efficient anticoagulant, right?
Yes, sir.
Do you have, is there any relation between, is there a relation, or do you understand the relation between the structure and the anticoagulant effect?
Oh, no, sir. Still, we are in the progress to identify the correlation between the various structure. For the preliminary work, we have synthesized limited compound to check the liquid crystalline property and anticoagulant study. In next step, we have check the correlation between the various compound and what is the chemistry of what is the morphology of the compound to treat the anticoagulant activity. We have not correlate the structure of the compound with anticoagulant property.
Thank you.
Okay, thank you, sir. Okay, thank you very much.
Do we have another? Okay. So I believe all the presentations of this sessions is over. Thank you, Professor Mark Telefer and Dr. R.K. Gunsaria. I request Professor T. Punyamurthy, who have joined us here from IIT Guwahati, to please come over the stage and present a memento to Professor Mark Telefer for his valuable time.
So,
Professor Mark Telefer, please come. I also request Professor T. Punyamurthy to present a token of appreciation to Dr. R.K. Gunsaria for his kind cooperation and coordination of this session. Once again, thank you, Professor Mark Jalifer and Professor R.K. Gunsaria. And ladies and gentlemen, now this is the time for the second session of today's. I take my pleasure in introducing our next chairperson of this session, Professor Akhil Kumar Sahu. Professor Akhil Sahu is a postdoctoral fellow. He did his postdoctoral fellowship from Kyoto University, Japan. He has been a postdoctoral fellow at James Pierce, Kyoto University, Japan, with RWTH in Germany. He did his PhD from National Chemical Laboratory, Pune, India. Currently, he is working in the University of Hyderabad. He has published over 90 papers. He has got 7 international patents. He has delivered lectures over 70 seminars in the national symposiums in India. More than 50s of invited lectures in the international conferences. And I can keep on reading his resume. He has got a big list of achievements. So instead of wasting and taking much of the time, I hand over this session to him.
Good morning to all of you. Good morning, Professor Lee. How are you? Is it audible?
Yeah, fine. Thank you.
Oh, that's fine. We miss you here. Last time we had a good time.
Yeah, sure.
Okay, so it's a great pleasure for the organizing committee and organizer to give me opportunity to chair the session, and I'm a very pleasure of inviting Professor Lee for this lecture. The first invited plenary lecture session in this today's second of the seminar. He is very well known in organosulfur chemistry and selenium chemistry. And without talking much of this one, the good thing of this, the organizer, Professor Satpal's worked with Professor Lee and learned many good things, and he is continuing of doing a lot of cis carbon sulfur bond forming reactions. So thank you, Professor Lee, for giving good training to Professor Satpal. And with these words, I would like to invite you to showcase your work to us. Thank you.
Yeah, thank you so much, Professor Sahu, for your kind introduction. So, yeah, thank you so much for the organizing committee for giving me this opportunity to share everyone our recent research. So I will focus on more sulfur chemistry today. So next, the conference in 2018, I joined this conference in India. So yeah, everything is perfect. It's not only food and also wine, and the most important thing is the friendship between Jimbali and also my friends in India, especially the Professor Basana. So thank you so much to invite me again to talk about our chemistry. So my group started from 2008, and so we focused on more sulfur chemistry, but not only sulfur chemistry, also we did some different area. For example, recently we reported the functional polymer and application for the gene editing projects, and we published a paper 2 years ago in small, and also some interesting topics. And as Professor Sahu just introduced, we did some sulfur chemistry, and including the synthesis of siloesters, siloesters, and not only the CS bond coupling, and also but also SP bond formation and carbon oxygen bond and CN bond, CP bond, carbon bond, and also PM bond. Because of the time limitation today, I would like to talk about two works in cycle ester synthesis and also more sulfur phosphorus bond formation. So the project one, the blue LED promoting the synthesis of dicyl esters and also oxacyl acetals, and also the blue LED can be used for the synthesis of cycle ester with very, very mild conditions. And also the NCS promoted the synthesis of actually such much is the synthesis of phosphorus. We used a different approach. The first example reported in Green Chemistry in 2014, many years ago, and also in 2019, we can carry out a reaction on the full electrochemical approach. And recently, we report a paper in ChemCom by using. Yes.
Can you make it to your this one slides more full screen so that it will be visible nicely?
You mean the slide is not green?
No, it is just this one. You just click that below there is a desktop symbol is there. So that if you click it, it will be like a slide mode.
Okay, so how can I do now? So how about this?
Yeah, Professor Sattal here. Yeah, you make it slide mode now. That umbrella sign downside. Otherwise, press F5. F5 on your computer.
And how about like this?
You make in presentation mode. In your whole screen, the presentation should be there.
Okay, so how about this?
No, your slides are perfect. Only thing is that you see side by you can see all the slides now. You have to remove that and you have to see only one slide. So you make it in presentation mode. On your screen, downside, right side down, there is a last, I think last one, last tab. Volume.
So how about this?
No, what we are saying, what I'm saying is that, see, in your on your screen, you are seeing all your other slides. I know.
So,
everything.
Professor Sahu, can you hear me?
Yeah.
Hello, can you hear me?
Yeah. We are able to hear you.
Okay.
Okay.
So, what's
you press F5 button, F5 tab. Okay, no problem. Continue. You continue, please. Professor Lee.
Okay, so
continue. It's okay.
Yes.
You continue. No problem. You continue.
Okay, but I think the slide didn't change from the conference room.
Okay. Yeah.
But I can handle my slide. But how about in the meeting room? Hello.
Professor Lee, whatever you are, you are just looking in your desktop. That screen is here. Yeah, now it's okay.
Yeah, now I can share the slide now.
Yeah, now it's okay. Yeah.
Now it's okay. Okay.
Very good.
So
you click last, just now you have taken this one pointer.
Okay, pointer, how to use
Yeah, last one.
Let me see.
Okay, then carry on, no problem.
Okay, okay.
Yeah,
So, so next slide. Okay, so let me. How about a full screen? I cannot use a full screen.
रेखा भेजा क्या आपने?
Okay.
Let it go. Roughly,
Yes.
Rekha sent a message to you. You follow that one on your phone. Line. Rekha sent a message to you.
That's the point.
Where's your phone? Line. Rekha sent a message to you online. Follow that instructions.
So how about now?
It's good. Please continue. It's good. It's good.
Okay.
Yeah.
But it's not consistent.
You start from slide number 2. Start from slide 2. See, he's not
from this line, application of thiophosphate.
You go to your first slide.
This.
He's not able to move for the next slide. So your file is PDF file or what file? This PowerPoint presentation, right? PPT? That is in Chinese.
Ladies and gentlemen, probably this is the drawback of having, the digitization, but yes, we are in the position to connect to him.
No.
So for this technical.
Will get distracted. Absolutely, you can start from the left side button, like you click on slide number 1, then use any arrow to go down. One by one slide you can speak.
Yeah, but can you use my slide in a conference room?
Yeah, we are able to see your slide perfectly. You can talk now, no problem. We are able to see everything on your slides, no problem. You can continue.
So I can continue my talk. Okay.
Can continue.
So in the first part, we start from the aldehyde and react with thiol to equivalent of thiol, then in the presence of using Y as the photocatalyst. On the blue lights, then 12 hours, we can obtain a diethyl ester in very good chemical yield. And actually, the synthesis of a diethyl ester in the literature, the Lewis acid is quite common for the synthesis of a diethyl ester, but we can use the very simple approach and very green approach by using blue lights to carry out the reaction. Only 0.1 more equivalent of the using Y as the catalyst. Then 99% of the product was obtained in very good yield. Also, we can try the different substitution in the benzene, the benzaldehyde. As you can see, the chloro and bromo and even iodo, and also electron withdrawing electron donating group in the benzene and diethyl esters can also be obtained in good chemical yield. So we also not only the alkyl thiol, but also the cyclic thiols can be used for the synthesis of diethyl ester. But the low yield was obtained when phenyl thiol was used as the starting material. So also we can form the cyclic diethyl ester with the good chemical yield. And in this system, we can carry our reaction in a grand scale synthesis of diethyl ester. As you can see, 49 grams of the diethyl ester was obtained by using this simple approach. Interestingly, when aldehyde was changed, react with the ketone. When ketone was used for this chemical transformation, no product was obtained. So we are thinking about another functionality is oxacyl-acetals. So in the literature, Lewis acid as a catalyst is necessary for the formation of oxacyl-acetals. And in this case, we're just thinking about how about photochemistry in the synthesis of oxacyl-acetals. So in this case, acetophenone was used as a starting material and reacted with the alcohol cycle. Then the product oxacyl-acetate was obtained in good years by using a 2% more of eosin Y as the photo catalyst. It's a very simple reaction, a very green approach. And also we tried different substitutes, different substrates, and the product was obtained, but the chemical yield is not very good. It's about 40 to 60%. So another project is light-induced the synthesis of cyanoether from aniline. Actually, aniline, in this case, aniline reacted with the tert-butyl nitride to generate the benzene radical, then reacted with cyano to get the cyanoether. So in the literature, the synthesis of cyanoether is well known and starting from aniline. So there are two cases here. We are thinking about is possible to use aniline react with cyphenol in the presence of tert-butyl nitride. Yeah, to our surprise, using Y as the catalyst, photocatalyst, only 2 hours at room temperature, then we can synthesize the cyanoether in very good chemical yield. Interestingly, when the reaction was carried out without photocatalysts, then 90% of the isolated yield was obtained. So there are some examples of the cyanoethers, not only electron donating group, electron withdrawing group, but also in the presence of a very bulky cyano substitution, then we can get the cyanoether in good chemical yield. Not only the cyanoether, but also selenium ether can also obtained in very, very simple approach without photocatalyst, without light. Only 1 hour at room temperature, 96% of the selenium ether can be obtained. And also many functional groups, including very bulky substitution, and also iodo, bromo, nitro can also be tolerated as the starting material. And another approach for the synthesis of the thiophosphates, we carry out a reaction by using electrochemistry. Actually, the background show you the synthesis of PS bond formation is well known in the literature by using nickel catalysts. And another interesting paper was reported by Professor Barsana in 2019 by using the silica gel promoted the synthesis, not only the PS bond, but also the P-selenium and P-telluride bond. I think it's a very interesting and very useful approach for the synthesis of such molecules. And our approach, actually, in 2012, we reported the synthesis of cyanoether by using the NCX promoted reaction. First, cyano reacted with NCS, then followed by reaction with Greenland reagents to generate ether. We are thinking about how about trapped by pH bond. To our surprise, we can synthesize such molecule in the formation of the pH bond, and we reported this paper in green chemistry many years ago. So we are thinking about how about electrochemistry. So in this case, we use the protein line, then we can obtain the pH bond formation. So many substrates are including hinder thiophenol in the meso, and also electron donating group, electron withdrawing group in the benylene of the thiophenol. Then we can obtain the such molecule pH bond formation. And also only the butyl in the oxygen, and also the azo group, and also a phenyl group on nitrogen, then we can obtain the pH bond formation. And also last year we reported the paper in ChemCon, and it's quite simple formation of pH bond. In this case, only cysteine carbonates was used as the promoter, 40% reaction, then pH bond was obtained in good chemical yield. It involves the chemo selectivity because the P was attacked by sulfur, not attacked by carbon. So in this case, we can obtain the pH bond formation. So there are many examples here. Then first, we prepare the cell ketone. In this case, just ketone react with Lawson reagents to generate the cell ketone. The procedure is quite simple from the literature. So first, we try the base and also solvent, temperature, and reaction time. The optimized conditions is use cysteine carbonates and acetate as a solvent. Then we can obtain the perla in 85% chemical yield. So based on this condition, then we carry out many examples of the thioketone and react with in this reaction, the product was obtained in good chemical yield. Not only the electron donating group but also the electron withdrawing group in the thioketone. And the X-ray shows the structure of the 3P. We also carry out a grain scale synthesis of this pH bond product. And also the comparative experiments was carried out in the presence of a ketone and thioketone. And in this case, only the pH bond was obtained in this case. So that means the nucleophilicity of the sulfur is much better than oxygen. So we also did some phosphorus and NMR to demonstrate the formation of the pH bond. And we also did some calculation and also to support our reaction profile. And the last part is the blue LED promoted synthesis. And in this case, I think, how about the time of my talk? So in this case, we can use the X is equal to oxygen or sulfur, then react with diphenyl disulfide, then in the presence of the blue LED at room temperature, then the pH bond was obtained in very good chemical yield. And this slide shows you the scope of the substrate. Then not only the benzene aerial cycle, but also the aqueous cycle can also obtain, for example, the compound 5F. Then we can obtain the product in 84% yield. And in this slide show you the some compound with bioactivity can be obtained by using this simple approach. And not only the PS bond formation, but also P selenium bond can also be obtained, and also the P telluride bond can also be formed. And we also did some control experiments, and the mechanism is show you here. It's quite simple. In the presence of the blue LED, then the disulfide will generate the radical, and the radical species that react with the pitch bond to form the final product. The last part of my talk will be the blue LED promoted the synthesis of the aerial azo phosphine oxide and phosphonates via NP bond formation. And in this case, start with aniline and also the isoamyl nitrite. To our surprise. What?
Yeah, you are unable to see your slides moving. It's the same slide. Can you share the slide? You have not shared the slide to this.
Yeah, so still could not share my slide.
Yeah.
Okay, so how about the time of my talk? I think I can stop my talk now.
You have 10 minutes, but if you just share the slide, it would be better.
Ask him to.
Firstly, open your student is there outside, they will help you. Open your office door. Your student is there. Master student is there in your office, they will help you.
Yeah, his question is but I think the time is not allowing me to share my talk, or I email you. How about I email you my slide?
No, you continue there, but you use the left side you are saying, yeah, like that you make it less. Yes, there is a moment. Yeah, like this you do. Next, like that.
Very strange. Hey?
You go for next slide.
Yeah, next slide.
Yeah, how you are doing? Yes, like this use. Yes, that's good. Like this you start. Yes, now you are able to. Yes. Like this you do. Yes, now, yes, that's correct. Click there only. Click there.
So now it works.
You click the button, like you just click.
So, and
it's okay. You go for the next slide, like you did last time. Same way you do. Go for the next slide and continue your talk. You have 5 to 7 minutes. Go one by one. Next slide and then continue speaking. Okay.
Okay. So the aniline can react with the phosphonates. Then we can obtain the interesting P dinitrogen bond formation of the product. So I think it's the first example of this approach by using LED to synthesize such molecules. So yeah, and also we did some control experiments, so we can demonstrate the reaction was performed under the theoretical the synthesis of this approach through a radical approach. So the conclusion is the synthesis of cell ether, we can carry out a reaction by using a LED promoted synthesis of cell ether. And also we can prepare the thiophosphonates by using green NCS and also electrochemistry approach and LED approach, and also the cysteine carbonates promoted the PS bond formation. And last part is the P dinitrogen bond formation. So thank you so much for your kind attention. But sorry for the technical problem for sharing my slide. So thank you so much.
Thank you, Professor Lee, for your elaborated talk. Although a little bit there is a problem of technical features, but we apologize for that. Yeah, now it's quite good. So, any questions or comments from the audience?
So I think the best way would be in person. So I hope next time I can join the conference in India.
Yeah, it would be pleasure. So on behalf of organizing committee, we thank you for your valuable time and for sharing your work to us. Thank you so much. Have a nice time. Good day to you.
Thank you.
Okay. So next, yeah. So connect this one. Okay, so the next speaker is Professor K.N. Singh from Banaras Hindu University. So he's a very good friend of all of us, and his working area is very diverse. And today he would like to speak about their contributions for exploration of new synthenes for decarboxylative coupling reactions. So Professor K.N. Singh, sir.
Thank you. जल्दी लिया भैया थोड़ा जल्दी
Thank you, Professor Sahu, for your kind introduction. Good morning, everybody. Before I start, let me express my gratitude to Professor Badsara for providing me this opportunity. The topic, as you can see, deals with exploration of some new synthenes and decarboxylative coupling reactions. It's not moving. Okay, thank you. So our current research interest lies in methodology development, and in that pursuit, we have made use of CH activation, some decarboxylative coupling reactions, and exploration of new synthenes. My today's presentation will cover the first two, sulfonyl hydroxides as a new synthene and decarboxylative coupling reactions. Okay. Let me directly start with the synthesis of vinyl sulfides, which are present in many natural products with remarkable biological activities. Vinyl sulfides, they are important intermediate in the synthesis of functionalized molecules, and they have extensive applications in material science. You see, the most prevalent approach for preparing vinyl sulfides involves alkyne and thiol. As you can see, these were three reports which were Okay. All right. So what people do usually, they make use of alkyne and thiol under metal catalysis. So the problem of these prevalent methods is that thiols are very foul smelling, they are volatile, and they readily undergo SS coupling, leading to the formation of undesired sulfides, rather disulfides. As regards transition metals, they are usually expensive, moisture sensitive, and harmful. So there has always been a demand to explore some alternatives of thiols, which may be practically used. Well, we were not carrying out any reaction to bring about the synthesis of vinyl sulfides. Rather, we were carrying out a reaction of idea was to have the formation of alene for some other products. But we were confronted with the formation of a product whose spectral data did not support the formation of alenes. Then we resorted to the use of single crystal X-ray, which conclusively proved the structure as vinyl sulfide. Then what we did, we tried to what we call simplify one of the reacting partners. We made use of simple toluene sulfonyl hydrazide, and that readily reacted with phenylstilene, giving rise to the same product in almost identical yield. Then we further went down to sulfonyl chloride, but sulfonyl chloride did not work under the conditions which we were put in. Now, actually, the idea was that this tosyl hydrazone might have reduced to thiol. That was the assumption. And then we optimized the reaction conditions while making use of sulfonyl hydrazide and alkyne to come out with some final condition. So we have carried out a good number of reactions while making use of sulfonyl hydrazide. And when we allowed sulfonyl hydrazide to react with alkyne in the presence of DV and acetic acid, it immediately gave rise to vinyl sulfides. So altogether 21 products were prepared with an yield of 67 to 92%. And we had made use of aryl sulfonyl chlorides having substituents like methyl, nitromethoxy, and tersibutide. We also made use of benzyl sulfonyl hydrazide that also gave rise to the corresponding product in rather better yield. As regards aryl alkynes, we had substituents like methyl, tersiributyl, and fluoro. We also made use of 2-ethynyl pyridine, and that also worked well to give rise to the corresponding product. So this was the first report on the use of sulfonyl hydrazide as thiol equivalent. Ionic liquid was recycled up to 5 times without any diminution in its catalytic activity. And in this regard, I would like to report this conventional condition in a sling tube at 120 degrees C that required almost 24 hours and gave rise to the product in just 52%. So, okay, these are various products which we have prepared by using our protocol. And as you can see, the reaction is reselective. It follows anti-Markovnikov addition. As regards stereoselectivity, we could see Z-oriented products in some cases, but in the great majority of cases, we ended with different cis-trans ratios, which I have seen shown here. Well, this is the probable mechanism that is based on some controlled experiments and literature. And as you can see, this sequential removal of sulfonyl hydrazide by DBU state in the presence of acetic acid gave rise to this one. I mean, thiodiazonium salt, and that thiodiazonium salt, when reacted with DBU and removed nitrogen, gave rise to this what you call N-sulfonylated DBU. And this N-sulfonylated DBU eventually reacted with what you call alkyne to afford vinyl sulfide. Now, according to our control experiment, this is another possibility whereby we could get the same product. Well, thus we could make use of sulfonyl hydrazide for some more reactions. What we thought, let us make use of sulfonyl hydrazide for some other reactions, and then came to our mind the synthesis of unsymmetrical sulfides, because preparing symmetrical sulfides is quite easy, but preparing unsymmetrical sulfides in good yield is really challenge. And what we did, we carried out the reaction of sulfonyl hydrazide with haloarenes, hetero aromatic derivatives, and some benzyl what you call halides. They all work well, giving rise to the product in 65 to 93% yield. Now, the problem of, sorry. Okay, sorry. Traditional methods had many disadvantages, which I have written here. Now, the only problem of this method was that it required a high temperature of the order of 130 degrees C. These are various products which we have prepared, and as you can see, when we made use of benzyl halide, it did not require copper salt. So that worked well without copper. Now, well, the only thing which always stayed in our mind was that how to develop a room temperature protocol for preparing unsymmetrical sulfides. So in that regard, we thought of our earlier report where we had prepared different N-related products while making use of chain lamp coupling, and we had carried out optimization to work with nickel chloride and 2,2' bipyridine. So the same reaction conditions we had applied in this case. What we thought that if haloarenes they are replaced by boronic acids, perhaps we may get the product at room temperature. And when we carried out the reaction, we were happy to see the formation of the product in 53 to 90% yield. And this was in fact a room temperature protocol under open atmosphere. And of course, it involves nickel catalyzed chain lamp coupling. So these are various products which we have prepared while making use of this approach. We also used N-octyl sulfonyl hydrazide, natural boronic acid, and pyridine 2-ol boronic acid. So they all work well under the present conditions and gave rise to the product. Similar type of mechanism we had proposed for preparing unsymmetrical sulfides while making use of chain lamp coupling. So let me skip it. Okay, now our next target was how to prepare vinyl sulfones. Because if you start sulfonyl hydrazide, the idea was that we should tune the conditions so as to get a sulfonyl radical. And once that sulfonyl radical is formed, that may eventually react with what you call this carboxylic acid. So these were two important reports before we could undertake our work. The first one required alkyne and sulfonic acid, rather sodium salt of sulfonic acid, triazole gold complex and gallium triplet. They were used as catalysts. In the second one, the researchers made use of styrene along with sulfonyl hydrazide in the presence of copper chloride. So the challenge before us was to provide a practically useful alternative to alkynes and sterines, because alkynes and sterines, they have their own disadvantage. Another problem was how to get rid of transition metals. And we could do that while performing this reaction. We made use of cinnamic acid as well as what we call phenylpropiolic acid. And the catalyst which we had used, iodine, TBHP, DBU in acetonitrile water mixture. Okay, now have a look over the use of carboxylic acids. Why did we use carboxylic acid? Because they are usually cheap as compared to other coupling reagents, and they are easily available in great structural diversity, either commercially or by synthetic methods. Then carboxylic acids are usually stable, non-toxic, and easy to store and handle. They are far less air and moisture sensitive as compared to organometallic compounds. And CO2 is the only product which is non-flammable and goes off during the course of reaction. Another feature of carboxylic acids is decarboxylation can drive the regioselective coupling against CH activation, where site selectivity is very difficult. So with this, we could develop a metal-free room temperature protocol for decarboxylative sulfonofunctionalization. And the stereo around this carbon-carbon double bond. Sorry. Around this double bond was E. As I said, we also made use of what we call phenylpropiolic acid that worked well and gave rise to this product. Okay, these are various products which we had prepared. The stereochemistry is exclusively E, and this is also applicable to aliphatic sulfonyl hydroxides and 3-phenylpropiolic acid. This is the mechanism. The reaction between iodine and TBHP gave rise to I dot, and that I dot was responsible to bring about the formation of this sulfonyl radical. And this sulfonyl radical eventually reacts with carboxylate to give rise to this product. This is actually a recapitulation of what we have done as regards sulfonyl hydroxides as a new synthon. You see, well, this sulfonyl hydroxide in the presence of alkyne gave rise to vinyl sulfide. When this sulfonyl hydroxide was made to react with haloarenes, we could get the formation of unsymmetrical sulfides, and this is actually room temperature protocol, making use of boranic acids, and this is what we call vinyl sulfones. The other reports I did not discuss just due to shortage of time. Now let me take up some decarboxylic decoupling reactions. See, the first one involved the synthesis of thiamide. Conventionally, thiamides are prepared either by thionation of amides while using P2S5, or they are prepared by using Wilgrad-Kindle reaction, which I have shown here. Now, these were three reports. They made use of alkyne, then aromatic aldehydes, and benzylamine, along with amines and elemental sulfur. So what we thought, can't we replace alkyne, benzyl, amine, and aromatic aldehydes with carboxylic acid? And with that vision, we could develop the formation of thiamides. We started with aryl acetic acid, and that aryl acetic acid was made to react with different amines, either primary or secondary. They all worked well in the presence of elemental sulfur, and that gave rise to this what we call thiamide. Now, when we implied aromatic amines in the same reaction, we could not get the formation of product in high yield. We could get a yield less than 20%. So we re-optimized these aromatic amines conditions, and we could get the formation of thiamides. Interestingly, when this amine was ortho substituted, like phenylene diamine or what you call, 2-aminophenol or 2-aminothiophenol, they eventually cyclize to give benzozoles. So we could achieve the formation of 2 substituted benzoxazole, 2 substituted benzimidazole, and 2 substituted benzothiazole. And when we replaced aryl acetic acid by cinnamic acid under similar conditions, we could get the formation of aryl ethane thioamidated products. So this was a useful approach to thiamides and benzozoles, and the reaction is metal-free, oxidant-free, and solvent-free. Let me skip this part. Okay, no problem. All right. Okay. Now we wanted to prepare some benzothiazoles while making use of orthochloronitroarene. So we could do the literature survey, and these were 2 reports which were available to us. The first one made use of orthoiodo and benzyl amine, and they had made use of K2S CuBr TMEDA. In the second one, they made use of orthochloronitrobenzene with benzylamine. So in the presence of elemental sulfur. And as you can see, the first one required a pre-functionalization, and they made use of some excess oxidant. So what we did, we also carried out a similar reaction. We just replaced the second reacting partner with aryl acetic acid in the presence of elemental sulfur and N-methyl morpholine. So this way we could achieve the formation of 2 substituted benzothiazoles in 46 to 75% yield. And as you can see, different functional groups were tolerable, like carboxylic acid or nitro or OH. They all were tolerable under the course of reaction. We also made use of naphthyl acetic acid and 2-or-3 thiophene acetic acid. So the reaction is actually a decarboxylative redox cyclization study. It has been reported for the first time, and we didn't make use of any metal external oxidant or solvent. Aliphatic acids did not undergo the reaction, and the reason was that it passes through a radical mechanism. So aliphatic acids and their corresponding radicals, they could not be formed, or they did not work under the conditions which we had reported. These are various products. Let me come to this part actually. Yeah, you see, this is the intermediate which was formed in this reaction, and that eventually reacted with orthochloronitrobenzene. So the mechanism was very simple, and thus we could achieve the formation of that product. N-acylated indoles, they are usually prepared by anilides along with alkyne, vinyl acetate, and vinyl carbonate. All of the reports, they made use of what we call anilides. And we thought, let us replace this second reacting partner with acid. So that we could do, and this is the outcome. We made use of cinnamic acid along with anilide, and thus we could achieve the formation of N-acylated indoles. So it is actually a cascade CCNCN bond formation and concerted ortho-CH activation and decarboxylative coupling. How much time is there? Over? Okay. So this is another report where we had made use of cinnamic acid and iodoarene to achieve the formation of 1-2 dicarbonyl compounds. Actually, the beauty of this reaction was the use of PDI, because it undergoes 2 successive photoexcitations in a single catalytic cycle, and that was disclosed by Koenig. So we just made use of PDI to achieve the formation of these products. Okay, So this is the last one. I will stop after this. We have also made use of cinnamic acids and aryl diazonium tetrafluoroborate to prepare what you call transoxylanes. And I don't want to take much time because these are various products which we have prepared, and this is the probable mechanism. So, Professor Sahu sir, time is over. Okay. So shall I start? Yeah, okay. Let me go to acknowledgement directly. Okay. All right. So finally, I'm thankful to my students and funding agencies like SERP, CSER, and DVT for carrying out this work. So thank you all for your patience.
Thank you, Professor Singh Saab. So I'm sorry, but you have a lot of result, but we could not endorse this one. But next time, but floor is open for one or two questions.
Yeah, right epoxide. That we had scaled up, okay? And the yield was around 74%.
That is very good.
Yeah. And that is based on 1H NMR and carbon 13. Okay, if it is in carbon 13, 36 to 38, okay, for trans, and it goes to 42 to 46 for cis. Although we didn't make use of no, actually we tried to do some reactions on cis, but only one pyramethoxy cinnamic acid in cis configuration was available commercially, no other. So we just, didn't use cis. But the reaction is stereo specific. When you start with trans, you are getting trans. If you start with cis, you'll be getting cis. Yeah, okay. Shall we go to that part?
Yeah, mechanism.
Okay. Yeah.
So here that intermediate whatever radical after attack.
Yeah.
Bond is please repeat it.
But we didn't get any cis, okay? Because I don't know how this works, but this has been reported by Virendra Agrawal, okay? Similar type of mechanism, because he had done a lot of work on epoxy.
Part of this one was
Yeah, right. Yeah, right. So that way, we could get the formation of only trans.
Okay. No more questions. So, thank you. And on behalf of organizer, it's my pleasure to hand over our memento to you.
Yes, sir.
Please come fast. Time. Huh? क्या गजब का काम है अरे कोई फोटो है Okay, it's my pleasure to invite the invited lecturer by Professor Anil Kumar. He is from BITS Institute of Technology, Science and Pilani, and he would like to discuss the CH1 functionalizations of heterocycles. So, with this, Dr. Anil.
Thank you, Professor Shaw, for your kind words. And at the outset, I would like to thank the organizers for giving us the opportunity to discuss some of the work which we have carried out at BITS Pilani in last few years. I will be speaking on direct CH bond functionalization of selected heterocycle, both by some catalyzed by transition metals and some by radical mediated pathways. So if I introduce to the student, the functionalization of organic compound is a very common approach in organic chemistry, and we can convert any functional group to the other one. That is a traditional approach, functional group to functional group conversion, functionalization approach. In CH functionalization, you directly convert the CH bond into some C functional group. And this is a very common or very important reaction, and it has got a lot of attention in last 2 decades or so. And in traditional modern CH functionalization reactions are generally carried out by 3 different pathways. One is a metallic CH activation or CH functionalization, where carbon metal intermediate is formed by different mechanisms. Second one is CH insertion through use of carbene or nitrines. And another one is a CH bond homolysis that uses the radical initiator followed by radical functionalization. So there is again, for a student point of view, that what is CH functionalization and how it is different from CH activation. So CH functionalization is you convert any CH bond into a group, C functional group. We can call it a CH functionalization, but CH activation is a subset of CH functionalization reactions, where you activate the CH bond in the rate determining step, and then it becomes the followed a process of the reaction. For example, this enolization cannot be called as a CH activation step. However, the overall process can become a CH functionalization reaction. So it has, many times we use CH activation, it has to be used very carefully in our reactions. So coming back to our work, which we have done, so the melamide has been used extensively in recent years as an source to functionalize several molecules with a directing group at based reactions using all type of rhodium, ruthenium, cobalt, iridium, and catalyst. And you can get a dihydro cyclized compound, you can get a annulated melamide derivatives, or you can get a spiro compound on this. Several articles by specifically from Jagan Mohan group and Professor Parvuz group has done a lot of work on this. So we thought of utilizing this melamide in preparing some of the important molecules, starting from 2-phenyl indole, and we thought that if it can annulate properly, so we can get this type of annulated 5 pentacyclic compounds. And if you have a heteroaryl at C2 position, then you can get some of the bioactive compound like isoglutenamide derivatives we can get. And if you have some protection at C3 position, then we may end up into spiro compounds as well. So we, based on the optimized studies, we could prepare a large number of compounds in this. And interestingly, if you see this, if you have a electron withdrawing group on the C2 aryl ring, it gives the best yield. In case of CF3 here, we could isolate in 97% yield. And compared to the methoxy at 4 position, you have 63. So the electron withdrawing substituted aryl, C2 aryl ring were giving better yield in this case. And that also shed some light on the mechanism of this reaction. And when we could also make several isoglutenamide derivative when you have C2 pyrazole derivative, C2 pyrazolyl indoles. And when C3 was substituted by phenyl, we could get some of the spiro compounds. So coming back to some of the control experiment to find the mechanism of this reaction, the reaction is not suppressed. The yield is not suppressed by use of scavengers like tempo and BST. And we could, if we have a N-methyl 2-phenyl indole, then we could not get the product that shows some importance of the NH or free NH compound. And if we look into the relative rate of reaction of C2 aryl having electron withdrawing group versus electron donating, we found that electron withdrawing was giving better yield in 3:1 ratio. And we could also find out the KH upon KD for this reaction by taking a mixture of C2 and penta-deuterated C2 phenyl, and the KH upon KD of this reaction is found to be 4. So based on these information and literature reports, we could explain that initially the rhodium coordinates with this nitrogen, leading to this Rhoda cycle, and that we could also detect by in situ HRMS of the reaction mixture that coordinates with the melamide, giving to this intermediate B, which can then undergo this insertion to give intermediate C. So if we have C3 position free, then it can go to roll over CH function activation or CH to give this Rhoda cycle D, and that can give us this intermediate E, which get oxidized to the final desired product. We could also detect the intermediate E in the HRMS. If we have this C3 position blocked by phenyl, then we can undergo this protonolysis, and we can get the this melamide substituted orthomelamide substituted indole, which can then undergo the Michael type, Michael type of reaction to give the spiro compound. So we also extended this methodology to C2 substituted imidazopyridines, and we could make several compounds on this, and some of the compounds were also characterized by the corresponding crystal data of this. Again, similar to previous report, the reaction with the nitro was giving a better yield compared to that of methoxy in this case. So we assume that it is going by a very similar mechanism in this case as well. And again, we did some control experiment like earlier. We could do the competitive reaction in this case. We could do the radical scavenging, was not happening. And when we added a O in this reaction, there is incorporation of deuterium at C2 position that also shows that the first step, the metallation part, is a reversible step, and KH upon KD was very similar to the last reaction, which is 3.99 in this case. And based on that, a very similar mechanism we have proposed here, leading to the Rhoda cycle B, which then can insertion of melamide leads to this, which then undergoes a rollover activation of this C3 proton and leading to this compound, which get oxidized to desired product. Recently, what we did, we took again 2-phenyl imidazole pyridines, and if you see in this compound, there are 3 hydrogens, HA, HB, HC, and we, based on the catalyst, we can annulate the cinnamaldehyde derivatives either between HA, HB or HB, HC. So when we used rhodium as a catalyst, and similar to last melamide kind, we could get this annulated product. And if we take palladium acetate as a catalyst in presence of asphos as a ligand, then we could get the annulation between HB and HC. This proton get so we could make some derivative this, and specifically in this case, the regioselectivity of the aldehyde is also fixed. We are getting the aldehyde on this side, and in this case also on the C9 position. So another approach we have done is functionalization of 3-aryl coumarins using palladium acetate as a catalyst. In this approach, we are using this lactone ring as a directing group, and it is a weakly coordinating group, so it is advantageous in, and we could make several alkynylated compound at C2 position having a substituent on phenyl ring. We could also use different alkynyl derivatives, and a structure of one of the compound was also confirmed by X-ray. Continuing this, we could also functionalize these 3-aryl to bring in halo here. We could do bromination or we can do the chlorination. And this is very interesting in the sense that we can get selectively the meta halogenation for electron donating group. So if you have an electron donating group at para, so this type of functionalization we can do using this chemistry. And then we continued same reaction, and we could do ortho-alkoxination, trifluoroethoxination, and hydroxylation of this compound. And this ortho-hydroxy compound are very interesting compound because they can be further cyclized to make the chemistine derivative. So you can use a DDQ as oxidant, and this can get oxidized to this furan fused coumarins, and we could make some of the chemistine derivative using this approach. So the mechanism of this reaction, for all these reactions are catalyzed by palladium acetate. So we assume that this we could detect in mass. So first palladium coordinates with this oxygen and ortho CH bond leading to this pallada cycle, and then because we are, this is oxidizing medium, so this can get dimerized and get oxidized to palladium 4, and that can undergo the reductive elimination to give this ortho acetoxy derivative, which can hydrolyze to ortho hydroxy compound. Very recently, we also started utilizing ball milling for several chemical reactions, and we are using ball milling for CH functionalization reaction here. So we use 2-phenyl imidazopyridine again and alkynes to give form this alkylated compounds. And this reaction is interesting because most of these reactions are done at high temperature and longer time. We could do it at within 4.5 hours. We get excellent yield by using this methodology, and several alkynes could be used for this. So giving the regioselectively this dialkylated compounds, and some of the compound were also characterized by single X-ray. When we used aliphatic alkynes, we were getting mono substituted product. In case of aliphatic alkyne, aromatic alkynes, we were getting the dialkylated product. So we also extended this for other two-aryl heteroarenes. We can have pyrazoles. We can also have simple pyrazole, indazoles and pyrazoles and other heterocycle compound for doing this alkylation under ball milling. So mechanism of this reaction is actually standard metal catalyzed thing. We could isolate this intermediate A, and that intermediate again catalyzes this reaction. So that shows that this is an intermediate of this reaction, and KH upon KD for this reaction was found to be 2.2, and there was a deuterium exchange at ortho position in around 13.5% only, and whereas the deuterium exchange at C3 position was 45%. So based on this information, we could propose the mechanism of that ruthenium first coordinate and forms this Rutherna cycle A, which then coordinates with alkyne and leading to this insertion product. That insertion product then again undergoes CH active again the other position, and that C3 position. After basically after protonation, we get alkylated compound, and then it gets rotated, and this another ortho position again forms this Rutherna cycle E, which again follows a very similar pathway to give a dialkylated product. Taking this ball milling to another reactions, we did a Friedel-Crafts hydroxyalkylation, and this is an interesting reaction because we can do a selectively hydroxyalkyl product, and we were not getting a bis compound in this case. If we perform same reaction under thermal condition, we generally end up into a bis product. So here the zinc triplet was used as catalyst, and we used sodium acetate as additive. We could isolate in 92%, and these are isolated real hydroxyalkylated products. And we could extend this to several substrate, several C2 substituted and other substituted imidazopyridines could work. It also works with formaldehyde leading to hydroxymethyl, and this is very interesting because getting this isolated is very difficult, and we could characterize this by a single XRD of this compound, and this work with different, we could make a different substituted compound with hydroxymethyl groups. So we also extended this to other esters, indole also, and get indole derivative in this case. So the mechanism of this reaction, to prove the mechanism of this reaction, we did some regioselectivity. When we substituted bromine at C3 position, the reaction was not going, we could recover the starting material in this case. That means this is a selective, because this C3 is more nucleophilic, so only that position it is going. A radical scavenging experiment shows that the reaction is not affected by the presence of radicals, and therefore the radical intermediate may not be involved in this reaction. Competitive experiment shows that the reaction with electron donating substituent on C2 phenyl were reacting much faster than those of electron withdrawing. That was 4:1 ratio, and deuterium experiment and deuteriation at C3 position and doing a H upon KD was around 1.05, again indicating that the CH bond breaking is not involved in the rate determining step. This could also be, we could scale up this on gram scale, both for indole and imidazopyridine. We could make a 1.3 gram of this hydroxylated and 0.98 gram of this hydroxylated compound. And this can be now, this shows that these are very useful. We can use them for doing further reaction like substitution of this with S-phenyl by making this Cl and followed by substitution. We can do heterocoupling, making this asymmetric bis compound and so on. And we could also hydrolyze this to acid. Now coming back to the radical mediated reactions, we have used tempo as a radical initiator case, and we can generate a carbon centered radical that can add to indole or imidazopyridine to give us hydroxyfluoroalkylated imidazopyridines, and these are some of the examples for that, and some of the compound have been also analyzed by single XRD. So these are control experiment, and this shows that this goes via radical mechanism, like BHT dimer we could isolate, and we could also detect hydroxylated tempo in the in situ HRMS. And I'll skip this to mechanism of this. So we propose 3 different pathways because we don't have exact evidence for this by which pathway this is going, but based on this we can say that tempo is involving in the generation of a carbon-centered hydroxyl radical. I think, and this is if you see from the last experiment, we got this as a side product when we were doing a tempo and this control experiment. So we thought we will extend this and make use of this reaction to make butylate quinone methyl substituted imidazopyridine and indole using tempo as a catalyst in this case. Again, same mechanism based on the previous. And this is the last one. Very recently, we have developed a very, what you call, very common method for functionalization of this quinoxalinones at C3 position. Using select fluor as a radical initiator, and we can do hydroxylation here, we can do sulfination here, we can do amination and salination using this approach. So these are some of the compound we have prepared for this. We can also use natural alcohols for this, and we could do salination, several salination and amination. This was a limitation that it was only going for primary amination, not secondary amination. Amines were not working under this condition. So we could isolate this intermediate, and we could detect this also in the control experiment when VST was used as a scavenger. So based on that, again, we have proposed two different pathway, either path A or path B is working for this reaction leading to this, because we could have B as intermediate. This was, we could trap in the. These are some other reactions our group has done at BITS Pilani. I'll skip this and I'll summarize that we have developed some novel method for functionalization of heterocycles through transition metal catalyzed CH bond functionalization reaction, or radical mediated CH bond functionalization, or by implying some mechanochemical condition under ball milling. And these are some scaffolds we have developed by these functionalization methods. I thank my past group members and present group members. The work which I have presented was mostly done by Dhananjay, Neha, and Sonam. And Bhavani did the ball milling work. I also thank my collaborator, Professor Benton Deboy from University of Rhode Island, Professor K. Parang from Chapman University, Professor Mukund Jha, Nipi Singh University, Professor Dalip Kumar BITS Pilani is actually my mentor also there, and Professor Benu Bhattadas, IAC. He did some of the biological work with us. And I thank the funding agency, SERV, DST FIST, and CSIR, UGC, and BITS Pilani for all the infrastructure. And thank you all for your kind attention and listening.
So thank you, Dr. Anil, such a elaborative work, but due to time constraint, I'm sorry. You can discuss the chemistry in during the break. Or anybody, any single quick question?
The ester group, that side is substitution the phenyl group, or
Esters we have not tried. Melamide it was working. Melamide also it is.
Cinnamaldehyde. If you replace the phenyl, simple acrolein, like this, did you try?
Let me see. No. No, we have not tried.
Okay, so.
Yeah, sure. Thank you so much, sir. Now I would like to request Professor A.K. Sahu, chairperson of this session, to present a token of reverence to Professor Anil Kumar.
Thank you,
Mahesh. Thank you, Professor Sahu and Anil.
So finally, it's my pleasure to invite our own alumni of the University of Hyderabad, Dr. Raju Reddy Garu, and then from Department of Chemistry in Osmania University, he has joined as a UGC sponsored assistant professor. And with this, Dr. Raju.
Thank you very much, Sahu sir, for kind introduction. Nice words. At outset, I would like to thank you for organizing committee to present some of our group work, and especially Satpal Singh Batsara, so for his kind invitation. So I would like to talk on the cycloannulation strategies using the beta iodovinyl sulfones. Now the question is what is the beta iodovinyl sulfones? The beta iodovinyl sulfones, such as this molecule having the multifunctional groups, which has the alkene, iodo, and sulfone. If you carefully look at this molecule, we can make these such a variety of analogs such as heterocycles. So this molecule or scaffold can be prepared using the alkynes, and variety of the sulfones in the presence of the iodine or NIS, we can able to achieve these molecules. So in the previously, there are only few reports they have been find the applications of this alkynyl beta iodovinyl sulfone, such as Suzuki or Heck coupling or Sonogoshia coupling. And our interest to develop using this beta iodovinyl sulfone to prepare the variety of heterocyclic using this center and this alkene center as like this. If you're able to make this useful beta iodovinyl sulfone by Michael addition, oxo-Michael addition or amine addition, followed by the cyclization using this active hydrogens. So with this idea, we started the working on the using the variety of the 2 iodophenols, and which will be active for the oxo-Michael addition of the beta iodovinyl sulfone under the basic condition, which is able to get the desired vinyl ethers, followed by the under Suzuki type Heck type coupling under palladium catalysis, we will be able to produce the desired 2,3 disubstituted benzofurans. So trust me, at the same time, the Benval and co-worker, during our work in progress, the Benval co-workers published the similar work, so which they use the beta iodoenols, which they can able to prepare the corresponding the benzofuran systems. So with this idea, we have gone through several optimization conditions, various parameters we have explored to turn to the best reaction condition. We are able to achieve the using the palladium acetate and triphenyl phosphine and base as the cesium carbonate. In DMF at 120 degrees, we can able to get the 2 steps which involve the oxo-Michael addition followed by the elimination and Heck reactions. And with this one, we have explored the variety of other substrates such as the iodophenols, and that's iodophenols can be provided the moderate yield of the desired 3-sulfonyl benzofurans, whereas as expected, the chlorophenols, we give the uncyclized product in the 68% yield. We also explored the understanding of the reactions using the 2-bromothiol and 2-bromoanilines, but however, in the case of the aniline, we found no reactions, and only the mixture of the products were observed in the case of the 2-bromophenylene. And instead of the iodo, we also explored the 2-bromo beta iodovinyl sulfone under standard condition. We able to obtain only the 18% of the desired product. Subsequently, to understand the reaction scope and viability, so various substrates were examined very carefully, and a variety of the sulfones can be studied, and including the different heteroaryls, and also can be explored the different other place of the substituents will be explored very carefully. All the cases we obtain the moderate to good yields. And interestingly, these reactions, this protocol can be extended to the PD substituted furans, and the scope of the reaction very limited to the when we use the two alkyl substituted beta iodovinyl sulfones, so the product was obtained in the low yield or traces in the case. So further, we explored the variety of the bromophenols. The bromophenols also facilitate the reactions and provide the desired 3-sulfonyl product, and a variety of the substituted phenols can be explored under the optimized reaction conditions, including the NASTyle systems also very nicely proceeding the formation of this desired product. And the ground scale reactions also explored to understand the viability of the reactions. So we performed the 2 independent reaction conditions. So since the cesium carbonate is very expensive, so we did the reaction with the cesium carbonate as well as the potassium carbonate. So for cesium carbonate giving the yield of the desired product in 81% yield, whereas we use the cesium potassium carbonate that the product is forming the 75 and 71% of the desired product with slightly lower yield as compared with the cesium carbonate. Subsequently, we conducted the several control experiments to understand the reaction nation. Here are two interesting points. So Soros are already published the using the internal alkynes, just phenols, able to produce the 2,3 disubstituted benzofurans. So keeping in that mind, so we perform the same reactions using our substrate like beta iodo sulfones. However, we could not see our desired product. We able to see only uncycled product of the vinyl ether in the 74% yield. So for a while, we also used the simply instead of the bromo-aryl compounds under the standard condition, we could not detect any of these products as expected. And also, we also used the terminal alkyne for the same reaction, and we able to prepare the other geometry of the isomer vinyl ethers, which will be allowed the reaction very restriction because of the geometry of the double bond, which could not allow, and we obtained the 13% of yield of the desired products. And under the basic conditions, it is well reported that the alkynyl sulfones will be formed. These alkynyl sulfones will be treated with the under standard conditions. We could not detect the our desired products. That indicates these alkynyl sulfones could not be the intermediate to the product formations. And also interestingly, we were able to prepare deuterated beta iodo vinyl sulfones. That beta iodo vinyl sulfones can be understand the rate of the reactions. We could the rate of the reaction slightly slower as compared to the standard reactions. The rate determinant steps is the kinetic isotopic effect was understood by the 1.5 ratio. Keeping in this mind, and we propose the reaction mechanism. The first steps is the well established and well known is the oxo-Michael additions, and it is able to produce the desired product. So by elimination of the iodo group, and it will be produced the desired product E3A, that is the key intermediate to the cyclization. So as we know that the Heck reaction will be proceed oxidative insertion, pi bond complexation, and carbopalladations will lead to the 2,3 disubstituted benzofurans under these conditions. And then we realized subsequently to use the same substrate, same scaffold, that is the beta iodo vinyl sulfone, to prepare the other type of the heterocycles. So we have been used the one amino pyridine iodide, and we perform the reactions, and we able to produce the desulfonylate product. So unfortunately, the desulfonylate product was observed under these conditions. In the various reaction conditions we performed, but however, we can able to see only two substituted pyrazolopyridines can able to get. So we would like to see this 3-sulfonyl compound. We are interested to prepare the corresponding the 3-sulfonyl pyrazopyridine. So we could able to change the substrate that could be achieve the desired the 3-sulfonyl pyrazones. Surprisingly, we can able to prepare the anamide, tosyl anamide, that is known as ylide. So this ylide providing the instead of the desulfonylation, sulfone group retained under these conditions. For the both conditions were optimized for the best yields, and substrate scope also further explored to prepare the variety of the two substitution pyrazolopyridines, and variety of the aryl groups has been successfully provided the desired product. And not only aryl, heteroaryl and alkyl groups also will be able to produce the desired two substituted pyrazopyridines. So here is the interesting one is the where we use the alkyl substituted beta iodo vinyl sulfones. We could not observe the two substituted pyrazolopyridines. So we are able to see here the desired what we want actually, that the sulfone is the retained under these the conditions. So further we extend the scope of the reaction using the beta bromo iodo vinyl sulfone under the standard conditions, the desired product obtained in the 69% yield. The scope of the reactions further extended to the using the anamide under the optimized conditions. The variety of the scaffold has been able to see variety of aryl groups and meta-substituted and naphthyl, heteroaryl and alkyl variety of the substituents able to get the desired product having the sulfone groups. Under these under the optimized conditions, we able to try the reaction with the methyl sulfone. Under these conditions, we unfortunately we able to produce, we able to see this the 3-sulfonyl and as well as the beta keto sulfones under the mixture of the products. Further, we control experiments were performed in the using the alkynes and no reaction was observed in the both cases. So, as expected, the under basic condition, beta sulfones will undergo to the to form this kind of the scaffolds, the iodovinyl sulfones, and these can be treated with the under standard condition. Here is the 23% of this. Two products was observed, the 3A and 4A in 23 and 56%, whereas the elide was used, no reaction was observed. Further, we have used the deuterated beta iodovinyl sulfones under the standard condition. The only thing we observed, the reaction was slightly slower as compared to the standard reaction, and we able to produce the desired product in the 73%. In the similarly using the elide, also we able to produce the desired product where we use the deuterated beta iodovinyl sulfone. Based on these control experiments, we propose reaction mechanism to deliver the desired product that will be known that the base used this will be free amine will be formed. This free amine will undergo to the formal 3+2 cycloaddition reactions such as like this. So which will be lead to the HI eliminations and subsequently desulfonylation for the aromatization for stability of the compound to deliver the desired. On the other hand, so we can able to use this elide, which will having the sulfone retained on the product that will be produced under 3+2 cycloadditions and which could be undergo such as the 3+2 cycloaddition reactions which could be able to deliver the desired product. And again, we perform the ground scale reaction of this strategy. So it works very well under these optimized conditions and we get the little sacrificed yields under the optimized reaction conditions. Further, we utilize the same substrate, the beta iodovinyl sulfone, where the other partner of the reaction can be used. So under using these 2-hydroxy chalcones, so that expected the oxamical addition will be expected a product, whether it could be undergo cycloannihilation product or not. So under optimization reaction can begin with at room temperature. The reaction at room temperature in 15 hours, the desired product, uncyclized product only we observed. Then we heated the reaction to the 90 degrees. There is the formation of the product, cyclic product was observed, further heating in the 120 degrees. So the reaction proceed the 77% yield and the desired product or uncyclized product was observed in the traces. And finally, we achieve the best result using the changing the base K3VPO4 and at 90 degrees, 94% yield. And the scope of the reaction further explode variety of the beta iodovinyl sulfone restricted with the corresponding 2-hydroxy chalcones. We able to produce, including the estrone derived chalk benzopyranes, we able to produce. This having the stereogenic centers that produce the 1:1 ratio of the desired product. Further, we explored the reactions using the different chalcones or cinnamides also, 2-hydroxy cinnamides also further explored under the kind of optimized conditions. So where we use the cinnamides that required the high temperature, at 90 degrees, the reaction was sluggish. So both uncycled and cyclized product was observed. So where we use it then 120 degrees, so that will be produced the desired product. Unfortunately, when we use the 2-hydroxy sulfones, that will be only produce the desired uncyclized product where we could not see the cyclized product under these conditions. So further, control experiments were performed using the alkynyl sulfone that expected under basic conditions, and we could not observe the desired benzofuran product under these conditions. Again, we used the deuterated compound to understanding the primary kinetic isotopic effect. There is a slight slower action was observed under the optimized conditions. So we used the intermediate to, there is a two condition without base. The reaction was traces where we used the K3PO4 and the product, the desired product was observed in 85% yield. So based on these control experiments, we are able to produce the propose the mechanism, and the ground scale reactions also performed. And also we can see these reactions can be going through the Witting use under basic condition directly providing the desired product. When 90 degrees the uncyclized product, at 120 degrees the cyclized product. And based on this idea, so we want to see this reaction in three component reaction directly in situ preparation of these chalcones that could able to generate in situ preparation of the chalcone that able to produce the desired product without isolation of the 2-hydroxy chalcones. And the ground scale reactions also performed using the three component reactions. Further functionalization also performed using these kind of scaffolds. So, and recently we able to also see again oxanucleophile 2-hydroxy orthoalkynyl hydroxy compounds. So we initially talked with we generated the E9s. These E9s will treated with the manganese acetate, which giving the interesting product. The expected product is the sulfone is the retained here. However, we able to see that the product is sulfone is the migrating. So based on this optimization reaction condition was achieved by the sequential one-pot reactions, first treated with the base, then followed by the manganese acetate. So which involve the oxamical addition and followed by the cycloamylations, and subsequently sulfonyl migration occurred under the sequential one-pot conditions. So substrates to further explode the variety of this beta idovinyl sulfones, including the histone derivative and the complex molecules. And further, we also explored the orthoalkynyl hydroxy compounds, various orthohydroxy salts also tolerated under the optimized reaction conditions. So here is the one interesting point I would like to mention, the way the electron withdrawing group here, so the opposite of the phenol or alkyl group. So the opposite of the phenol, there is the only 5% product was detected. And on the other hand, the opposite of the alkyne, only 23% of the desired product was obtained. So when the electron withdrawing group significantly affecting the formation of the desired product of this substrate. So again, the control experiments were performed for the understanding the reaction mechanism. Here is the two interesting points is there, whether the sulfonyl migration is going to through intramolecular reaction or intermolecular. In order to know this one, we able to add this sulfonate salt. That salt will be provided the two mixture of the products. So this indicate the reaction will be proceeding the intermolecular sulfonyl migrations. Further, to understand the reaction mechanism, we treated with the tempo or BHT, whether the reaction sulfonyl migration occurred through radical or ionic mechanism. So that the results clearly indicate that the reaction proceeds the radical migration. To isolate the sulfone, to trap the sulfone precursor, so we performed with the reaction with the diphenyl ethylene, and we unfortunately we could not detect the corresponding the sulfone radical. However, we able to see detected this kind of scaffold under by using the HRMS analysis. And the mechanism is proposed here that involves the oxamical addition followed by the sulfonyl cleavage and initiation and termination cyclization followed by the sulfonyl radical migration, which involves in the for the mechanism. And gram scale reactions were to understanding the reaction protocol. Gram scale reaction was performed. The reaction works well at the gram scale reactions. Subsequently, we introduced the desulfonine and sulfonylation under these kind of basic conditions. We are able to achieve with the using the disulfide as well as the corresponding the thiocresol. And to summarize these reactions, we can see this. We started with the using the beta iodine sulfone somewhere in the 2017. We start publishing the work in the variety of the scaffold sulfone content scaffolds. And today I discussed about these two and this one and these scaffolds. However, there is another interesting papers on this using this one. And we communicated this using the same beta iodine sulfone. This works in being in the progress. And we are also working in another area where we used the extensively thiosulfonates. This is known as the thiosulfonates. We can be able to prepare the variety of this scaffold using the thiosulfonates. And unlike others, we also used the time, the COVID time, we able to publish a couple of reviews. And if you want information further, you can visit my website. And the first throw of the student significantly contributed on the aspects of the beta iodine sulfones, and other two already graduated from a group, and they found the applications of the thiosulfonates. So here is where I would like to thank you, Dr. Ramakrishna, who supported me single crystal XRD from the NCL. So, and also I would like to acknowledge the various funding from CCSAR, DST, UGC for the support of my research. And if you have any question, I would be happy to answer.
Okay, so thank you, Dr. Raju Garu. So, but you have to exceed a lot of time. So, in that case, there will be no question. I'm sorry, but thank you very much.
Yeah, sure. Thank you, sir. I would like to request the chairperson to present a token of appreciation to Professor R.J. Reddy.
Support, please. No.
He's asking over the stage. With this, I also request Professor Maya Shankar Singh, who have joined us here from Banaras Hindu University, Varanasi, to please come over the stage and present a token of gratitude to Professor A.K. Sahu for his valuable time.
Okay, so thank you all the four speakers. They have taken the time on time, but I'm sorry I have taken 7 minutes more in this whole lecture, but end of the day, it's okay. Good. Have a nice day.
Thank you so much, Sahutar.
आपको लेफ्ट में आपको मैं आपका राइट एंड लेफ्ट आर्म तो चलते हैं
Only the Banaras wala people can know the left difference between left and right standing.
Yeah, they don't know actually then. वो प्यार जो करते हैं ना वो लेफ्ट राइट को समझ में नहीं आता हमको.
Ladies and gentlemen, this is the time for a break. We are going to have a 10-minute break. Please be back by 12:25. I request please be back here by 12:25. It's a short break. I also request the participants who are supposed to give talk or speak in the next session, please come over the stage and hand over the PPTs to Dr. Pragati Pagaria. May I request all the attendees of this conference to please come back to the indoor auditorium and be seated. If you are outside this auditorium and you can please come back and take your seats. We are about to begin the session.
चालू कर दो आप
Ladies and gentlemen, this is session 12. The chairperson of this session is Professor T. Punyamurthy from IIT Guwahati. We have heard him twice, and now I believe there is no more introduction needed of him. We all are very well known to him. Sir, may I request you to please come over the stage and take this session forward. Professor T. Punyamurthy from IIT Bangalore, Guwahati.
Very good.
So very good afternoon to all of you. In this session, we have 4 speakers. I request all the speakers, please stick to the time. And let me now introduce the first speaker of this session, Rotni from IIT Bombay. He is one of the well-known organic chemists, all of you know very well. He doesn't need any introduction, but just I will briefly tell 1 or 2 words about his academics. He completed his PhD from NCL Pune, then pursued postdoc research at Tokyo University, as well as University of Freiburg. After that, he spent a short time at the Institute of Chemistry, UNA, Mexico City. And following that, he returned back to India, joined in IIT Bombay. In 2007, he is full professor. Today, he will speak about step economic total synthesis of selected natural products. Please, Courtney.
Yeah, thank you, Professor Punyamurthy, for your kind words of introduction. And I thank Dr. Satpal and his team for the invite and the hospitality. Yesterday, we had a series of talks on total synthesis and how natural products synthesis is significant and it is important. Today, I'll take you to a little different topic on step economy. Okay, that is how to strategize total synthesis and make it in the most shortest possible way. Okay, because we cannot really run total synthesis today the conventional way. Okay, that it should not be having a large number of steps. So what total synthesis today demands, okay, that it demands economy and efficiency in synthesis. So these two words are very important, and some way to address these two issues is that, or what we try to do is to avoid a large number of steps, to avoid protecting groups and so on. Okay, so how to build a molecule in a shortest sequence? We need today convergent processes. Okay, we all know how to make molecules by building blocks and then combining them towards the end. We need cascade reactions, tandem reactions, so that molecular complexity can be created in the shortest possible way. We need divergent or unified approaches that not only make one molecule but make an intermediate and try targeting more than one natural product. We also need today modular synthesis, because this is very important when it comes to medicinal chemistry, that if you have made modules, you can make analogs and so on. So although there is a great deal of creative art and imagination within the realm of synthetic endeavor, success is rooted in sound synthetic planning. Okay, that is, you really need to strategize a kind of sequence for making total synthesis. If it is a first synthesis, you do have some liberty to make molecules with n number of steps. But when you want to do a second synthesis, your synthesis has to really beat the earlier synthesis. So we have been advocating doing synthesis is protecting group free. Okay, that is, it avoids several steps in the synthesis, and I was fortunate to even edit a book, Protecting Group Free Synthesis. You can read this book. It has more than 100 total syntheses which have been performed by avoiding protecting groups. There are follow-up articles also we have written in ChemCom and EJOC on how to do protecting group free synthesis. So today I will tell you two stories on natural product synthesis. One is the gonolide chemistry, how we have achieved the synthesis of these natural products, and secondly, a very important compound called mycalol, which was earlier synthesized in NCL Pune, followed by many others. So I'll show the synthetic attempts in our laboratory to make these molecules. So gonolides fall into three classes of compounds. You can see this is 612 when it is electronized here, 812 when it is electronized with this hydroxy. If the C110 bond is broken, you have secogonolides. And there are a large number of molecules which basically fall into this class of compound, which have this 5-7-5 ring system. And they are very important. Okay, they have very good bioactivity, especially anti-cancer properties. I have listed here many molecules which have anti-cancer property. Capsigargin, which is on the complex molecule, if you replace this with a polypeptide, okay, this is called mipsagargin, which is a pro-drug in the phase 2 clinical trial for several cancer cell lines. So that tells us the significance of synthesizing these molecules. So our approach was a divergent approach, that we thought that we will make this compound and then build all others through a common intermediate or through a common compound, so in a divergent way. Now the approach comes from a simple literature precedence that Wang tried to add this bromolactone to an aldehyde in a Barbier type reaction, and he employed this ligand, okay, so that he can have a very good diastereoselectivity here, which is NT, and then carried out reactions to make paraconic acids. What we hypothesized is that we don't need this chiral ligand. If this bromolactone has one chiral center here, it itself can impart chirality in a diastereoselective allylation. So our whole strategy is over here, that if we have this bromolactone which has one chirality here, it can induce chirality in the subsequent steps. So to make these molecules, the base molecule was ligustrine. So we started with R-carbone, okay, from where this aldehyde can be prepared. A Barbier type reaction can give this one. And now you do a trans-lactonization, so that you have this lactone with an exomethylene group. The diol that is obtained, you can cleave this diol and do an aldehyde in reaction to make this base molecule. And then you can decorate it all sides to make all other natural products. So that was a simple strategy in our hand. And so we went to make this aldehyde, R-carbone, which is commercially available through a nucleophilic epoxidation followed by opening. We made this halohydrin. And this strategy was planned in such a way that during the wrong ring contractions, we should also have this hydroxy eliminated, so that we have a double bond created. So this hydroxy was converted into a tosylate, and the Favorsky reaction gave beautifully this ring contracted product, as well as the O-tosyl eliminated, so that we have this double bond. Now, diol reduction normally gives a mixture of aldehyde and alcohol. So we went for complete reduction of this with diol, and then oxidize the alcohol to have this aldehyde prepared. Okay. So this was prepared on gram scale, requiring only 2-column purifications. The second one, that is the allyl bromolactone, was prepared from a D-mannitol based aldehyde, which is also commercially available, by a Baylis-Hillman reaction to get this compound, followed by deprotection and cyclization with removal of this OH group and adding a bromine. So this could be prepared in just 2 steps in a good yield. So having got these 2 compounds, we went for the coupling of these 2 compounds by the Barber type annihilation by using chromium chloride, and it gave us 2 products. And in fact, this was a failure, because you can see the double bond here migrated. Okay, so this double bond, probably acidic nature of this double bond migrated here, and then annihilation happened. This was the major product, which is unwanted. So these 2 were inseparable, and so we still had this compound. So we thought we'll go ahead, and then we did a trans-electronization, first hydrolyzing this treatment with HCl. We could get this trans-electronized compound. The same happens for this one also. Then cleavage of this diol with pyridate, and then the aldehyde in reaction. We could get this molecule apart from this unwanted compound. Now we thought we made ligustrin, but then we were lucky to have an X-ray structure of the compound, and we saw the stereochemistry at this 3 position was reverse than what was present in ligustrin. So this is a diastereomer and not a ligustrin molecule. So we had to solve 2 problems here. Okay, one was how to avoid this olefin isomerization, as well as the required stereochemistry at this 3 chiral center. So this came luckily with a small change in the reaction conditions, that we employed the Barber reaction in presence of zinc. In DMF, these are neutral conditions. So this reaction occurred very well, giving these 2 products, okay, which are diastereomers at this 2 chiral center. The major product was as what was required. The minor product was this one. And we confirmed the structure of this one by converting to the earlier compound where we had taken the X-ray. So this major product, which could be separated, we took it for the trans-electronization, followed by cleavage of this diol here, and the aldehyde in reaction. We could set up the 7-membered ring, and at this time we had ligustrin prepared in 10 steps, nearly 20% overall yield. The data was perfect match with that reported in the literature. Now, one thing came to our mind, that you can see the diastereomery is only 3:1. Okay, this is low. Okay, so, and second reason is that if you see this chirality present here, it is actually cleaved here. Okay, this chirality is destroyed. So the question comes whether we really require this chirality. So what we thought is that we will use this simple bromolactone with no chiral centers, but this has chiral centers itself. It can impart chirality, and we got very good diastereomery 90:8. Okay, so this was enough, and of course these 2 diastereomers were inseparable. The minor one we could not separate it, but during the trans-electronization we could get the major product in pure form, and then a simple oxidation and aldehyde in reaction, we could prepare now ligustrin in a more than 10% additional yield in the same number of steps. Again, the earlier one, the 3:1 diastereomery, is possible that since these 2 chiral elements are maybe working together or may not be working together. So we prepared the enantiomer of the earlier compound to see if this becomes a synergistic approach, and then we may get higher diastereoselectivity. And indeed it gave, okay, 1:7.5. Okay, see, this was better than the 3:1. So and these 2 were not separable, but we went ahead for the trans-electronization. At that time, the major product could be separated in the pure form, and then the diol cleavage and the aldehyde in reaction, again a slightly improved yield of ligustrin. So having this base molecule, we went for the diversification. We did the epimerization here under Mitsunobu conditions to get the alpha hydroxy compound, pyridate based epoxidation of the more substitute double bond, and rearranging the epoxide, we completed the synthesis of groshmane. So this was the first asymmetric synthesis of groshmane. Coming to euphenolide synthesis, when we started this work, there was only 1 synthesis on euphenolide, which was published in JACS in 19 steps by Ziegel. And these last 2 steps were obvious, so that involves the reduction of this to the alcohol and epoxidation and opening over here. It's a 2-step process, so we did not go for this last 2 steps. We made this intermediate, so it's a formal synthesis. This hydroxy was converted to an ester by tiglic acid, and a selective oxidation over here with chromium trioxide. We completed the synthesis of this intermediate. While this intermediate in Jack's paper was published in 17 steps, we could prepare it only 12-step synthesis. So this was the work on gonolide chemistry, and this is published in JOC with a beautiful cover picture for this paper. Coming to my second part, that is mycolol synthesis. This was a very interesting compound, which is a polyhydroxy lipid molecule. It was isolated in 2013, published in Engavante Chemi. This was the originally proposed structure of the molecule, and then through the first synthesis by Reddy from NCL at that time, the structure was revised. Okay. Now the revision is very interesting in the sense that, of course, the chiral centers normally people try to change and get to the structure. So Reddy did one change here to make it alpha, but this is really surprising. Okay, that at 20 position acetate was proposed, but Reddy moved it to the 19 position. This is really beautiful that the structure he synthesized was not matching. Okay? And this change from here to here was remarkable, and this structure is actually the actual natural product. So if you see that the literature on these molecules, it's interesting that Reddy and Goswami both started this work at the same time. I believe so. Okay, so Reddy was lucky that he made only 3 molecules. Okay, and he could fix the stereo centers as well as this one. Okay, Goswami really struggled in the synthesis. He prepared 12 compounds. Okay, and he was fighting the structure because every compound he prepared, the structure was not matching with that of the natural product. Okay, and I believe that there was a difference of 1.5 months here. Maybe Goswami, after Reddy's synthesis was published, that he must have done the final synthesis. Otherwise, he would have not struggled 12 structures. Okay. Even Goswami went for even putting 3 carbons here instead of 2. Even that structure did not match. But Reddy was really clever in putting this acetate from 20 to the 19 position and solved the whole structure of the molecule. There was one more synthesis by Ghosh in 2018, but this synthesis requires a large number of steps. You can see the synthesis of Reddy requires 13 steps, 2.1% overall yield. Goswami's synthesis requires 16 steps, 11.1% yield. Ghosh required 19 steps, 8% overall yield. So when we took up this project, okay, almost 3 or 4 years back, it changed hands from 1 to 2 to 3rd student. And then finally we settled in a strategy that we had to beat this 13 step as well as this 16 step. And we came up with only an 8-step synthesis. So it goes by this way that we can have this bond cleaved here and do a metathesis reaction. And we already have one more double bond here. Both these double bond can be reduced at one time. So this fragment can come through a metathesis over here between these 2 fragments, and an allylic alcohol can be actually resolved to get this compound. This fragment can come, okay, from very easily from our lactone, but you see this is a syn alcohol, so we needed an anti, and we have a strategy to even epimerize the center, whereas this part can come from D-mannitol. The whole synthesis sequence is so simple, modular. You can have D-mannitol, protect as an acetonide, cleave this and reduce, and then convert the hydroxy into a tosylate. This one side fragment is ready. The second one came very easily from D-gluconolactone because this chemistry was ongoing in our lab. We prepared this lactone in one pot, a 2-step sequence. Here we epimerized this, okay, by using palladium acetate from a syn-diol to a trans 1, okay, and then reduced this with lithium aluminum hydride to a triol. The diol was protected as an acetonide with this free hydroxy. This was reacted with this tosylate, and we have this fragment ready. This fragment was prepared in only 5 steps against 8-step synthesis reported by Reddy in his paper. The second fragment was prepared again very easily, that we took this aldehyde, did an allylation reaction, which is called the Baber allylation, and then acetylation of this converted into the acetate. The other part came again very easily, that we have this aldehyde, we did vinyl Grignard, you have this allyl alcohol, resolve it with Sharpless kinetic resolution, we have this epoxide, okay. And then when we have this epoxide, we did a metathesis reaction to get this fragment, and the sulfur ylide based opening of this, we get this olefin, and the diol was protected as an acetonide. Okay, so this also requires just 5 steps from compound to prepare the 2 fragments, and then the joining of 2 fragments was easy from Grubbs Haveda catalyst, and hydrogenation of this double bond, we prepared this acetonide. This was reported by all the 3 authors before, so comparison of data was very easy, and 1 step of deprotection, we had mycolol with all these 6 chiral centers properly placed and finished in that way. We have completed many other syntheses recently, like dihydroetomaylene, first total synthesis, Kumauza-Alene, cis-anoxylipids, petromyroxol. We have back-to-back 2 papers in ChemCom. We have also completed a very interesting molecule synthesis, tri-wine sensitive molecule. We have back-to-back 2 papers. Earlier synthesis on this was done in 15 steps. We have reduced it to only 7 steps. And in this paper, we have made this molecule in only 4 steps. So to conclude, I have shown use of this allyl bromolactone or this one or the enantiomer we can convert this to an aldehyde and prepare this base molecule ligustrin. And then from here, we have prepared all these molecules. So all this work was single-handedly done by G.V. Ramakrishna, who is now a postdoc in Texas. Then the microlol synthesis is done by two students, Priyanka and Gulenur. One of them is there presenting a poster. This is a totally a modular synthesis, because we can actually use D-mannitol, we can use ribose and take the enantiomer of this D-gluconolectron, we can use L-gluconolectron, make an enantiomer of this. Sharpless conditions from plus diethyl tartrate, minus diethyl tartrate, we can make the enantiomer of this. And K chelylation by changing binol, we can make either enantiomer through a modular synthesis. So this is my group. Ramakrishna is here, Priyanka is here. So I thank them this contribution. I had many syntheses, but I could not present them here. And the funding received from DST, SERB, and Humboldt Foundation is gratefully acknowledged. And thank you very much.
therapeutic value to the patient. If you see, this is the biomolecule and this is the binding site. We have the binding site. Suppose you are taking say chloroquine. The question is how the chloroquine approaches to this and whether it is the correct way approaching that groups so that there is a perfect interaction and that can inhibit the therapeutic value. Okay. So, and what we are calling normally EC50 or IC50, whenever you are loading the drug with some nanomolar or micromolar at the exact concentration, whether the cell viability is good for 50% synthesis of the protein or that inhibits the protein synthesis. So mainly, I'm working on this alkaloid. The last one day you have seen starting from Professor Ganesh Pandey, Alakes, Professor Singh, and many others, Professor Punyamati also, they told this is the simple, structure. You can see it starts with from tetrahydroacetylcholine core. You just remove this 3 oxygen from this side. It is just a simple tetrahydroacetylcholine. This is a simple alkaloid, and this alkaloid mainly rich in these plants. As you know, plant is the master of synthesizing this natural product, and we are nothing. Just we are following, making rotosynthesis and getting it. So they synthesize in a very important way. And why I am telling, because I know the hemostatic activity of cottonin, and that you can just modify to different natural product, and they have different properties. So here, so I have started from the papaverine plant because direct synthesis, the cottonin, I will tell the story. Cottonin, 1 milligram, it costs like 56 euro, and that is not available, that's not easy. So I thought, how fast I should do the cottonin. Then I can choose the best heat molecules for the clinical trial. Okay. So with this, if you see this plant is more available in the south side, putting this 1 to 10% of the SR noscapine available. If you see SR noscapine, you have the upper part is the cottonin and the lower part is the meconic acid and that anhydrate. And initially, this is very good for cough suppression. Okay. You see, if in the 9th position, if there is a nitro in the bromo group and it has some therapeutic value. As I told, for synthesis of this, I take the help of this because the SR noscapine is chirally available and commercially is very cheap only. I started my work to make the molecular library from cottonin. So my aim is now I will fish. That means I am collecting all the cottonin scaffold. Then I will do that. I will find out the heat molecules by biological study. And from a lot of molecule, I will come to the best 3, then best 1, I'll go for that preclinical trial. Okay. So with the same, I started, but before going there, the cottonin is a very interesting molecule. As I told, this is a very level group. And cottonin, if you just make it and keep it for some time, you will not get the NMR again correctly. Because that is very level and it exists as hemicottonin form. So understanding this thing, and then as I told, this is very expensive. You can see here. So I started preparing from this is cheaply available SR noscapine. So you just do treat nitric acid for degradation and subsequent neutralization with potassium hydroxide. You will get this cottonin along with the meconic acid. And that, as I told, cottonin is not stable. So how to solve this problem? You just add either HBr or HI in ethanol. You will get the salt, and you can store for years. There is no problem. And another also, you can use bromine waters, just simple bromine water, not bromine, and that iodine direct you can use, which will give the same thing. Okay. So I have done published this one. I could have done it a patent, but I made it open for the public, and I published this in Organic Synthesis. So and after that, what I thought, I want to do the diversification as I told. So this position, then you can insert alkyne and acetyl, many functional groups. So I done this, and this is standardization table. And what this is very beautiful reaction. You just add both of them in methanol. After 30 minutes, you will get the precipitate. Just filter it. No need to do any column. It is very, very interesting. And with this, as I told, so this is the way you take this noscapine and nitric acid. Exactly after honor, but the temperature maintaining is very important. 55 degrees is very, very important. You have to make it minimum 1.1 to 2 hours. After that, you will get the endpoint you can get by this. And this opionic acid will come out as a white solid and leaving that cotton in the filtrate. So filter it. After that, what you do? you utilize the potassium hydroxide, you get this product. But immediately after that, this is the product. Immediately after that, you make it salt. Otherwise, within 2-3 hours, again it will go some roasting. It's very difficult. Okay, next. So as I told, first we have done reacted with the acidophenum to get this product. It's very easy. And you can do, I have done like 50 gram scale in a laboratory, but for organic synthesis, I send for 20 gram, you get the 10 gram product directly without any problem. And yeah, the video is there for it will work. So I was very happy to repeated my work in that laboratory, some of the laboratory, and they gave me that confirmation chapter that your procedure is repeatable. Okay, so as I told, I am now finishing collecting a lot of molecules, then I will go for biological evaluation. So I first, you can see para product, ortho product, and meta product. You can synthesize very easily. And similarly, we can see disubstituted that phenylalanine and phenylalanine also I synthesized. Next, I synthesized in naphthyl course as well as the heterocyclic course. And for all the cases, you just filter the product without any hesitation. Don't need no base, nothing, just methanol at room temperature. But in some cases, you will not get the product, so you just cool it type to 0 degree and just decant the liquid. It will be easier. Okay. So next, I am interested to also synthesize other benzofuran and in that indole core. For that, I take the commercial available that kaolin is very cheap. You just do oxidative degradation by potassium hydroxide. You will get this acetyl with this core. Then this one, you just play with the alkyl iodide, adding potassium carbonate. We can do many chain, elongation of chain, so that you get a lot of molecules. Again, also, if you understand the effect of this, remove the hydroxy group you want to do, just to do the react with triple anhydride and followed by pyridine reaction, you get this. So now I have this benzofuran moiety with me to do this. So I synthesize a lot of molecules like this. And the same thing. And moreover, also the aliphatic and anthridines and benzofuran indole to one another, most of many derivatives I synthesized. And this is very easy just. So now I have a lot of molecule. Okay. To understand the mechanism, I thought how it worked. And what I thought, I have done DFT study. Finally, I came to know this open form of this is more stable. And this amine takes this proton and make it nucleophilic enough to do this reaction. And similarly, but the question was, since 9 position is very important, that's why I try to insert that bromine or some functional group in 9%. But for that, first I tried bromination and in ethanol, but I end up with this salt. There is no bromination. And similarly, I tried NBS and acetonitrile, but instead of bromination, it goes to oxidation. And again, I tried this condition, but I'm not successful. Then I thought I should go for a subnoscopine. Directly, I do that. Okay, I was frustrated, but okay. Again, directly I do bromination with bromine water, that subnoscopine. Then I neutralize the ammonia. Then you get a lot of bunch of white product. And that's done. Then I followed that same procedure in organic synthesis. I degraded with nitric acid, followed by potassium hydroxide neutralization. You get this product. And also you store the same thing by this way. Okay. And also, next also, I want to increase the number of softness. So I prepare some alkyne insertion here, because alkyne you can do again click reaction and make a heterocyclic. So it will be beautiful again. So I prepared many substrates, you see. But then I check the chemoselectivity. If the substrate contain this and this, acetyl and this alkenyl, then alkenyl product is dominated by this acetyl product in more range. Okay. And here, but unfortunately, I'm not able to get success for the acetylene having CA2 in between. So the same thing, I done the DFT study, and I found this type that open form is good for that. Okay. Now almost I have done the collect a large number of molecules. Fishing is done. So I am interested for this doing biological evaluation. So the student asked you to start this work, and he started immediately. So as I told, I was interested in mouth oral cancer. So I collected the oral cancer cells from like Tarandon Institute, Calcutta, NCBS, and initially I got frustrated. Out of this, I didn't get any result. So I thought, what shall I do? But I should not waste a lot of molecules, because the parent skeleton has a very good property. That's why what I have done, I thought, okay, that nucleophilicity of nitrogen might be causing problem. That's why I thought to end methylation and change the nucleophilicity of nitrogen. See what I have done. I have added this methyl iodide. I have done quaternized that nitrogen salt with counterand this. Again, another also I can do the triplet also. Because triplet and iodine, they have a different nucleophilicity pressure on that nitrogen atom. But again, I want to extend it with different counter ion. So if you add, this is a very wonderful reaction. If you just add methanol and NaBO4, you will get a salt easily. You just filter it. Similarly, so this is the procedure you can do many other counter strategy. Okay, after that, now I have that bunch of molecule. So I gone for this, I synthesized many of this, okay, with that strategy. So benzofuran core as well as the general core. And finally, I selected these molecules. And I was very happy. First case, 2 heat molecule I got. You see, this and this. 2,4 dichloro and this. And okay, I was much interested for that in vivo study after that. But in vivo study, I will show you. You see that 2 molecule. You see this percent of cell viability is very good with that cancer cells. And I have initially taken this as Cal 33 FAD. Cal is that cancer cell taken from the tongue, and that FAD that is collected from ATC USA. That is this side neck, near that mouth, that pharynx saline. And that is very successful. There are some, you can see actually what happened in the mitochondria, the MTP18 protein is responsible for making the tumor growth. And after addition of this drug, and that was able to control this MTP18. So these are some biological things like concentration, you have to control many things. And finally, the in vitro study was done. And because I cannot tell anything with short of time, but this is now the 3 successful molecule. Finally, I have done it out of many molecules. Okay. So then now the next step will be in vivo study. That means we will take the human cell and insert in the mice model. Okay. This one, that student gone to that NIT Rourkela with one of my friend. He have done it. So what we have done, but before that, doing that mouse model, we have to go for ethical guideline, and also you have to see the histology of the mice. What disease you have, how many RBC, what is the weight of initial body weight, and okay. With all the data, I started this. You see, you take 2 tumor mice. That means we have taken the cell from the pharynx and inserted in the mice. And that are the tumor cell mice. And the tumor mice, death after 3 weeks if I don't give any drug. But when I apply my drug, you can see after drug treatment, this belly is normal. But the longevity increases up to 10 weeks. That means that we understand our drug is working. Then what is the exact concentration? We have to check the other parameters. You see, so first we injected the drug, and after 10 days we do surgical dislocation and take out the cancer cell and check it again. What happens there? But another thing is, whenever it is controlling that MTP18 protein, there are other proteins that will create problem. But for that, the concentration maintenance is very important. Finally, we see 50 milligram when I treat, the initial body weight, final body weight, RBC, WBC, everything is constant. But if you increase the concentration of drug from 50 milligram to 200 milligram, that also there is no problem. So what we have done, you see here total cell count also in the particular volume is constant. That means that is not killing this normal cells. Okay. So here you see minimum survival time when our control is 18 days, but when I give 50 milligram of drug, it's 28 days. Again, it is going to 35 days when I give 100 milligram, and 200 milligram, it is going to 35 days. That means minimum survivability of that mouse is increasing with the introduction of this thing. Okay. So finally, then another question comes, then what is the protein responsible for that? Which protein? So there is 2 ways. One, you can do molecular simulation, and you take this and do simulation. I found this SIRT1 protein, this fellow from Paolo, Germany, and the John from Paris, they're my collaborators. They found this is the protein responsible for that. Now, practically, we have to then find out, is it really true? For that, I have to do isothermal titration calorimetry, ITC experiment. That we have to do it. It is under process. And for this, again, you have to go for preclinical trial because this is a little difficult. So we have to collaborate with the doctors, so they should be interested. And but up to this, we are done. And you see, very interestingly, I can tell how biomolecules that they inhibit the drugs. You see, this one is 24-fold active. Here is both hydroxy and methoxy group. If you remove hydroxy group, okay, just do a triplet anhydride treatment, then palladium, then it will go out. Okay. So you get this is pure totally inactive. But if I remove this only to fold active. So this is a very complex thing that to understand how these biomolecules they interact with this drug molecule and inhibits the therapeutic value. Okay. And so this I am interested for modulating to other places to increase the efficiency of this drug in future. And these are also other hit molecules are here. This is very small, but see, okay, I have other hit molecule. So we are working. It will take a lot of time. And another interesting thing you see, this 2-bromo position, this drug is active. If I replace by chlorine and the fluorine, it's nothing. That means halogen, the halogen bond interaction to the drug, and bromine is a very good candidate for this. However, chlorine and fluorine is making the problem. That means selectivity for binding the drugs, if due to that halogen bonding is also very important. Okay, these are the now I have a lot of molecules in my laboratory. If somebody want, I can give. If somebody interested, in good amount, we can do it. And all are characterized. So these are my collaborators from different places. And this is my laboratory at Berhampur University. These are my students. And that this fellow is here. That fellow doing most of this cell line work. And see here, all of my I have produced till now 4 students. Many MSc dissertation students are there. And 2 of my students are they already in USA for PhD directly after the dissertation. And so this is the beautiful Berhampur city. I welcome all of you. If sometime you get a chance, I'll be happy to host you people. And of course, I arranged an international conference, but due to COVID, I stopped last 2 years. But I think it will be soon. This is the beautiful city. So I also thank to Berhampur University, UGC, ACRV, Planning Convergence Department for giving me the generous funding. So if someone is interested, may not be interested, but if someone is interested, I will be very happy to take. Thank you.
Okay, thank you, Lakshmidhar. On time you have completed. We may have 1 or 2 questions, please.
Yes.
Yeah. We have 2 questions. Yes.
Yes, sir.
We go on these different experiments. Did you check the pK of these molecules?
Yes, because whenever we are doing that mechanism, that time the DFT study, that pK is very important to find out the mechanism. I don't know. I'm sorry, I don't know. But that means my student knows better. Yeah, tell me, please. No. This is as I told, we have to collaborate with some doctor and or some clinic, and if they're interested, because my limit of this, we have to take because a lot of problems for that permission. Yeah.
Okay, we may have 1 more question. Then yeah.
Yeah, very nice talk. So here.
Yeah, I can hear. Yes.
Yeah. So in your in vivo studies, what was the frequency of dosing and what was the mode of administration? Like
dosing means is we inject that tumor cell and leave it for 3 days.
Yeah.
Okay. Then you do after we'll see what happens. Is there any increase of that body weight or something, belly is swelling or something? After 10 days, we will do the cervical dislocation. Then only we took out that cell and check it. And this is, as I told, this is not over. Now we have to do a lot of things to optimize this.
Okay, so I mean, you don't inject into the animals, I guess.
Yeah, no, already inject. I told the mouse model.
Okay.
Looks.
Yeah. Okay. I mean, and the other question is like, what was like? It was IV or topical, or what kind of application it was?
IV means I don't understand. Can you please?
It was like intravenous or like topical, or what was the mode of application? That's what I want to understand.
No, they're directly in that stomach of the mice directly.
Okay. Thank you very much.
Looks there are several questions. Please ask during lunchtime since we are running late. With this, let us thank Lakshmidhar for the excellent talk.
So I would like to request Professor T. Punyamurthy to present a token of appreciation to Professor Lakshmidhar Raut.
Yeah, this is, I'm really fortunate. Thank you.
Thank you.
Thank you, sir.
Next speaker is Ajay Kumar from CDRI. He completed MSc in Delhi University, PhD at CDRI. Following that, he pursued postdoctoral research at Seoul University. He returned back to India in 2011, joined at IICT Hyderabad. After spending 6 years, he moved to CDRI Lucknow in 2017. He will speak about development of isocyanide-based synthetic methods towards NCEs. Ajay, please.
A very good afternoon, everyone, and thank you, Professor Punyamurthy, for nice words of introduction. So it's a great to be here for the second time, and thanks, Dr. Satpal, for organizing this nice conference and giving us the opportunity to interact in various manners by presenting our work, by talking some of the exciting stories that we are developing at CDRI. So, considering the time of the talk, I think is just 15 minutes and maybe 5 minutes for discussion and all, I would be kind of going through glimpses of some of the research work that we are doing over there, and I would try to highlight one of the stories that actually has recently matured at our lab. So being at CDRI, we actually mainly focus on doing medicinal chemistry, where we try to kind of develop lead molecules that can be further taken for clinical trials and marketing approval. So these particular process is a kind of highly systematic and requires a lot of discipline, because it kind of, if you do not do that, then actually you end up with a lot of failures. So here our projects are driven by unmet needs. We try to choose a disease where there are limitations with the existing drugs, and where we can find some of the molecules that can actually tackle this problem in a significant manner. We do not kind of randomly go and start working on any of the disease area. When we choose the disease, then we immediately try to look for the biological targets that are actually being discovered every day. If you go for learning the biological targets for different diseases, you will come to know that a lot of biological targets have been widely explored, and the molecules that were developed had several problems. So new targets, new mode of actions are coming up, and accordingly we need to plan our strategy. Immediately after that, we create a target product profile. There actually we kind of fix ourselves that we should not cross these kind of boundaries, and there we try to limit ourselves, and also we define when do we call a molecule a hit or a lead, when do you want to take it for academic studies, what kind of talks are required. So as Lakshmidhar kind of very nicely explained a story, and one thing that I spoke was, before you take any molecule for animal studies, you must do a lot of talks, a lot of PK studies, then only you should go for animal studies. That's what we do. And after doing this, we have to kind of look for molecules. So there, to minimize our synthetic efforts, we use a lot of computational approaches to kind of have a scaffold, and then we do SAR and then look for patentable NCs. So in this particular area, one of the area that we actually started working in the year 2017, this is a PCSK9, that is one of the receptor that is responsible for kind of controlling the lipids in our body. And this was another target which is kind of mostly expressed in the patients where the statins don't work. And two of the antibodies, they are actually there in the market. We do not have any small molecule inhibitor for this particular target. So in 2017, we started looking for small molecules. We were fortunate enough, we got this particular, this particular S017062. This is a lead molecule that actually was kind of synthesized at our lab and is now at IND enabling study. This actually molecule has a very nice IC50 of 300 nanomolar. It has a ED50 of 50 milligram per kg in hamster model, and that is through oral route. The oral availability was very nice. That was around 22.9%. That is far better than a lot of statins that are there in the market. The molecule doesn't have any hERG liability and CYP inhibition. So now the molecule is safe, and we are proceeding further for this particular compound to complete the IND-enabling studies and then go for phase 1 clinical trial. So maybe some other time I'll be like when this story either it becomes successful or when we drop the molecule, I would be probably in a better position to describe the whole story. Along with that, we actually do process chemistry. This actually is again driven by the need. It is not like we choose a molecule and start making it. We go for API selection depending on the kind of high valued API where the price is very high, and we want to kind of have a affordable synthesis, and that synthesis should be patentable so that we can transfer the technology to industry where it can be marketed at a much cheaper price. So in this, actually during COVID, we were kind of looking for a lot of antiviral drugs, and there was one drug that was Arbidol that was being used in China and Russia for managing COVID. So we wanted to check this particular molecule in our country, how this actually affects, and can we use this for managing COVID. So unfortunately, this molecule was not available at our country, so we had to develop a process and make it here. So during COVID time, within 1 month, we actually developed a process that was done at 50-gram scale, and then we transferred the technology to Medis Pharma Goa. They scaled up this particular compound and then prepared the tablets. We approached DCGI. We got phase 3 clinical trial permission, and that was completed for 129 patients. And because that time COVID-19 was going down, so DCGI asked us to go for a larger clinical trial. That has also been completed, and now it is submitted to DCGI for marketing approval. In this particular case, another beauty of this molecule was that this can, along with COVID, this can also be used for influenza kind of symptoms. So we thought if this gets approval, we can try to find other uses of this drug. Along with that, we also look for another molecule. This is a Centaquin. This is a drug in the market that was actually synthesized for the first time at CDRI. And it was also used for the critically ill COVID-19 patients. So not exactly for managing antiviral things, but it was for managing the lower blood pressure. So we developed the aqueous process to prepare this particular molecule at gram scale. We patented it, and now we are in talk with Cipla to transfer the technology. So now we'll come to the kind of our interest in doing a synthesis. So being at a drug discovery lab, we always try to kind of have the synthesis that is simpler, that doesn't require multi-step, and also can be scaled up at any gram scale. So post-UGI modification was one thing where actually we prepared a different kind of UGI templates. So this was the first template that we started working, and then this was the second one, and this was the third one. So using these UGI adults, we actually prepared a lot of sp3 enriched heterocycles. You see, mostly we try to avoid flat molecules because they actually have been explored a lot, and in drug discovery, always they end up with kind of creating a lot of toxicities. So in this, most of the reactions were diisoselective. They were one pot, and the product could be scaled up at multi-gram scale. Along with that, because when you develop these methods, the two things we look for. One is like if we can use these methods to synthesize some drug, or we can use these methods to synthesize some natural product, so that kind of we also do a little bit of complex synthesis, and also this kind of gives us a lot of excitement. So there we completed the total synthesis of these 3 molecules, and these 2 we are currently pursuing, and we are hoping it to be completed in coming days. So now I'll come to one story which actually I wanted to focus on. This was alkyne, because in our lab, while we are working on post-UGI modifications, we had a lot of alkynes, alkynoic acids and isocyanates sitting at lab, and we thought, what else can we do from away from UGI? So in this, actually, we looked for some of the vinyl sulfones, and you see 2 of the previous speakers, they spoke a lot about it. Raju, actually, I saw his structure. He has done a fantastic chemistry with idovinyl sulfones. So these sulfones are obviously widely expressed in a lot of biologically active compounds. Along with that, I would also like to mention one thing, that the compound we are looking for, PCSK9, also contains vinyl sulfone. So that actually interested us to take our chemistry in slightly different direction, so that we can explore a larger SAR around this particular molecule. So in this, there was a drug called bicalutamide. This is a drug used for prostate cancer. This is a very old drug, and this is a non-steroidal compound. This targets androgen receptor. So during COVID times, we're looking for different molecules that are actually being repurposed for managing COVID. So we found this particular compound was actually it got approval for phase 3 clinical trial in male COVID patients, because it was also known that this particular COVID-19 virus can actually affect the androgen receptor. So somehow that was also another interest that we thought, can we come to this particular drug using this particular sulfonyl acrylamide kind of thing. So keeping this idea in mind, we started looking for the literature. We found that this particular idovinyl sulfone is widely explored, and there are a lot of methods. Raju is making, I think, in kg quantities, and this was kind of a lot of methods were reported to make this, and then there were 2 reports that actually say that you can do a lot of nucleophilic addition by oxygen nucleophile as well as nitrogen nucleophile. So with this, they actually made this phenoxyvinyl sulfone, and then this was a vinyl sulfone. So these were the 2 reports that came in the year 2019 and 20. So looking at this scaffold, that we actually thought, can we do something with isocyanates here? So with this idea in mind, we thought, let's try the reaction of isocyanate with these idovinyl sulfones. And here, this was that we can go away from this C hetero bond to C bond, which is usually kind of gives us another approach for homologation. So keeping this idea in mind, we prepared this idovinyl sulfone. We added the cyclohexyl isocyanate, and we characterized the product. This 5A, the yield was good. We did little bit of optimization. We could isolate the product in with a nice yield, and it was kind of single product, I think, in the reaction. So we also tried to do this in one part, like we prepared this idovinyl sulfone in situ and added cyclohexyl isocyanate, and it gave the product. And what we found was this was giving exclusively Z isomer that we can't of another molecule in the series. So that actually kind of little bit of, because if you see the steric hindrance, this actually is kind of more here. So we were kind of intrigued why this particular sulfone is going to that direction. Anyways, we'll come to that story later. So this is for the students. If you see, isocyanide actually is it works as nucleophile as well as electrophile. So it attacks here in a kind of a Michael type of addition, and then elimination of this iodo happens. With that, you end up with this ammonium salt that actually gets quenched with water and use it sulfonyl acrylamide. So in this particular case, this was the isomer that was isolated, and this did not happen. So we later on, obviously, we moved to check whether this reaction works nicely with different isocyanates and then sulfones. And in one of the cases, we found that this particular ammonium probably got trapped with ethanol. So this was one byproduct in one of the cases we observed as a minor product. So this was further we checked with alkynes, and all these alkynes they worked well, and we used aliphatic alkynes and then these phenylacetylene derivatives as well. This was one of the example where we had biacetylene moiety at this phenyl, but somehow it reacted with only one, and then this particular product also we subjected for the second transformation that did not happen. So later on we started wondering what exactly is happening, why this particular Z isomer is the major product. So we proposed a cyclic intermediate. Here actually we proposed probably this O minus is getting reacted with this ammonium to create this cyclic intermediate, which is more stable, and because of that the water radiation can happen, it can open up and give the Z isomer. Somehow when we started creating a 3D model for this particular cyclic intermediate, we found this seems not to be stable at all. We also tried to check the possibility of formation of this through DFT and all. Again, we end up, no, this is not possible. So then we started looking for what could be the other reason. Then we end up, probably there is an intermediate which is in the open form. However, there is charge stabilization at the similar site. Because of that, probably this is staying at the same site. So we did S2O18 labeling experiment. So here actually we did the reaction and quenched it with S2O18. So we felt if this cyclic intermediate is formed, then actually this O18 can attack at sulfur as well as with this particular carbon, ammonium carbon. And if this happens, we will, so we actually did this, and then after this, we got the 18O incorporation of 56%. Then we did epoxidation. Actually, the reason was that if you can do epoxidation after that, you can do desulfonylation. And if there is O18 incorporation in this sulfone, then actually you will see reduction in the 18O incorporation. Somehow we did not see any reduction in the 18O incorporation, that remained intact to 56%. So that's how we felt that that also confirms there is no possibility of cyclic intermediate formation. Later on, we conducted time-dependent 1HMR experiment to see if this can give us some clue. However, this was not very supportive. But what we could see that the sulfone, this orthoprotons of this particular sulfone aryl ring were actually getting desealed and that were coming back. So we feel probably this two are lying in the same site because of some charge stabilization, and because of that, this is kind of favoring more to the Z isomer rather than the E isomer. So with this, we proposed a reaction mechanism. It is a similar thing what I explained in the last slide. So now actually we also moved on to check whether this particular approach works with internal alkynes. Somehow we found that this kind of vinyl sulfones that were derived from internal alkynes do not react under these conditions. So considering that, we thought that maybe we can develop some sort of palladium catalyzed coupling. There is a head type of coupling where we can make the this sulfonyl acrylamide. So somehow this reaction with a lot of optimizations worked fine, and we could get a condition and we could make this E-selective. We confirmed this through X-ray. There was no additional elimination product. So that's how this actually, the geometry remains intact. Later on, we tried the same with the iodovinyl sulfones that were derived from terminal alkyne, and we failed. Actually, here the method did not work. We actually got both the products. We got typical isocyanide addition product through palladium mediated coupling, and also we end up with getting the product that was arising from the addition elimination mechanism. So considering that, again we did optimizations. We changed the ligands. We found a very bulky ligand, DPPF, that could help us in making this product in a selective manner. So with this, again we could make all these E-selective vinyl sulfonyl acrylamides from both terminal as well as internal alkynes. So this is a typical hair type of mechanism. So I'll not go into the details of it now. We actually again look for the further transformations that we mentioned, that we wanted to use this particular method to synthesize bicarbonate. So we did epoxidation that could be done easily, and then we also opened it up. Fortunately, it opened from the right side that we wanted for bicarbonate, and after that we actually are now pursuing this method to make the bicarbonate log. So along with that, we also had the exhaustive desulfonylation by using LAH of this particular epoxide that ended this product 12 in 66%. Along with that, we could do reproduction of this tetrabutyl, and also we did hydrogenation to get the completely alkane kind of sulfones. So now with this, we moved on to check other congeners of the alkynes, and we found that this kind of phenyl propiolic acid kind of substrate also give the product. However, if we check this with the typical butanoic acid kind of substrate, it did not work, and we end up with getting this particular amide. So with that, we started because this was something which we cannot call a alkyne difunctionalization, because we are doing the reaction in 2 steps. We are going through iodovinyl sulfones. So we were still wondering, well, let's see what happens when you do not have this particular iodine in between. So we took the phenyl propiolic acid and this aryl sulfonate and then isocyanides and reacted with palladium chloride. So we immediately end up with this sulfonyl acrylamide. But this obviously we did not know from the NMR whether the sulfone is sitting on this or that. Obviously the NMR that we matched did not match with our previous products. So we took the X-ray and then we saw there is a complete reversal of regioselectivity. Now the sulfone was towards internal carbon rather than the terminal carbon, and amide was sitting on the terminal carbon. So this is the new story coming up, and here we have created a library. Now we are kind of doing a lot of studies to understand how this particular transformation is happening and why the isomer is not forming. So with this, I will summarize my work. So we actually could make this Z selective just by using typical addition elimination approach. we could do palladium mediated Higg type of coupling to create E-selective sulfonyl acrylamides. And this is a new story coming up where we have made a completely different radioisomer of sulfonyl acrylamides. So with this, I thank all the funding agencies, CSIR, CDRI, and then SERP and DBT for helping us through different projects. These are the students, and the work that I presented today was mainly done by Sasank and Monty. This is a picture of the group. Sometimes we went out a party. So, and this is a few pictures of Lucknow. You can come here to a beautiful campus of CDRI. This is a new campus. This is an old campus. We moved from a palace to a completely new palace of science. And these are a few of the old buildings at CDRI you can come and see, and places famous for food. So you are all welcome to CDRI and Lucknow. So once again, I thank you all for presenting. Thank you so much.
Thank you, Ajay. We may have one question. Okay, if no question, let us thank Ajay for the excellent talk.
So I would like to request Professor Punyam Murthy to present a memento to Professor A.K. Srivastava.
The last speaker of the session is now Dharmendra is here. He completed graduate studies at the Veer Bahadur Singh Purvanchal University, then PhD at NCL Pune. Following that, he did postdoctoral research at the University of Bodax. He returned back to India to accept BST Inspire faculty position at IICT Hyderabad. He was there nearly 6 years. In January 2019, he has moved to CDRI. He will speak about development of new methodologies using BMSO as a carbon source. Please.
Hello. Thank you, sir, for kind words. First of all, very good afternoon to all of you. I am very thankful to this organizer of this conference for inviting me here, and a special thanks to my friend Satpal Basra for giving me this opportunity to speak about my works, what we have carried out in last 6-7 years as an independent author. So the title of talk is that the development of new methodology using DMSO as a carbon source. So before going to my work, I would like to highlight that what kind of works we are doing. So in our lab, we are working on the azide chemistry. This is nothing but 1,1,3 dipole. So what we are doing, like we are taking this keto vinyl azide. So under photocatalysis or thermal reaction, it cleaves and it forms a vinyl aziridine. So that transforms. We have a lot of decent papers on this azide chemistry. Apart from this, we are also using isocyanide chemistry as a 1,3 dipole. So that is nothing but you can call it like a pseudo click chemistry. And next, what I'm going to talk here is it is the dimethyl sulfoxide. So I will next 15 minutes I will take you to this work. Apart from this, I'm also working on this anthranil chemistry. This is what yesterday Ramana sir has spoke about. And recently we have started this new adventure where we are using rhenium catalysis. So in last 4-5 years. So now it is taking baby steps, and we have some positive results on this one. Probably when next time I'll visit here, so I will show some results on this one. So coming to DMSO chemistry, it is a very simple and very basic chemistry, you can say. So DMSO is very well known as a aprotic, highly boiling point solvent. And I will come to this one, but apart from this, it has a various medicinal property. For example, it can be used as a NSAID, that is the what you call that non-steroidal anti-inflammatory drugs or agents. Apart from this, it has a property to penetrate our skin, so it is basically used in the painkiller. So please don't try it as a if you have some pain, because the concentration is very important. So, and we all know using it DMSO as a oxidant in oxidation state for where we oxidize alcohol to ketone. So most famous reaction is like Swarn oxidation. Coming to this solvent things, what I wanted to convey, that where is the problem? I mean, we are using DMSO, we are throwing, I mean, once it goes water into the water, it is very difficult to isolate. So I had this idea, actually, I was working on azapine rearrangement. If someone knows that one, this is nothing but 2-amino epoxide, where we open epoxide by trimethyl sulfoxylamide and cyclize, and we get hydroxypyrrolidine. So based on that, we have made like alexine and castorospermine molecule, polyhydroxylate molecules. So in that step, actually, for 1 millimole, I used to use almost 30 ml of the DMSO. So that was clicking in my mind. This is, we are going generating a lot of waste. So I had this idea during my PhD that whenever I'll get chance, definitely I will use it as a reagent, because there is a lot of chemistry known. So presently, just trust me, we have broken all the possible bond in DMSO. We have used it as a methyl carbon source, 1 carbon, 2 carbon source, and we have used like a SME source. Recently, we have some result where we have taken this oxygen of this DMSO in our reaction. So next 15 minutes, I will take you to my DMSO journey. So the first idea came into my mind that there is a alcohol. If you treat with this TMS chloride and DMSO, you will get the chloro. So we'll go to the mechanism. We'll see that the DMSO gets activated by this trimethyl silyl chloride, and you have this intermediate, and this TMSO comes out, and then the alcohol attacks. Here you will have this one. So this chloride, which is here, that attacks on this one, you will get chloro. So based on this, I was thinking, so can we take instead of TMS chloride, if we'll take the TMS azide, probably we'll have this intermediate sulfonium ion, and this azide may attacks here, and you will get this azide. So once you have azide, the simple reaction comes to your mind is the click reaction, because I believe that is one of the simplest reaction on the planet. So we also thought we'll try this reaction on this one. So based on this hypothesis, actually, we have taken this phenylacetylene TMS azide and DMSO in copper chloride in our initial result. In first attempt, we got this 27% of the yield. So we have done a lot of optimization. So finally, we came up where you can use either copper acetoacetate, that also gives almost 96% of the yield, and this copper on ferric oxide that is commercially available. You can use this also. It is like heterogeneous catalysis that also gives the 96% of the yield. So I have chosen this over the copper acetoacetate because it is very easy to isolate. Just put a magnet, and all this catalyst will come out, and you can use it for 5 times without much apprehension in the loss of the activity. So these are the some azide we have prepared. Triazole we have prepared. I will not go to details of this one. As you can see, I mean, just I have given some examples, like you can have electron donating, electron withdrawing, and you have this like a heterocyclic alkyne like thiophenol or sulfur. When we have taken simple NH, it was not working. We have protected this one. So extended polyaromatic aryl alkyne you can take. This reaction work with simple aliphatic alkyne and this cyclohexyl also. So having this encouraging result, so we thought to try some more reaction on this one. As we all know, that the DMSO in the presence of oxygen, it gives this sulfidinium ion, cation, whatever say. So we thought, okay, if you treat this with the simple inolate, I mean the acetophenone, probably we will get this one. And once we got this one, then probably we can have this one. Based on this, we tried this reaction, and just believe me, in the first attempt, we got this product in almost 23% yielding, and this one in the 56% after 12 hours. When we have extended the reaction time for 24 hours, the 74, this product was formed in 75% yield. So, you know, this preparation of this alpha beta unsaturated ketone is very difficult. Not very difficult, but definitely it has some problem, because you have to have aldehyde, then you have to do vinyl grignard, then oxidation. Else you have a here propiophenone, then oxidize it, then you can make this one. At this stage, we thought to publish this one, but somehow I did not get the confidence. But later on, some person, they have published this work only after my work. So at the same time, as I said, that we are working on the enthranil chemistry. So probably Ramana sir has given a lot of introduction about this one. So it has one nucleophilic center and one electrophilic center. So what happens if you do expose to this one to electrophile, and immediately this polarized worm, it break down and it give this free aldehyde. So based on this, we thought if you can treat this with this alpha beta unsaturated ketone, they will get quinoline. Based on that, we perform a reaction where we have taken acetophenone, DMSO, and enthranil in the presence of K2S28, and we got this product. And finally, we optimized the reaction yield. And so the best yield was observed was like 76%, and we have made a lot of entry. The reason was taking this project ahead because this compound is very important. You can see in the, it is a core structure of the beta-choline, and this ERW 1041, ERW 1016. So we have taken forward, this is a project, and we have prepared a lot of 3-keto-quinoline. You can see here, these are some examples, representative examples have been given here. This reaction work with the heterocyclic ketone also. You can see sulfur, NTS and oxygen, and the extended aromatic ketone you can see. And most importantly, the reaction work with the free hydroxy acetophenone also. But the yield was obviously it was little less than others. And there are some different enthranil we have employed in this reaction. So whatever we are doing, it should have some application. Yesterday, VK Singh sir was telling, so all methodology without application is is not complete. So we have shown this application of this methodology in the synthesis of this agricultural bactericide. This is a very important molecule. It is used for this grapes forming, where there is a disease called black rot. So it has been used there. So the previous method was people are using this Suzuki coupling and this diazepine and boronic acid to get this one. So we prepared in one step 67% yield. Before going to mechanism, we wanted to prove that DMSO is a methane source in this reaction. So we have performed several control experiment. You can see initially we have taken the acetophenone DMSO, and without enthranil, we got this one as I have mentioned. And then we have taken this ketone and treated with the enthranil under the standard reaction condition. We form the desired product in 80% yield, and we have taken this also, and then separately we have tried this reaction and 84% yield. And the last, we have tried this reaction in DMSO D6 because it is very easily available. So we got here 100% deuteration. Apart from this, the partial deuteration was observed here. The exact reason we don't know. And based on this, actually, we have proposed the reaction mechanism that the polarized form of DMSO attacks on this K2S28, and it forms this intermediate, and then the potassium mono persulfate gets knock off this one and form this sulfonium cation. So this is the reaction mechanism. I think that first you have this alpha beta unsaturated ketone undergoes to azamical addition, and it opens up this one, this give free aldehyde. This undergoes to annihilation reaction followed by elimination and oxidation to give this 3-keto-quinoline. So I have said that we have used azide as a nucleophile on this one, and then the acetophenone, like ketone, as a nucleophile on this one. So next idea came to our mind, we can use aniline. Based on that, we have performed a reaction where we have taken aniline and the oxidant, and we were trying to isolate this one. I know the all of our first were butyl, because this is a very difficult to isolate. What we were getting this polycyclic compound. So I was asking student whether we have got this one or not, but yeah, he was saying that we are getting in the mass, but proton is not coming. So I was asking that what we are getting in the proton. He said, sir, we are getting a 3-methylene group in DEPT. So we have taken this one and we have characterized that this is the compound. So how it formed, that is very encouraging. So it formed that iminium cation, then this way it goes and it formed this one. This is nothing but this is a Troger's reagent, or Troger's base. So it was enough to understand that the iminium cation is forming here. So based on that, what we thought, to trap this iminium with some dienophile. So first idea came to our mind to try this reaction with the alkyne. Just trust me, this in the very first attempt we got the 4 substituted quinoline in 79% yield. While we were writing this manuscript, we saw one paper. It is Chinese guy, some WY, and he has done the same reaction, but he has used some cobalt catalyst and silver triflet, and then the K2S2 and same condition. There is no much difference. Only thing, he has used these two catalyst and one additive. So far, I did not understand why they have used this one. So we have submitted this paper to organic lecturers, and it has gone through a lot of litmus tests, because almost 5 reviewers they reviewed and 4 of them accepted and 1 rejected. But then still it got rejected. Then it has transferred to JOC. Then again 4 reviewers they have reviewed. So almost 9 reviewers they have reviewed this paper, and really it's very surprising. So it has a very decent substrate scope. As you can see, different alkyne it can be used. These are some representative examples, and you can use different anilines to make. Important is that this polycyclic anilines can be used and it gives a decent yield. So again, to prove the application. So what we did, we have taken this particular compound, make this N-oxide in the presence of MCPBA, and you can treat this with the N-morpholin, and you have this one compound. This compound is known to show this one anti-proliferative agent. It acts as an anti-proliferative agent. And the reaction mechanism, again, we have performed some control experiment where we have taken alkyne, aniline, and we have taken this diphenyl sulfoxide, and there was no product formation observed because there was no methyl. So we have taken this alkyne DMSO, and in the presence of oxidant, we were getting this compound. And if you treat this acetophenone with this aniline, and under the standard condition, you will have this compound in 63% yield. And again, we have done the deuterium labeling experiment where you can see 100% deuterium at this place. So this is a very crucial. So based on this, because this is also forming, I mean, I was not sure whether it is forming or not, but with this oxidant, this product was forming when I separately tried without aniline. So based on that, we have given 2 pathways. One is the like simple 4+2 cycloaddition between this imine and this alkyne, or you can this can also be possible for formation of this 4 substituted quinoline. So having encouraging result, we thought, okay, we have used azide, we have used acetophenone like ketone, we have used amine as a nucleophile. One can use like this less activated methyl. I mean, this is the amide. So we thought if we can do this one, probably we'll have this one. So based on this, we have performed reaction where in the presence of K2S2 we did not get the desired product. So we tried several oxidant like K2S2, ammonium thiosulfate, or we have tried this selectofluor. We were not getting the desired product. But when we have used the acid, we got this product. And it was completely unexpected. Actually, we were expecting that we'll have this iminium cation, but it was going through this azide intermediate. and we have this pyrazolo quinoline. So the structure was characterized by the single X-ray, single crystal X-ray. And these are some substrate scope we have shown here. All the most of the compound they give decent yield. The important part of this work was like the application. So we have taken this as starting material, just did bromination here, and we can have this bromo compound and react this with the amino diethanol, and we will have this one. This compound is very important. This acts like a OLED. And if we'll go ahead and treat this compound with this triethylene glycol paraditosylate, make this microcyclic compound. This is acts as metal chelating agent. It is very useful for this Wilkinson disease, where the metal quantity increases in our body. So this actually useful for to catch that metals. So again, we have done some like control experiment to understand the reaction mechanism. It is almost same. When we have blocked this ortho position, we did not get the desired product. And again, when we have taken this methyl sulfoxide, then reaction product was forming, but in 55% to yield only. And again, the deuterium labeling experiment suggested that there is 100% deuteration at this place. And based on that, we have proposed a reaction mechanism. So this actually in this reaction, this DMSO gets activated by the this trifluoroacetic acid, and it forms this intermediate and undergoes elimination to form this one. Then this pick up this proton, and this CF3COO minus gets knocked off to give this sulfonium cation. So this is the mechanism. As I said, this reaction probably goes through this azadine intermediate to get the desired product. So I have done some more work on the DMS activation chemistry, but due to short time, I am not going to return to that. But this is the very recent work we have. So if you take the aniline, anthranamide and DMSO or acetophenone in the presence of iodine, you will have this quinazolone. So I think I have done the DMSO activation chemistry here and here also. So to club both, then and see that what happens. So when we try this reaction, and just we have this unalkylated quinazolone, and it happens that one of the compound, it is known for the analgesic property. So this molecule we can prepare in one step. And the work was actually completed in 2019, but I was in transition. I was moving from ICT to CBMR. Then the COVID came, and some work was pending, so it took a lot of time. But in between, there is Dr. Sandeep at IIM Jammu. He has published, I mean, this part of the work with the DMSO, and this part of the, so the novelty is the that we got additional this unalkylated part. So we have shown some decent substrate scope of this methodology, and we have done a lot of experiments to prove the reaction mechanism. These are here. I think it is almost same what I have explained earlier. In the presence of DMSO, we got this deuterium at this place also, and this is the reaction mechanism we have shown. So take home, let me conclude my work. So I have taken this simple solvent and used it as a reagent. So we have made this triazole, we have made this quinoline 3 ketoquinoline, and we have made 4 substituted quinoline. And this is the part which I have not included here. I mean, that's I have cleaned this with the inaminone also. And we have made this pyrazoloquinone, and we have also made this quinazolone, unalkylated quinazolone. So having said that, we have taken up the oxygen of this one. So there is an unpublished work where we have taken this propionophenone, and in catalyst, in the presence of some catalyst and oxidant, we are getting this product. So far we are struggling to get the oxygen 18 labeling. So finally we have one collaborator, and he said that he can give me this DMSO 018. Probably once we'll get, we can prove this one. And this is another work we have completed. And there are some more results. I mean, they are at the initial stage of the journey. So let me thank my students. So as I started as an Inspire faculty, I was having only 3 students initially. And this person, he was at Belgium. He came back. Now he's working at the Sai Pharma, and he was working exclusively on the DMSO activation chemistry, and he has completed his PhD. Now I have the group of 6 students. So presently, this Pushpendra, he is working on this DMSO chemistry. This is our small group, and yeah, these are some new joinees we have, and these both person they are working on our rhenium chemistry. Let's see. So I'm very thankful DST and SERB because this DMSO project is funded by SERB and the UP government for generous funding. Finally, I would like to thank you all of you for your patience hearing.
We are running late. To let us thank Dharmendra for the nice talk. Thank you.
Thank you so much, sir. May I now request Professor T. Punyamurthy, chairperson of the session, to kindly present a token of appreciation to Professor D.K. Tiwari. And with this, I also request Professor R.D. Kaushik from Gurukul Kangadi University to kindly present a memento to Professor T. Punyamurthy sir for his kind cooperation and coordination in this session. Thank you, Professor Punyamurthy and Professor. Hello. If you can hear me outside, I request you all to please be seated in the auditorium. We are about to begin our session. If you can hear me outside this auditorium, please come back and take your seats. We are about to begin the session. I also request to the participants of this session to please come over the stage and submit your PPTs for the talk. I request Professor Siddharth Sharma to please submit your PPT. It's a humble request. Those who are outside this auditorium, I'm requesting you once again, please come inside so that we can begin with the session. Ladies and gentlemen, we have with us Professor Anil Kumar. He will be chairing this session. Professor Kumar is working in Department of Chemistry, Birla Institute of Technology and Science. He has been awarded with Bronze Medal by Chemical Research Society of India. He has been also awarded with ISCB Young Scientist Award in Chemical Sciences by Indian Society of Chemists and Biologists. He has been awarded with Harrison Mackin Foundation Award 2012 by Acadia University, Delhi, Canada. He has published a number of research articles in the journals of national and international repute. I now request him to please take over the session further.
Thank you, ma'am, for the kind words. So this session we have 5 lectures. All are invited lecture, and each is of 20 minutes. I request all the speakers to restrict in the scheduled time. The first lecture is by Dr. Jivan Jyoti Panda. Dr. Jyoti is from Institute of Nanoscience and Technology, Mohali, and her group is working in different nanostructures for treating brain CNS associated disorders. And I request Madam to start.
Hello. Yeah. Yeah. Yeah, good afternoon, everyone. I would like to thank Professor Kumar for the kind introduction. And of course, I would like to thank Professor Satpal for giving me this opportunity to present my work here. So today I would be talking about blood-brain barrier traversing peptide amino acid. This is not working. Okay. The pointer is not working. Okay. Yeah. So today I would be talking about peptide amino acid based nanostructures and their utility as a blood-brain barrier traversing molecule so that we can use them for treating different brain disorders. So in this conference, we have been listening to a lot of talks in which people have been talking about synthesis of different active molecules, smart molecules. So through my talk, I would like to give you a flavor like how can we use the most important biochemical molecule, if I can say, the peptides and proteins into various biomedical applications. So why this is important, why it is important to develop nanostructures or nanotherapeutics for combating different brain disorders. And you all should agree with me that brain is the most important organ of our body. It controls anything and everything we do, so we cannot neglect it. So that makes it very important that we should protect it well, and we should try to, develop effective formulations for treating different brain disorders. So having said that, what is the problem? Why brain is so difficult to treat? So when we talk about that, there is a blood-brain barrier which is formed by the tight layer of endothelial cells which line the inner surface of the brain capillaries. If you ask me, the brain cells really love it because it protects them from harmful and noxious substances, infectious agents and all. But when we talk about brain drug delivery or treating neuronal disorders, it becomes really difficult and it gives nightmares to the pharmaceutical scientists working in the field of brain drug delivery. This is because it has been shown that 98% of small molecular drugs and 100% of large molecular drugs actually cannot cross through the blood-brain barrier. So we can make the drugs, we can make potent newer molecules to treat different brain disorders, but they cannot cross through the blood-brain barrier to raise the brain tissues. So they may be effective under the in vitro conditions, but they cannot cross through the barrier and reach the brain. So effectively, they are of no use. So that's why it's really become important that we should develop different strategies to enable the molecules to cross through this barrier. And conventionally, people use different strategies that include invasive strategies like the transcranial drug delivery or intranasal drug delivery. And sometimes, we go for chemically modifying the molecules so that they can cross through the membranaceous barrier. So, but when we do all this, there are lots of demerits, because when we go for invasive drug delivery, it's not good, because the person needs to be admitted in the hospital. So they need proper care. Many times it comes with a lot of risks. So it really becomes important that safe and effective therapeutic strategies should be developed to tackle these brain disorders. So in my group, we try to develop peptide and protein-based nanostructures for various applications. We synthesize the peptides and then we try to self-assemble them to form variety of nanostructures which can be used for different types of applications. We synthesize the peptides, they form vesicular structures, bowl-like structures, tubes, vesicles, so many different types of structures. Why peptides are important? Because peptides are our own biomolecules. So if they degrade, they will degrade into amino acids and they will not cause any toxicity. So that's why working with peptides is really interesting. And then when we talk about, chemical modification, with there are lots of functional groups, peptides can be easily and chemically modified to present us with tunable properties, desirable properties. So that's why it's really important that, so it was interesting to use these peptides for forming different types of nanostructures. And the most important thing for my research is that these peptides can cross through the blood-brain barrier by themselves. So there are lots of peptides, amino acids, which can cross through the barrier. So when you make a nanostructure out of these peptides, you don't need to work hard for making them, to cross through the blood-brain barrier. So that is an added boon which we always get while using peptide-based nanostructures. So this is our strategy. We use we synthesize the peptides using different design principles. We use solution and solid phase synthesis methods. And then we try to develop different types of nanostructures using them. And then these nanostructures are being functionalized with different blood-brain barrier permeabilizing molecules in some cases. And then we test their ability to cross through the blood-brain barrier using different assays, the PAMPA assay, in which we test their ability to cross through the barrier using membrane permeability assay. Then we go for different co-culture assays in which we culture brain cells, different types of brain cells to make the model of the BBB permeability. And then we try to use them for various brain drug delivery applications. So this is my group's work here. As I just mentioned, we use the peptide-based nanostructures for various applications in the field of brain drug delivery. So now I will take you through few examples, like maybe brain tumors and Alzheimer's, where we have tried to use the structures as a delivery agent. I can say specifically as a theranostic agent. So now what are theranostic agents? These are agents which can simultaneously act as therapeutic and diagnostic and imaging agent in one platter. So we develop one kind of system and it can serve all the three purposes. So that's our motto, to develop brain theranostic systems. So and we kind of try to use them for brain tumors, and here we are trying to develop different types of structures, photoresponsive, stimuli responsive structure, phototheranostic structures. So we develop structures which can respond to different pH conditions. We have this bowls, pH responsive nanovesicles. Then we have redox responsive systems, photoresponsive systems. We also have multi-stimuli responsive system which can respond to different type of stimuli. And then in a very recent piece of work, we have piezoactive peptide-based materials which can be used for anti-cancer therapy. So just to give you, I will try to take you through some of the examples. So this is a very interesting piece of work where we use an amino acid modified amino acid, I must say, and we use this to make nano bowl-like structure. So this amino acid could self-assemble to form this very beautiful bowl-like structures. And I would like to mention here that we used here microfluidic based self-assembly to make this structure. So unlike conventional assembly where we got vesicular structures, when we use microfluidic as the self-assembly, we got this bowl-like structure. And you can see and agree with me that these bowls can load a lot of drug-like molecules and can act as a very good carrier for the drug molecules. And after that, we tried to coat them with another amino acid, which makes them pH responsive. So these are the coated bowls. And when we subjected to different pH condition, you can see that now they are forming, these are the coated bowls, and you can see that there are different pores are being formed in this bowl, like the coated bowls, and through which these pores, the loaded drug molecules can come out. So these are our bowls after being loaded with doxorubicin. And then, through this pores, the doxorubicin can come out. And then we tested for their efficacy under cancer cells, the glioma, brain tumor cells. And you can see here that the glioma cells, the drug-loaded bowls exhibited enhanced killing and lesser IC50 as compared to the free drug. Then we tested their efficacy in cancer models. Here you can see this we developed in nude mice, and you can see that the tumor volume has significantly come down when we used our bowls, drug-loaded nano bowls for treating the animals. So that was interesting. In another piece of work, we use these bowls for combined dual drug delivery system. Here we use two drugs, the doxorubicin and curcumin. We loaded the bowls with both the drugs, and then we use the system, the whole formulation for combined chemo and photodynamic activity. So where doxorubicin acted as a chemotherapeutic agent, curcumin has got both anti-cancer and photodynamic activity. So we use them as a combined chemo and photodynamic agent. So these are also some of the works where we use modified design dipeptide-based nanocarrier system for pH responsive drug delivery and for redox responsive drug delivery. But in the interest of time, I'm not going into the details of this work. Anyone interested can follow these articles. So now I'll show you another very interesting piece of work where we developed multi-stimuli responsive system for anti-cancer activity. So here we synthesize this tetrapeptide in our lab. And this tetrapeptide is actually, it has got lysine, cysteine, and like aromatic groups containing phenylalanine, and also it can self-assemble to form 3 different types of structures. So that was really interesting because one peptide monomer can self-assemble into 3 different types of structures. So this peptide forms sheets, fibers, and nanoparticles. So generally, people do it in nanoscience. They use, but they have to modify the monomers with different groups. But our system was interesting because we can use the same monomer. We just tweak the self-assembling condition, and this system can shuttle between 3 different types of nanostructure. So we characterize these sheets and the fibers and the vesicles using various techniques. And to our interest, what we found that these structures are self-fluorescent in nature. So not only they form variety of structures, they're autofluorescent. And this makes our life much easier because if they're fluorescent, we don't need to tag them with something. They can be used as an imaging agent. They can enter, if they're entering inside the cells, we can track them very well without conjugating any other molecule to them. So these are the fluorescence confocal microscopic structures, images of our structures. And you can see that they're very beautifully being taken up by the cancer cells. So the cells are glowing very after taking up this structure. So we have different types of structures. We thought, will they show different type differential uptake behavior in the cells? So that is what we got. we observed that while the sheets and the fibers went inside the cells through macropinocytosis, the vesicles went through the cell to the cell through endocytosis. Means these are different types of mechanisms through which nanoparticles or any other molecules are taken up by the cells. So since they are having different shapes and structures, so they exhibited differentiated cellular uptake behavior. So that's also interesting to note. And we also found that they could kill the cancer cells. We loaded them with doxorubicin and they could kill the cancer cells based on their shape. Like we found that the maximum killing was observed in nanosheets. So that was again interesting to note that actually the shape is impacting their behavior so much. So after that, we also work with peptide metal hybrid structure. So this is one piece of work where we try to develop theranostic systems. We use levodopamine. It's a small molecule, and this levodopamine can cross through the blood-brain barrier. It is the precursor of dopamine, which is being used in Parkinson's disease. It's dopamine cannot cross through the barrier, but levodopa can cross. And interestingly, we found that this levodopa can self-assemble to form this very beautiful structure. And these structures, we synthesized one anti-glioma chimeric peptide in the lab, which can cross through the blood-brain barrier and has got anti-glioma activity. So we loaded this levodopa structures with that peptide. And since it has been reported that polydopamine and levodopamine has photodynamic activity, these structures, when this polydopamine self-assembles to form nanoparticles, they have photodynamic activity, which means they can, if you shine light on them, they will release reactive oxygen species, which can be used to kill cancer cells. But their activity is little feeble, so we loaded these structures with another dye, NIR responsive dye, indocyanin green. So this is our complete multifunctional nanostructures, which can kill the cells through anti-cancerous, chemotherapeutic effect, photodynamic effect, photothermal effect, and can label this, this can be used to track the tumor. So here you can see this molecule can cross through the blood-brain barrier, and we did in vivo imaging system to track them inside the brain, and you can see they stayed there inside the brain for quite a long amount of time. And then they killed the cancer cells. We tested them in 2D and 3D models, so they're very effective in killing the cancer cells. So having said that, this is a phototheranostic system for the brain tumors. Similarly, we developed anisotropic gold nanoflowers. This also was synthesized on levodopamine, and that we use for combined PTD and PDT theranostic agents for glioma. And in this work, this is just we published in last month, we synthesized a design peptide carrying a dihydroamino acid in this, the dihydrophenylalanine, and this form self-assembled to form nanotubes, and these tubes, they exhibited piezoactive behavior. So which means when you subject them to mechanical stress, they release, they get electrically active. And we use this piezoactive nanotube for killing cancer cells, the glioma cells. So that was a glimpse of the work which we carry out for brain tumor, and we do work in the field of Alzheimer's. How much time I have? Yeah, so we develop different types of nanostructures which can act as anti-aggregating molecules for Alzheimer's anti-amyloid therapy. So we use tryptophan as a scaffold to make these structures, and we tested their so these are the structures. They exhibited anti-aggregating behavior. We tested them in the peptide-based models we developed in the lab. They protected the neuronal cells from, amyloid is a protein aggregation disorder. So they protected the neuronal cells from A-beta aggregated induced toxicity. And we tested their efficacy in animal models also, where we found that tryptophan nanoparticles, the nutrient, the amino acid which we use as a nutrient, and it's the essential amino acid, these nanoparticles can protect the animals from, streptozotocin induced cognitive decline. So similarly, we developed dopamine and tryptophan nanoparticles, and they also behaved as a theranostic system for amyloids, Alzheimer's.
You can summarize,
Yeah,
please.
so just to summarize, we developed different types of peptide, amino acid, and protein-based nanostructures, and we use them for various application in brain drug delivery, cancer, and other fields. With this, I would like to thank my students. They are the real workhorses, and my collaborators, Professor Chauhan, Professor Kundu, Dr. Bhanu, Dr. Dipankar from INST, Dr. Shubha Shukla, she helped with me in the amyloid studies, Professor Vidya Kampela, University of Colorado, USA, they helped with me some with the photothermal work. And of course, thanks are due to funding agencies. And thank you all for your kind attention.
Thank you, Dr. Panda. The floor is open for one question. Any question from students? If not, let's thank Dr. Panda for a nice lecture.
Thank you, sir. Thank you, Professor Jyoti. I now request Professor Anil Kumar to kindly present a token of appreciation to Professor Jivan Jyoti Panda.
Our next speaker is Dr. Sandeep Murarka. Dr. Sandeep has done his MS from Rutgers University, USA. Then he moved to Germany to do his PhD. This is WWU Münster. And then he did his postdoctoral work at Max Planck as a Max Planck postdoctoral researcher in the lab of Herbert Waldman. He did a short-term work at industry, Syngene International Limited. In 2017, he joined IIT Jaipur as an assistant professor, and since 22, he is an associate professor at IIT Jaipur. His work interest is in development of chemoselective and sustainable transformation towards synthesis of bioactive molecules. I request Dr. Sandeep to share his work. Dr. Sandeep.
Okay. Hello, everyone. So thank you, Professor Anil, for your very kind introduction. So it's been a pleasure to attend this conference. We have had a couple of series of great talks. So thank you to all the speakers for set the stage. So at the onset, I would like to thank Professor Satpal and the rest of the organizing team for organizing this such a wonderful conference and giving me the opportunity to attend this. So today, in the next 20 minutes, I would like to talk to you about the photodecarboxylative alkylations involving redox active esters and the recent developments in our field in this area. So to begin with, let me first introduce the research program we are trying to pursue at IIT Jodhpur. So our research basically focuses on sustainable organic synthesis, where our prime motto is to develop novel activation modes to activate unsaturated pi bonds and unreacted CH bonds. To do so, we are using chemical tools such as transition metal catalysis, photocatalysis, metal-free catalysis, and of course, radical chemistry. Having these chemical tools in hand, we should be able to have synthetic methods for CH functionalizations and cascade annulations, which in turn should enable us to construct a compound library that should involve drug-like molecules and based on biologically pre-validated heterocyclic scaffold. Finally, this chemically and structurally diverse compound library will be screened in a biochemical assay or in a cell-based screening in a collaborative approach to find that particular compound which will infer the biological activity which we desired for. So if we look into a pharmaceutical company and the drug discovery program, we would realize that the venerable Suzuki-Miura cross-coupling reaction and classical amide-Formis reaction contributes almost 50% of the synthetic methods to develop drugs. Now, the reason behind this intriguing phenomena, as said by Georgi Kesru, who is a medicinal chemist at Hungarian Academy of Sciences, is the robustness of this chemical transformation. To add to that, the easy availability of the corresponding reacting partners and the cheap chemical reagents also contribute to the robustness. Now, in the heme side, we can also think having limited number of synthetic methods and disconnection approach may limit the accessibility to the chemical space. So it would be always wise to develop novel disconnection approaches for the exploration of chemical space, which is untapped. Now, to look at the wish list of medicinal chemists, it would always be a method for late stage fluorination, late stage alkylation, specifically of heteroarenes, development of methods for the modification of heterocycles and CSP3 coupling. Through this particular research program, we have tried to address these last three challenges, where we are focused on development of novel and chemoselective methods for CH functionalizations, and as well as cascade annulations for the construction of different sort of heteroarenes that are biologically pre-validated and embedded in many biologically active compounds. In this slide, you are seeing a couple of drugs which are already marketed. Interestingly, if we would like to disconnect these molecules in the highlighted bond as it is shown here, we would not only need a robust synthetic methods, but also the corresponding alkyl surrogate, which is easily available. Now, a quick browsing through the literature and the availability of the corresponding alkyl surrogate would tell us that alkyl carboxylic acids have the broad representation among all the alkyl surrogates, such as alkyl halides and alkyl metals. But counterintuitively, the methods involving alkyl carboxylic acids, especially those which goes to radical intermediate, are literally scarce. Although there are challenges associated with alkyl halides, such as their instability and as well as like propensity towards beta hydride elimination, their methods are much large in the literature. So technologies, especially radical technologies that leverage these advantages of carboxylic acids, such as versatility and stability, would always be highly desirable. Now, the decarboxylation of carboxylic acid to generate the corresponding alkyl radical, the classical Hans-Dicker reaction, dates back to early 1980s, where such kind of decarboxylation can be done with scrupulously dry silver salt under high oxidative condition using K2CS2O8. Of course, these exhaustive reaction conditions limits the method of this, limits the scope of this method, especially with regard to functional group tolerance. One can also prepare the not so stable and not so easy to handle Burton ester and then subjected it to classical tin hydride reduction reaction under AIBN condition. But of course, no one want to use the toxic tin conditions. And of course, in modern day, we have seen many methods for the decarboxylative couplings involving carboxylic acids using precious metal-based iridium catalyst. The method works well, but the strong oxidation potential required to oxidize a carboxylate anion, of course, deals with the problem of functional group tolerance. So having these challenges in this mind, when I started my independent career, I became interested in the chemistry of redox active esters, or to be precise, N-ethyloxythaliamides. So these N-ethyloxythaliamides can classically be prepared by simple coupling methods between feedstock carboxylic acid and N-hydroxythalamide. These N-ethyloxythaliamides have readily been used as acyl donors towards the synthesis of amides, ketones, esters, and so on and so forth. Not until 1990, when Okada discovered that these molecules are predisposed to accept an electron into the pi star orbital, which then jumps into the sigma star orbital, leads to the reductive fragmentation and form eventually alkyl radical through concomitant decarboxylation and elimination of thalamide. These alkyl radicals can participate in a plethora of cross-coupling reactions and has immense applicability. So I got fascinated with the chemistry of N-ethyloxythalamides and decided to sum it up in a concise review in Advanced Index Catalysis. Interestingly, this review was very well taken by the scientific society, and within 2 years, we have got more than 250 citations. In the subsequent 2 years, almost this field got exploded and 300 research articles were published, which compelled us to write the next comprehensive review, which was published in 2021 in ACS Catalysis. So all this while browsing through the literature, what we have seen that mostly these alkyl radicals generated from N-ethyloxythalamides were added in electron deficient alkene under photoredox catalysis condition using metal or organic photocatalyst. There are many leaders in the field, of course, starting with Okada, who is the pioneer, then Larry Overman, Fu, Liao, Megers, and so on and so on. They have contributed immensely. So, but our group mostly focuses on the rapid increment of molecular complexity. So this particular paper by Larry Overman attracted our attention, where he develop a cascade involving addition, cyclization, and fragmentation events, where he could couple this trans hydrogen fragment with this butanolide to get this advanced intermediate, which was taken over to the synthesis of chromodorolide B. So we asked ourselves, can we take inspiration from this and develop a series of cascade methods to access to the heterocyclic compounds? With this vision in mind, we first documented a cross-coupling reaction between allyloxybenzaldehyde and N-acetylhalides to access 3 alkylated coumarin derivatives, chromanone derivatives. The method was quite broad, and several functional groups were tolerated under the reaction condition. Next, we tried to address the challenge of preparation of Z-selective desuccinimide derivatives, and we coined a redox active esters, which is derived from aryloxy acetic acid and HPI ester. So we could develop an organophotoredox catalyzed cross-coupling between maleimide and such an alkyl NHPI ester to develop the corresponding Z-selective aryloxy alkyl leading succinimide derivatives. Lately, we have also looked into the problem of addition of alkyl radical in electron-rich alkenes, and accordingly we developed a cascade annulation between alkyl NHPI ester and this electron-rich alinamide derivatives to eventually access 3 alkylated indoles. So far what I have shown you, that we developed mostly methods which deal with organophotocatalysis. We have tried to address the challenge of addition of alkyl radical to electron-rich alkenes, and also we have tried to access compounds that are based on biologically pre-validated heterocyclic scaffolds. So about the future directions, we were looking for the alternative activation modes for NHPI esters, and that's when we became interested in the chemistry of electron donor acceptor complexes. So the 2 stories which I'm going to share with you, that fundamentally depends on the chemistry of EDA. So before I share those stories, let me first briefly talk about what is EDA. So in an EDA complex formation, a suitable donor comes in close proximity with a suitable acceptor, and in solution, once they come closer, there is a charge transfer takes place from the donor to the acceptor, which is arrested or which is recognized by simple color change by naked eyes, and as well as a charge transfer band in the visible region. Once you irradiate light on this EDA complex, there is a single electron transfer takes place, which leads to the formation of the radical ion pairs. Now, if the kinetics of the radical ion pair is proper, then one can lead to the formation of product formation through some reactive radicals. But what problem often encounters in this is that the back electron transfer is quite fast. So what happens if the product formation, if it is Kp, is slower than Kbet, which is a back electron transfer, then it goes back to the ground state EDA complex formation, and you do not get the product which you have wished for. To circumvent these issues, one can do, they can attach this leaving group to the acceptor moiety, which eventually, once it forms a radical anion, then classically through AZN1 mechanism, where this electron jumps to the sigma star orbital, and then eventually through reductive fragmentation leads to the formation of the product. So now coming back to the story, the first scaffold which I would like to share is this fused tetrahydroquinoline derivatives. If we look into the literature, we will realize that the methods mostly depends on non-photo-induced and photo-induced, but in general, conceptually they depend on oxidative cross-coupling, or rather to say radical annulation between these NN-dialkyl anilines and maleimide derivatives. So most of these methods involve expensive and toxic metals, stoichiometric strong oxidant, and high temperature. But the most drawback is that the application of symmetrical NN-dialkyl aniline. Whenever one takes the non-symmetrical NN-dialkyl, you would get the corresponding possible regioisomers. For example, in this literature, you can see that when they took N-methyl aniline, they could not distinguish between a proper regioselective oxidation that eventually lead to the formation of the possible regioisomers. So we wanted to solve this problem, and how? So we thought that if we have a site-selective reductive auxiliary into the NN-dialkyl anilines, which can actually accept electron, then we can have a suitable donor easily available, such as tertiary alkyl amine, that can form a EDA between the lone pair and the pi star orbital of the NHPI ester. Now, upon irradiation of this complex with visible light, that should lead to the formation of the amino alkyl radical in a site-selective fashion, and then we can discriminate between the sites which are possible. And then eventually a 4+2 formal radical annulation should give rise to the formation of regioselective tetrahydroisoquinoline derivatives. Such a method would be metal-free, would be regiospecific, and most importantly would lead to difunctionalization of alkenes, which are not so trivial. Now you would appreciate this design even more when you see the latest state of the art in that field. People like leaders in the field like Molander, Bach and Melchior, they have come up with different donors, and they have shown that these donors can form EDA complex with NHPI ester, but most of their methods leads to the monofunctionalization of alkene, whereas our method leads to difunctionalization and formation of heterocycles. Now, we quickly change all the parameters which are involved in the reaction and find out that 3 equivalent of DiPA in presence of 440 nanometer LED is the suitable for the reaction. Few control experiments reveal that donor DiPA and the photo irradiation, constant photo irradiation is necessary for the success of the reaction. Now, having this optimized condition in hand, first we explore the scope of the maleimide. And as expected, several substitution patterns on the phenyl ring of N-aryl maleimide were tolerated. Electron-rich, electron-deficient, ortho-meta-para substitution participated in the reaction. N-alkyl maleimide also were tolerated to the reaction condition, and we obtained our desired product in good to excellent yields. Then we varied the scope of the aniline partner in the alkyl NHPI ester, and we found out several substitution patterns can be attained there, and the reaction could also be carried over by different N-alkyl substituents. Most importantly, other than the primary alkyl NHPI ester, we can do the reaction with secondary alkyl NHPI ester also. So basically, through this example, I'm trying to tell you that the problem which you saw, that the regioselectivity issues, which we can solve it, and this compound could not be prepared by any of the earlier methods. And so to sum up this story, so we developed a method to access regioisomerically pure tetrahydroquinolines, and the reactions were carried out under mild condition. Now, to begin with the second story, we recently became interested in the chemistry of azauracils. The reason is obvious. They are absolutely present in many nucleosides and as well as in natural products and marketed pharmaceuticals. Now we know that alkylation plays an important role in modulating the medicinal chemistry of a particular compound. Especially, we all know the effect of magic methyl, right? So what we thought that can we do alkylation of these azauracils in a completely regioselective fashion. So when we looked into the literature about the methods which are available, we found out there are two methods for the non-photo induced condition. The first method involves this trialkyl sulfoxonium salt, and that happens at high temperature in the presence of a strong base in super stoichiometric amount. Moreover, this method only could do alkylation of primary alkyl. The second method was developed by the same group. In this case, they used these dihydropyridine derivatives. In this case, the scope of the reaction was quite broad, but probably the reaction condition were a little bit exhaustive, and the alkyl partner was also taken in quite excess. There is one method for the photoinduced condition, but that allows alkylation only like having alpha heteroatom substituted alkyl group. So basically, this method is applicable only for alpha alkoxylation and alpha amination. And one biggest drawback of this method is that in case of alpha alkoxylation, they have to use the alkyl partner as a solvent. So we asked ourselves, can we do alkylation of this azauracil by taking NHPI ester as the alkyl surrogate? And if we want to make this method more adaptable, cheap, and widely applicable, we have to come up with an activation method. So we find out that if we can get an easy donor which can coordinate with alkyl NHPI ester, then we can actually serve the purpose. So at this point of time, this great reaction developed by Sang and Fu, which was published in Science, captivated our attention. So in this particular reaction, they did the alkylation of silylenol ether using cheap and commercially available donors such as NAI and PPS3. The reaction goes to the formation of such this charge transfer assembly complex, which upon irradiation of a suitable light leads to the generation of alkyl radical. So having this conceptual design in mind, we started the optimization process, and gladly, after quick browsing through all the reaction parameters, we found out that sodium iodide PPS3 and timida is required for our reaction, which was not required in the Sang and Fu's work. We do not really understand the role of timida till now, but through control experiment, what we have found out that timida indeed plays a big role in the formation of the EDA complex, and we suspect that it also stabilizes the transiently generated iodine radical. Nevertheless, to give you the glimpse of the effect of timida, without timida, we got 14% yield. Further control experiment revealed the indispensable role of sodium iodide PPH3 and constant irradiation of light. Finally, we explored the scope of the reaction, and in contrary to the reported literature, this method is absolutely can encompass all sort of alkyl group, starting from secondary alkyl, activated heteroatom substituted alkyl, and also sterically congested this adamantyl derived alkyl radical as well. We could easily plug in many pharmaceutical derived from tertiary alkyl radicals such as gembifrozil. Moreover, more challenging 1-degree alkyl radical, which has got a very less lifetime and more challenging to integrate in your system, can also be used so that we can, as you can see, there are many primary alkyl radical derived from a range of fatty acids, and as well as this pharmaceutical isozapac could be integrated on this azauracil scaffold. Finally, we explored the scope with azauracil, and in this case also several reactive functional groups such as ester and ketones were tolerated. More importantly, we could do the reaction with monosubstituted azauracil as well. And then we saw, tried to understand that if we can integrate many biologically active compounds and natural products or not. So we found out that azauracils embedded with monoterpenoids such as citronellol, menthol, and isoborneol, and as well as nucleosides could also be alkylated under our optimized conditions. So, as usual, we tried to see if the method can be commercialized, if the method can be scalable. So we found out that the method is indeed scalable, and we can easily prepare multi-gram compound under the optimized condition without having any effect on the yield. Once we started to do control experiment, we found out with tempo, the reaction was absolutely shut down. And when we used another radical scavenger such as this alkene, the yield plummeted to 60%. Of course, we detected these transient adducts by HRMS. And we performed some photophysical studies to get some better insights into the reaction. So for the first one, we recorded the UV visible absorption spectra of the individual reaction components and as well as the mixtures. We found out that when NAI, PPS3 and redox active esters was used together, we found a clear bathochromic shift. And when timida was added, the peak got further shifted to the visible region, which clearly demonstrates that there is a quaternary EDA complex formation between NHPI ester, NAI, PPS3 and timida. We also realized that constant irradiation of light is essential for the success of the reaction, and determination of quantum yield, which is like 0.54, clearly ruled out the possibility of any radical chain mechanism. So based on this wide range of control experiments, we predicted that the reaction goes to the formation of this charge transfer assembly, and then upon irradiation of light, it leads to the formation of alkyl radical and an iodinyl radical, which is probably stabilized by timida. Finally, this alkyl radical undergoes addition on azauracils to form this nitrogen-centered radical, which upon oxidation by this transiently generated iodine radical give rise to the formation of this nitrogen-centered cation, which upon deprotonation by timida or the phthalimide anion leads to the formation of the desired product. So, in conclusion, in this story, we have developed a alkylation for the azauracils and tried to solve an unsolved problem. Finally, this brings me to the non-scientific or scientific last slide, where I would like to show you that I hope I was able to convey you in the last 20 minutes some of the recent developments in our group in the area of NHPI ester chemistry. We are also extensively engaged in the field of hypervalent iodine chemistry, where we are mostly focusing on the chemistry of diaryl iodonium salts and as well as carbene chemistry and medicinal chemistry. So finally, I would like to thank all my group members, PhD students, project staff, and MSc students. This work was entirely carried out by Sudhir Hota and Satya Prakash Panda. So Satya is here, he's presenting a poster, and Rupasri also has recently joined the team of Redox Active Ester. And finally, I would like to thank IIT Jodhpur for the infrastructure facility and SERV and CSR for the funding. And thank you all for your kind attention.
Thank you, Dr. Sandeep, for a nice lecture. We can have one quick question.
Thank you, Dr. Sandeep, for your nice presentation.
Thank you.
So my question is, for most of the photochemical reactions, people use this 3 equivalent of DPA and DMSO combination.
3
equivalent of DPA. Diisopropylethylamine. So in fact, if you look into the mechanism, mostly one electron being transferred. So possibly 1 equivalent is enough. Why everyone use 3 equivalent? So is there any reason behind it?
Well, DIPA is a base classically, right? But it depends on the photo kind of reaction you are doing. If you are doing a reaction where the reaction is going through the inductive quenching cycle, then the DIPA is reducing the catalyst. So there DIPA has a different role. If your DIPA is not involved in the photocatalytic cycle, rather it's just serves as a base, then that's a different thing. So your question is quite valid. In fact, in our case also, in the first story, this alkylated chromanones, we also had to use high equivalent of DIPA. But although through control experiments, we did not find any role of DIPA in the photocatalytic cycle. So we concluded that it is acting as a base, but we don't know that where this DIPA is utilized. So I personally feel probably what may happen is that some of the DIPA get lost through some undesired photochemical pathway. And that's the only answer I have at this point of time. But yeah, that is true. But in some other projects, we are using actually less equivalent of DIPA also. So as of now, no clear cut answer.
Okay, and Dr. Sandeep, I have a very small question.
Yes.
In your NHPI melamide work, the yields are around 50-60% for many of the derivative.
You mean this,
the melamide making a cyclic heterocycle.
The fused tetrahydroquinone. Okay.
So can you shed a light on that, like in this type of reactions?
Yeah, so the problem with that reaction, what we have found out is that particularly designed redox activator. So we designed that redox activator particularly for this project, and we initially were very excited. And I can tell you that, to be very honest, we found out these redox activators are not very stable. So in many cases, we found out these redox activators are going decomposed. And also some of the redox activators, we could not even detect the HRMS. So the reason behind, and that is why we have to use also high equivalent. If you saw that we have used 3 equivalent. Yes. So this is one of the drawback of our method. And probably this also explains that some of the cases why we have less yield, because probably we have the less availability of the NHPI ester. Yeah.
Thank you. Let's have a good round of applause for Dr. Sandeep for a nice work.
Thank you, Dr. Sandeep. Thank you, Professor Anil Kumar. May I now request you to please offer him a token of appreciation for his wonderful lecture. You just. Thank you very much. May we have the next lecture.
Our next speaker is Dr. Ajay Kumar Shah. He is my colleague at BITS Pilani. Dr. Shah did his MSc from IIT Kharagpur, moved to IIT Bombay for PhD. He then moved to Japan for a postdoctoral fellowship, where he was also a JSPS fellow from 2003 to 2005. For a very short period, he also joined Syngene International. So that's a link between two speakers. And then he moved to BITS Pilani in 2007 as an assistant professor. Currently, he is a professor of inorganic chemistry over there. And Professor Shah works on carbohydrate chemistry, basically metal complexes of carbohydrates. I request Dr. Shah to present his work.
Thank you, Dr. Anil Kumar, for your kind introduction. So, unlike earlier presentations, my presentation will be slightly most on inorganic and little bit organic portion. So, my work mainly focused on basically biologically inspired chemistry. So, the copper, as we know, copper is the third most abundant D block element in biological system. It is present as type 1, type 2, and type 3 proteins. Now, type 1, 2, and 3 is just based on their geometry, nuclearity, and binding mode. If I come to outside the biological system, then copper has been used by in number of researchers for various organic reaction, whether it is a coupling reaction, click chemistry, or a lot more, which is summarized over here. So this is the biological and chemical background of chemistry. If I come to the sugar, so glucose is basically the one of the prime product of nature, and my research work moves around this glucose. Now, glucose looks like simple molecule, and this is good for organic work because it is multiple hydroxyl group for doing a lot of chemistry. But once we are trying to do anything on this, then the major problem is the time we put into the solution, solvent will be always water, and the time we put into water, it will keep cyclizing. And in cyclic mode, it will have 6 number and it will have 5 number with the OH orientation up or down with respect to this cyclic ring. So that we will have alpha and beta form. So even though we are starting with one compound, effectively we are getting 5 different compounds in the solution, and all 5 have equal probability of binding the metal ion. So that way there is a complexity, and so in organic chemistry in general, do either modification by protecting the solubility or by selective modifications. So in my work, what we do is generally we protect the hydroxyl group partly and then modify at the C1 position. The necessity of modification at C1 is we know that in DNA and RNA, all sugar and base connection is in such a way that it takes beta orientation. Here also, whenever we do this glycosylation formation, it goes to the beta form, and then we can have one selected H and one in the solution. So now it will give me whenever we do the reaction. Now in this ligand, you can see it will have a nice metal binding core, and this can be used in complexing n number of metal ions. And that we have proven by reacting with various metal ions. So it binds with vanadium, molybdenum, which are the oxo metal ions. Beyond, we can bind with first order transition metal like nickel, copper, zinc. So this way, once we modified the sugar, now it is a very good ligand and it gives crystal clear complexes. Now, interestingly, when we explored the copper chemistry, what we found is complex is discrete. Depending on the recrystallization process, we are getting different geometrical environment. For example, here you can say both the copper in a square planar, here one in a square planar, one in a square pyramidal, here both in a square pyramidal. This in general conveys that copper is flexible with respect to substrate binding, and that can be utilized for catalysis. So that way this compound can be a good candidate for catalyst. Second aspect is, you see here, this part and this part of the ligand, they are coming closer over here. So another chemistry suggests, what if one can put here the bulky group? Then the structure will be like this, or it will break. It will give you a mononuclear complex. So when we do that one, what we found is, when we put here the tributyl group, rather than mononuclear, we got a trinuclear complex where the two units move farther apart by incorporating a copper ion over here, and this copper is only bound with the alkoxy sugar moiety. And we know that pKa of alkoxy sugar is very high. It is very big ligand, but still it is able to hold this copper, and this copper is now holding through encapsulation. If you see here this distance, because of this methanol binding, this distance is 2.5. So if I put here one hydrogen, it is very difficult to say that hydrogen belongs to methanol or it belongs to this sugar wedge. So we have explored who is belonging that wedge, and it is one different chemistry. So I'm not going into that one. What I am concentrating on is the chemistry of dinuclear complexes, because when we look into the biology, we found that at a enzyme is based by oxygen, and here this copper separation is 3 angstrom. In our complex also, whatever the dinuclear complexes, we found this copper separation is also less than 3 angstrom, 2.5 to 3 angstrom in between that. So we thought, can it act as the functional mimic of catecholase? For that, we did the reaction on a model compound, that is not going to substitute this catechol. If it oxidizes this molecule, we will have this one. And then if I talk about the difference in the spectroscopy, this compound gives a absorption peak at 400 nanometer. And in general, all researchers monitor that peak. So that way, we also did this reaction. Generally, people report this reaction in methanol, but our compound is not soluble in methanol. So we took a mixed solvent DMSO and methanol, and then we did this reaction. And we found that yes, this compound is able to oxidize it. There was a development of peak at 400 nanometer. This is the kinetics of absorbance with respect to time, and this gives K catalyst roughly 58.4 per minute. So that way we are able to establish that it is having oxidizing ability. And once it is proven, then we explore this or extend this for the organic substrates. So for organic substrate, we took the literature reports and whatever the literature input, based on that we proposed this protocol. And with this protocol, in this one, solvent we used as ionic liquid for the better substrate isolation. And we could able to get selective oxidation of primary and secondary alcohol into corresponding carbonyl compounds. During this oxidation process, we did not get traces of acid using various spectroscopy. So that way we can claim that this is basically the selective oxidation process. In this result, again, we found it oxidizes primary and secondary alcohol. However, the product yield for secondary alcohol is slightly lower than the primary ones. So this was the oxidation. Now for our compound, only problem was the solubility. So what we thought that if we can put some substitution, which is aliphatic, then it may dissolve in organic solvent and we can do more catalytic work. So we developed our plan for amphiphilic ligand, where we basically developed this type of molecule so that this can again interact over here, and we will have a polar end and non-polar, it can act as amphiphilic ligand, and this may enhance the solubility. Once we got this ligand as per our plan, now it is soluble in almost all the organic solvent, starting from pentane and hexane. So that is one of the good aspects. But when we did again the copper action, as expected, it also given a dinuclear complex, but we again the solubility problem came, and then this compound is again soluble only for DMSO. So solubility thing was not overcome in this process. Well, so this is the crystal structure of this molecule. Here also you can see we are having a square pyramidal geometry with the copper. So one can plan to knock out this, so substrate can bind over here. So we assume that it will have catalytic ability, and since we have done earlier case oxidation, we tried other reaction. So in this time we searched for A3 coupling reaction to give this propazyl amines. Why we selected this type of reaction is because we are doing sugar chemistry. So a genuine question comes, what is the advantage of chiral centers of the sugar moiety? So we thought if you do this, then this center will be a chiral center, and if there is any stereoselectivity in the reactions. So for the catalytic reaction, first we did the catalytic optimization. So here is basically the solvent optimization, where we found that under the net condition, it gives the best result in 6 hours. So after solvent optimization, we went for the catalyst loading. So catalyst loading, you can say for 5%, 2%, almost equivalent yields. So we assume that 2% catalyst loading will be optimum. And then really catalyst is required or not. For that, we did some alteration, either the temperature or without copper or without ligand, only the cupric acetate. If it is cupric acetate, yield is only 12%. With ligand only, no reaction. With 2%, if I go higher time, it has enhanced. So that way, these all the control reaction suggest that catalyst requirement is necessary for this reaction. So once we had optimized reaction condition, we did under this condition the synthesis of various products. Roughly, we could synthesize 20 different compounds, and almost all compound were having stereoselectivity. If I talk stereoselectivity, if I see this compound, so this compound, if you see the starting material, then this unit, piperidine, will be optically inactive. Okay. And if I do this reaction with any chiral catalyst, then the chiral formation happens. And as per rule of thumb, if I start with a chiral molecule, I should get a chiral behavior. So that way, this gives me racemic mixture, and we are having the HPLC, chiral HPLC with roughly 50-50 mixture. But when we use our catalyst, we can see out of these two, only one here, it means we are getting chiral selectivity. Same is true if we take this compound. So this is basically what we used as the commercial compound, which was a chiral, means 50-50 mixture. So when we use this normal cupric acetate, since if I take this is a chiral molecule at this point, so it will have whatever RS notation. Then because of this, it will have a diastereoisomer, so you will get 4 peaks over here. In this case, we got only 2 peaks. So that way, in both the sets, we got basically the chiral selectivity. Then we looked for or explored its mechanistic aspect. So literature report is also there that in this type of reaction, first acetylene interact with this. So we will have this one. After this proton becomes more acidic. So either it loses or it may absorb by something else. So if it is absorbing by this piperidine, which is a base, it will have this, it will have a positive charge. So now we are having a lot of active molecule in the system. I will have this positive charge. I will have electron deficient, electron descent over here. So it can interact with this, or it can interact with this molecule. So here we will have a number of reaction. Now when we explore the reaction mixture, we got this mass value which correspond to basically this molecule. And then this can interact with this, and that also will look one of the component will crystallize, we got this molecule. So we had the mass support for this, we had the support for this, and then further, since we had the product, so naturally one of the path will be this, where we will have this, and then it will go away, and this catalyst is getting back. Now here in this scheme, apparently it looks like this is a catalyst, but whatever our observation is, there is a slight difference. The color of this catalyst is green, but whenever we isolate the catalyst, we get yellow. It means this might not be an active catalyst, but active catalyst may be something different. So that is again a point of discussion. I will come to that one. Okay, then since we are talking about the mechanistic aspect, so we did control of catalyst loading with respect to time, and this way it shows with different catalyst loading, we are getting a straight line. And if I plot slope of this with respect to the mole percent of copper, this also gives a straight line indicating it is first order with respect to catalyst loading. Same type of experiment was perform with each individual component, and in all the cases we got first order reaction only. Okay. Then second we did the reusability. So this is the reusability report for 5 cycles. During the process, neither E nor percentage E was compromised. So that way this catalyst is robust and it can be used for a number of times. Now coming to the what is the active catalyst? Once we got the 5 cycle, we taken out the catalyst and try to analyze. We analyzed by XPX, which gives copper is in +2. We did UV and IR. So that also gives more resemblance. IR is one where we can have slight difference. Like if you are having phenylacetyl here, there will be a peak at 2368 for serial bond C. And then this is the complex where we don't have anything, but the catalyst gives a peak over here. So this basically supports that active catalyst is whatever I said you, that one. This will be the active catalyst where your copper and this is having connected. Okay, so in conclusion, I have basically discussed about the various complexes of glucopyroxylamine derived complexes. Out of that, I have talked only the chemistry of dinuclear complex in the form of catalytic oxidation of primary or secondary alcohol, as well as the stereoselective synthesis of propazyl amines. This whole work, the first part of the work, that is the alcohol oxidation, was done by Dr. Soni, and the A3 coupling reaction has been performed by the Santosh. I'm also thankful to the chairperson, Dr. Anil Kumar, because organic section I always consult with him. And then Professor Krishnan from our sister institution for crystallographic help, and then funding from SHERV, UGC, CSIR, and BITS Pilani. Thank you all.
Thank you, Dr. Ajay. The floor is open for 2 quick questions.
Generally, this A3 coupling and copper catalysis obeys positive nonlinear effects.
Okay.
So did you study any of such kind that may give further support to your mechanism, whatever you support demonstrated?
No, that way I'm not done. I mostly done the mechanistic
Professor
aspect.
did that one. Can go through. One more. Did you separate those diastereomers? So where you had propazylamine?
Yeah, I separated each one.
They are separable.
Yeah.
Fine. Thank you.
Any other question? If not, let's thank Professor Ajay for a nice presentation on carbohydrate chemistry.
Thank you, Professor Ajay. Now I request Professor Anil Kumar to present a memento as a token of appreciation to Professor Shah. Now we can have the next speaker.
So our next speaker is Dr. Siddharth Sharma, Assistant Professor from MLS University, Udaipur. And Dr. Sharma has done his MSc from MLS University, Udaipur. It's Mohanlal Sugadia University. He did his PhD from CSIR CDRI Lucknow and has a postdoctoral experience from Post-Tech South Korea. He was in DST Inspire faculty at Guru Nanak Dev University Amritsar from 2014 to 18. And since 2018, he is in Udaipur. I request Dr. Sharma to share his work.
Hello. ये वर्क नहीं कर रहा है क्या?
Hmm. Hmm.
Yeah. Okay. Thank you, Professor Anil, for this kind introduction. And I would like to thank organizing committee and especially Dr. Satpal for giving me this wonderful and rare opportunity to speak our work, what we are doing at Udaipur. So today I'm going to talk about the work which we have started in 2018 when I joined Mohanlal Sukharia University. And it's all about some of the electrochemical organic reactions. At the same time, we are very much interested in isocyanide based multi-component reaction and these multi-component reaction, how you can assemble these molecules into the complex molecule, or we can make how to make these molecule into the diversified manner, where we can generate a skeletal diversity, or maybe if we can generate the point diversity over the molecules. So basically, our group's interest started and lies in the isocyanide in MCRs, and these isocyanide multi-component reactions are very important, and that I will show in my presentation. At the same time, after 2018, there is a sudden renaissance of the electroorganic synthesis in the literature, and we were also equally fascinated by this chemistry. And today we are going to show some of the work which we have carried out in electroorganic synthesis, specifically related to the isocyanates. Apart from that, some of the work we have started where we can functionalize the CH bond, specifically heterocycles, by using the electroorganic synthesis. However, I will concentrate majorly on to these two areas. So these isocyanates are a very specific functional group in one aspect, that it has very stable divalent carbon nucleophile, where electrophile and nucleophile both can react with the electrophile and nucleophile both at the same time. Whereas you see that generally it is considered as an isoelectronic chameleon because ultimately this is the carbene center. It can act as a nucleophile. At the same time, because of the carbene, it reacts with the radicals also. At the same time, these isocyanates are considered to be the non-lethal weapon because lot of under chemistry development of the chemistry hampered because of this unpleasant odor, which is a legendary. Although the importance of the isocyanate has been found in the number of the natural product and hundreds of natural product containing these isocyanate, where you can find specifically in the antibiotic compounds. We are very much interested in isocyanate multi-component reaction because it can give us lot of handles to play with. Specifically, when we talk about the UoR reaction, UoR reaction is a 4-component reaction. It reacts amine, aldehyde, isocyanide, and carboxylic acid. Another initiative we have taken for this multi-component reaction is this: out of the four component, the 3 acid, amine, and aldehyde were very cheaply available. Even it was readily available in the market also. At the same time, another reaction which we were very much concerned about, the Passaroni reaction, is a 3-component reaction, and amidines can also be synthesized. So overall, these multi-component reaction can provide handles where we can manipulate the functional group onto the specific handle and generate the different heterocycles. So we have synthesized molecule or precursor, I should say, by using the UoR reaction, and a small modification into these UoR reaction can play a certain role to synthesize different heterocycle. In this case, what we done is we have introduced a OH group, or after this OH group can selectively cyclize into the corresponding 7 endocyclization or 6 exocyclization using the gold and palladium catalysts respectively. At the same time, a small modification when we can make this electron amine electron rich, then it can do the hydroannihilation reaction and generate the 2-quinolone structure using the similar precursor. What we also can do via using the same molecule, if we can do the cascade or domino mediate process iodocyclization, then we can generate a spirocyclic heterocycle. Recently, we have also developed, using the same precursor, we have developed a thiolated spiroindolin 2-1 via the small modification into the reaction. So it gives you an idea that using a similar kind of precursor, we can generate a skeletal diversity into the molecules. That's why we were fascinated by this synthesis. Specifically, I would like to concentrate on this work which we have carried out years back. This is a metal-free diastereoselective modular construction of a phenanthrene fused tetracyclic ring system, where this precursor have been synthesized using the Ugi reaction, where aldehyde, amine, isocyanide, and alkenic carboxylic acid react together and generate this precursor, followed by the iodocyclization and cascade process to synthesize this compound. All these compounds were synthesized in a very diastereoselective manner because of the domino process. This reaction has important advantages. First of all, this is a metal-free approach. At the same time, reaction was so cascade that it was happening into the diastereoselective and regioselective manner. Number of the chiral center can also be generated, and multiple bond CC, CN, CO, and CI bond formation was taking place in a single molecule. And at the same time, these also might have a biological activity because these complexity generally interact with the biological system in much better manner. Our inspiration about this work came from the 3 very good work which previously have been published, and one of the work was by the Ajay Srivastava group at CDRI, where what he did, he also have taken a similar scaffold and synthesized these important tricyclic skeleton. At the same time, this Eric work, where also have utilized this amine, at the same time alkyne and aldehyde and isocyanide to generate this polyheterocyclic frameworks. It means that still we thought that still there is a scope in this area, especially when we saw that most of these researchers have utilized this methoxy group as a activating agent or activating group to initiate the cyclization. What we thought is whether we could cyclize these kind of product in a domino and selective manner via the similar strategy, but at the same time we did not want to use a methoxy group here. That's why we carried out the cyclization, and even without the activating group, this reaction takes place, and we confirmed this by the first we confirmed by the single crystal analysis. The number of the compounds have been synthesized. In this case, the good observation were that even the unsymmetrical amine also has been taken, and at the same time we got the product. Apart from that, we have varied number of the carboxylic acid, amines, aldehydes, and isocyanide. We did not restrict ourselves even to generate more complexity. So what we did is we were delighted that we have an alkenyl iodide. So in a general Suzuki cross-coupling reaction, we further diversified the product. At the same time, our obvious next step was to generate the alkyne via the Sonogashira coupling to further complexify this molecule. In this case, the mechanism was much inspired by the previous report. In this case, this alkyne iodo complex followed by the ipsocyclization and Friedel-Crafts type cyclization to generate this final molecule. So we were interested in the cytotoxic activity of this molecule, although I would not like to comment much, but still we found 1 or 2 compounds which are reasonable in comparison to the doxorubicin. So after this era of 2018 and 19, suddenly we were become very much interested in organic electrochemistry. In short, I would like to introduce this area that organic electrochemistry is a branch of, you can say, organic chemistry, where these concerns with the reduction and oxidation of the organic molecules at the electrode. It might be by the direct electrolysis. At the same time, we can do indirect electrolysis. Although there are a number of the parameters which are different in the electro-organic synthesis when you compare with the traditional organic synthesis. But at the same time, one of the two approaches which has been developed continuously is direct electrolysis and indirect electrolysis. In general, sometimes this indirect electrolysis is compared to the photochemistry, where mediator takes up or give us electron and followed by the transfer to the substrate, and finally we get the product. So somehow this is one of the case study which shows the power of the electro-organic synthesis in the current scenario. At the same time, the industry took this electro-organic chemistry 50 years back. Even then, academia realized the potential of the electro-organic synthesis very recently. In this case, this is the pioneer paper of the by the Baran group, where they can do the Birch reduction, where previously it has been reported at the -78 degree. They can do at the room temperature without using a specific catalyst. So our motivation, because ultimately we were having a very small experience in the isocyanate chemistry, so we thought whether there is a chance to merge this isocyanate chemistry with the electrochemistry, that would be easy for us to take out the chemical from the shelf and start doing some electrochemistry. So ultimately, this electrochemistry approach, where this anodic oxidations and cathodic reduction generate the radicals. These radicals may be further convert into the cations or anion on the base of the cathodic reduction and anodic oxidations. So and at the same time, we also knew the little bit reactivity pattern of the isocyanate. We thought there is a strong chance that this carbene, the two electrons on the carbene sometimes react as a radical biradicals, and this is a previously it has been established that these radical can attach onto the carbon center of the isocyanate and further can transform into some other heterocycle synthesis or functional group transformation. So. And another thing is, we have also in-house developed this electron flow microreactor, and this flow microreactor also have their own added advantage over the batch electrochemistry, where better phase transfer, better mass transfer can also take place. This is the first work which we have carried out in electro-organic synthesis, where this amine and isocyanide can directly couple to generate the carbodiamide, and finally we can convert it into the urea. But the most importantly, I really want to concentrate on this part, where we have modified a number of electrodes. At the same time, we have changed the iodine source and solvents, current, temperature, divided cell, undivided cell. So after doing a lot of parameters, we found that this is the optimum condition. When we started doing this work, we came to know that this is a well-known process in isocyanate chemistry, where this, in the presence of the iodine, this carbon center takes the iodine and generate this diiodo complex, which was previously published in Organic Matter. We thought if we can do this diiodination of the isocyanate, whether we can convert a similar kind of approach using the same doing the methodology. Finally, what we did is we have taken this iodine source, this iodide source, convert iodide oxidation by taking the 2 electron to convert iodo, this iodine, and this iodine convert into the diiodo complex, followed by the simple alkenic nucleophilic substitution reaction to generate the carbodiamide. We have synthesized a number of molecules. We wanted to change the substrate in such a manner that we can take an aliphatic isocyanate which are readily available in the market, but at the same time we also took aromatic isocyanide and amines also have been varied. At the one instance, we could synthesize the DCC, although but in very poor yield. Still, to date, we are trying so that if we can directly combine this isocyanide and amine to generate the DCC without using these other catalysts. This is another simple work which we have carried out, the multi-component reaction with the water, thiol and isocyanide. We knew the reactivity pattern of the thiol in one sense. This thiol can generate the radicals, and these radicals are well accepted by the isocyanate previously. So what we thought is whether we can generate something where this thiol and isocyanide can merge together to generate some compound. We have initiated this 3-component reaction where the thiamide can be synthesized. This reaction previously reported under very high condition, and now it is oxidant free, metal free, and almost catalyst free. So in this case, we will not restrict our methodology to only the basic thiamide synthesis, but these two commercially available insecticidal also have been synthesized over the 2 steps, although the yields were less, but still we were happy that we could give some application in this scenario. At the same time, almost at the same time, this Lee and co-worker published a similar strategy where in place of the water they have taken the benzotriazole, and they have published in Chemical Sciences. Anyways, so we have done a lot of CV studies and control experiment. After that, we thought we found out this thiol radical can attach into the isocyanate to generate this C, and this C can be oxidized to at the anode to generate D, and this D take up the water and rearrange and convert into the thiamide systems. So we have in-house developed this flow microreactor, and this flow microreactor have like I said, it has an obvious advantage. The specific advantage I want to explain here is that you can control the inter-electrode distance. Basically, in the batch electrolysis, if the inter-electrode distance is much higher, in this case, the resistance will be higher and ohmic drop problem will come at the later stage. So these flow microreactor can solve these problems. At the same time, it has obvious advantages where the fast heating exchange, multi-step reaction, a multi-phase reaction condition can also be done. So we have carried out the gram scale synthesis, and almost the same reactivity pattern and the yield were obtained in the case of the flow system. This is the another work which we have developed. Till now, we have developed the method in which the anodic oxidations were involved. At the same time, we were very much interested that how these molecules, if we can use the cathodic reduction potential of these molecules, where this we have generate the one-part reaction where this amine in situ generate the diazonium salt, and this diazonium salt at the cathode reduce and generate these heterocycles. Oh, how much time? Finish? Oh, okay. So this is the proposed mechanism, and some control experiments also I have done. In very short, I will summarize. So till date, we have developed this methodology development specifically in the electroorganic synthesis, but we wanted to synthesize some new molecules. Our group was very much interested to develop a new heterocyclic skeleton. Specifically, we had small expertise in post-Ugi cyclization reaction. Therefore, our first instance to develop something which are more familiar to us. So we have generated this skeleton using the Ugi reaction, and followed under the HFIP and room temperature condition, we could cyclize this product to generate this 4-imidazolidinone molecules. This is the importance. In this case specifically, we have done more than 150 optimizations, although the best results are here. At the same time, this reaction was specifically going in HFIP, and no other solvents was good enough to carry out the reaction forward. Even we tried in methanol, but the reaction was very poor. A lot of substrate scope has been done. Specifically, I will highlight those functional groups which are generally considered to be the electrochemically labile because of the low oxidation potential. So these although but reaction was not reasonably good, specifically with the lithocholic also acid also reaction was going quite well. This is the proposed mechanism. Yes, what happened?
Is it not moving?
Yeah, okay, just a minute. So as far as the mechanism is concerned, we were ultimately in Ugi reaction, there are a number of functionalities available. So we never know that which part of the functional group is first oxidizing. So what we did is we did the knockoff studies where when we have taken the cyclic voltammetry studies of this molecule, we found out the two oxidation peak, but we did not know that which oxidation peak belongs to which specific functional group. So what we did is we knock off this methoxy group and we found the two again oxidation peak. At the same time, this time we knock off this methoxy group and we got this corresponding 2 peak. It means that the 0.7 volt belongs to this functional group, and that's where the oxidation is taking place. We also did some NMR studies also to prove that that why HFIP is important. At the same time, these are the few works our lab is doing, especially in the heterocycle CH bond functionalization. So I am blessed to be living in a very beautiful city, and this is considered to be the Venice of the East. And frankly speaking, I am inviting all of you, if you really want to come Udaipur, just drop me a mail and I would be very, very happy to host you personally. So I was at the same time also blessed with the funding, where my independent journey started with the Inspire Faculty Grant. Previously, I also had a SRG grant. And these are the two students which have done the work. And the Bhavardu recently got the regular position in the class for doing this in the company where these people are doing some electroorganic work. Apart from this, these are the 3 hardworking students currently I am working with. Thank you. Thank you.
Thank you, Dr. Siddharth, and thank you for inviting all of us to the City of the Lakes. Actually, it's a beautiful city, you know. We can have one quick question.
Please take the mic so that your question could be heard by others also.
It's a very good work, Siddharth. One thing just I want to ask you, you have chosen the electrode, both of the electrodes are platinum based electrode, but I have observed during your reactions. So it means it depend upon the metal electrode that you have chosen of your reactions will depend on that.
Yeah, so in that respect, what I can say is it's just like you are with the solvent when you are optimizing the reaction. But there are some certain parameters when you want to choose the electrode. Specifically, if you want to do anodic oxidation as a preferential reaction, then the carbon-based electrodes are more preferable, especially the carbon which have a high porosity like carbon cloth or revitrified carbon sources or glassy carbon. These are the electrodes generally considered for the oxidation reaction. But if you want to do the preferentially hydrogenation as one of the side products, then you can take PT as a cathode because it can effectively reduce. So again, you can say that you have to optimize a bit, but more largely it doesn't affect a lot. Like
in your case, both are the same.
Yeah, in my case, actually, both were the same.
So where is the preference, like anodic and because it's the same, both are the same.
Yeah, but we generate the potential, right? Why doing the generate the potential via the potential state. That's where if you use different electrode or same electrode, it doesn't mean it will generate the potential via the potential state. So
and normally your reactions are taking 2 to 5 hours also. So
sometimes even 24 hours also. Yes.
Okay, so it should not be a bit fast because the mechanisms that they are going to be follow, it's
in my, I just, yeah, the thing is that it depends upon the how much electricity you are giving. Ultimately, you can also calculate the how much Faraday you have transported into the whole reaction, because suppose if it's a 2 electron system, 2 electron total transferring into the reaction, it means that overall you have to transfer the 2 Faradays. So that Faraday can be manipulated by using the more electricity, or at the same time you can use the low electricity also.
Thank you, Siddharth. Maybe we can discuss little more during tea time. So with this, let's thank Dr. Siddharth for a nice talk on electrochemical oxidations.
Thank you so much, Professor Siddharth and Professor Anil. May I now request the chairperson to please present a memento as a token of reverence to Professor Siddharth Sharma. Thank you, both of you. We may have the next speaker.
So the last speaker of this session is Dr. Virendra Singh. Dr. Virendra did his MSc from Kurukshetra University and he was a gold medalist. Then he moved to CDRI to do his PhD. So he completed his PhD from CDRI in 2011. Thereafter, he joined Central University of Punjab, Batinda as an assistant professor. Moved to Department of Chemistry, NIT Jalandhar in 2012. Worked here as an assistant professor for 8 years and then moved back to Central University of Punjab, Batinda as associate professor in 2020. He has published over 71 research articles, 8 book chapters, and has participated in more than 32 national and international conferences. And Dr. Singh is recipient of MMM Dhar Memorial Award, Professor S.M. Mukherjee Award of Excellence in Chemistry, Nanlal Talesra Memorial Award from the Indian Council of Chemists, and Young Scientist Award from Science Congress. He was also selected for prestigious CSIR and postdoctoral fellowship. I request Dr. Singh to share his work.
Thank you, sir, for kind words. At the outset, I would like to thank the members of organizing committee and especially Dr. Satpal Batsara for giving me this opportunity to share my work. I will be discussing on exploration of chemoselectivity of Pimogean C for the synthesis of beta-carbolene based privilege scaffolds. We are basically working on a simple molecule. First of all, what is beta-carbolene? Beta-carbolene is basically a tricyclic framework where indole ring is fused with the pyridine ring. And it is a tricyclic system. We call A, B, and C ring. And this is the way we call its beta-carbolene depending upon the position of this nitrogen. If it is at one position, we call it alpha carbolene, beta, gamma, delta carbolene. And we are basically working on this natural product. This is Pimogean C, a very simple one. We also call it 1,4-mile 9H beta carbolene. Now the question arises, why beta carbolene based natural product? Yesterday and even today, the great eminent scientists of India and other also have explained the importance of natural product, that they contribute a major portion to the drug discovery program, and there are several natural products that are directly or their mimics or derivatives serve as commercial drugs. The beta-carbolene derivatives are ubiquitously present in nature, in plants, marine organisms, insects, mammals, and even human beings, including their body tissues and body fluids. That's why it's considered as a privileged scaffold, and they display a wide range of biological properties. And these are some of the commercial drugs that contain this beta-carbolene framework. Majority of beta-carbolene derivatives display anti-cancer properties by interacting with the various receptors like topoisomerase. They act as topoisomerase inhibitors. They intercalate into DNA and they bind with variety of kinases. These are just few examples of beta-carbolene C1 substituted natural products and alkaloids. These are beta-carbolene N-fused alkaloids. These are some beta-carbolene containing natural products, where 2 beta-carbolene rings are present in a single natural product. Now, why we were interested in this framework? The reason is that commercially, conventionally, beta carbolene derivatives are prepared by pictet Spengler condensation, but the scope of reaction is limited with the deficiency of electrophile. They are selected electrophiles only. So we envisage to develop a alternate approach where a beta carbolene containing scaffold that have good functionality can be further explored for the diversity oriented synthesis of beta carbolene derivatives. You can see in this molecule, it have electrophilic site at C1 position, and it is in fact a fast reacting electrophile. And there are two nitrogens that are nucleophilic in nature, and they are in close proximity to this formyl group. So this can be selectively used for the synthesis of beta carbolene C1 tether derivatives, but at the same time, they can be chemoselectively enforced to undergo C1N9 or C1N2 cyclization reactions. Therefore, first of all, we developed a gram scale methodology for the synthesis of Cumulgian C, and it was achieved through a three-step synthesis where L-tryptophan was treated with dimethoxyglycol in presence of 10% TFA in DCM. In fact, this reaction also works very well in pure water also. Further, when it was treated with N-chlorosuccinamide, in one step, decarboxylation as well as aromatization of C-ring happen to generate beta carbolene C1 acetyl derivatives. Further, demasking of acetyl in acetic acid water furnish this 1,4-mile beta carbolene. The advantage of this methodology is that it is very economic because tryptophan is very cheap material. Further, high yield is obtained at each step and no column chromatographic purification is required at any stage. Further, we demonstrated the scope of this natural product for the synthesis of beta carbolene N-fused imidazole derivatives. When it was treated with the naturally occurring alpha amino acids in presence of iodine in sodium bicarbonate, just within 1 hour, it resulted in formation of beautiful tetracyclic framework, beta carbolene N-fused imidazole derivatives. The scope was further demonstrated with various amino acids containing aryl, alkyl, aliphatic side chains. Interestingly, when reaction was executed with L-serine in presence of iodine, it afforded this pentacyclic framework. Further, this was the library of compounds that were generated. Interestingly, these molecules were found to be highly fluorescent in nature, and their photophysical properties were evaluated, and we found that these compounds displayed excellent quantum yield, and up to 50% quantum yield was obtained. This is the proposed mechanism. Initially, in presence of iodine, amino acid and 1,4-mile beta carbolene undergo condensation to give the imine. Further, in presence of iodine and iodination occur, and base enforces the decarboxylation to generate this yelid type of intermediate. This yelid type of intermediate undergo 6-pi electrocyclization, which further rearranges to beta carbolene N-fused imidazole derivative. The reaction was found to be chemoselective in nature. The reason is that when this NH is free also, even the same product is obtained, though there is another possibility that 6-member ring may also be generated. Further, in another study, we were interested to generate nitrostyrene of derivative for the preparation of eudiostomin analogues. However, when in actual we performed this reaction, we treated Cumulgian C with ammonium acetate in acetic acid. We furnished this bis carbolene derivatives with the beta carbolene N-fused imidazole skeleton. A retrosynthetic analysis revealed that in fact 2 molecules of 1,4-mile beta carbolene, though they are sterically hindered, but they coupled with each other in presence of ammonia, and optimization of reaction condition was achieved, and at 2 to 4 hours at room temperature in acetic acid, simply the reaction afforded these bis carbolene derivatives, and no column chromatographic purification was required. Further, the scope of reaction was also investigated for cross condensation of aldehydes. Here it was observed that the aldehydes, aromatic aldehydes that were more reactive than 1,4-mile beta carbolene can only undergo cross condensation. Otherwise, bis carbolene derivative was obtained as the product. The scope was also successfully demonstrated with aliphatic aldehydes. These molecules were also found to be highly fluorescent in nature, but moderate quantum yield was obtained, where 26% quantum efficiency was found. Then in an extension to this study, recently we thought to explore the phenyl glycol derivatives because they were thought to be more electrophilic in nature than 1,4-methyl beta carbolene. And when we executed the reaction, we were expecting this product. But in actual, this product was not obtained, and reaction followed the other route, and in a chemoselective manner, beta carbolene C1 substituted imidazole derivatives were obtained. And recently, parallelly, a report was appeared in JMedChem. These compounds have been found to be very excellent antidepressant agents. These molecules displayed excellent quantum yield, where up to 90% quantum yield was obtained. And these molecules were also found to be selectively bind with the metal ions, and quantum yield, quenching of quantum yield was obtained. So there is further scope for development as chemosensor and imaging agent. Today also, Professor Fernandez demonstrated the scope of butyrolactone derivatives, that they act as excellent anti-cancer agents. And they are present in about 5 to 10% of natural products. This gamma butyrolactone is obtained, and because of this exocyclic double bond, they act as very good Michael acceptor. So we thought to merge two privileged scaffolds in the form of 1,4-methyl beta carbolene and this butyrolactone. And this was achieved via Barbier type homoalylation reaction by using these substituted allyl bromides that were generated from Bailey-Silman adduct after bromination. And when the reaction was executed under semi-aqueous condition in indium THF, within 5 to 30 minutes, this homoallyl alcohol was obtained with trans conformation. It is important to mention that all the previous reports generally shows that this cis product is found, but in this case the trans product was obtained, and no further additive was required, no further activation. It resulted in cyclization in situ with trans stereochemistry. In this condition, it was found that actually water play a very important role in this reaction. If reaction is executed in anhydrous THF or freshly distilled THF, no reaction take place. But water was responsible for the trans stereochemistry of the product. In an extension to this study, we achieved the synthesis of beta carbolene C1 substituted benzimidazole derivatives. A variety of catalysts were explored, but it was found that N-free substituted benzimidazole derivatives can be prepared only when zinc oxide nanoparticle is used in chloroform. Otherwise, this side product is obtained in generally 50-50 ratio. The scope was further extended to synthesis of beta carbolene C1 substituted benzothiazole and benzoxazole derivative. In this, we found that only few selected derivatives can be prepared because of the commercial unavailability of 2-amino thiophenols. So to circumvent that limitation, we explored the sulfur insertion chemistry. Elemental sulfur is a very powerful sulfurating agent. It is stable under normal condition, non-toxic. However, it can be easily activated by polar aprotic solvents as well as iodine and some transition metal catalyst also. So we explored elemental sulfur and aromatic amines, where aromatic amines were reacted with 1,4-methyl beta carbolene and elemental sulfur in presence of iodine. It resulted in formation of beta carbolene C1 substituted benzothiazole derivatives. The scope was demonstrated with a library of around 35 compounds, and these compounds were also found to be highly fluorescent in nature, where quantum yield maximum up to 92% was obtained. This is the proposed mechanism. First, this Cumosian C and aromatic amines undergo condensation to generate imine. This imine is further activated by iodine. Then elemental sulfur, that is also in situ activated by DMSO as well as iodine, may attack this imine carbon or may attack this aromatic ring may attack over the sulfur. This elemental sulfur can react, behave as electrophile as well as nucleophile. And further insertion followed by this anion formation and in situ oxidation result in formation of beta carboline C1 substituted benzothiazole derivatives. Then we again further explored the sulfur insertion chemistry for nucleophilic substitution of aromatic nitro functionality. There are just very few limited reports where nitro group is known to act as a leading group, and generally copper, rhodium or palladium catalyst is used. But in this case, we used simply elemental sulfur as a sulfurating agent and denitration was achieved. Here the triethylamine, 5 equivalent of triethylamine was used as a activator for activation of sulfur. We developed one port strategy but sequential manner. When Cumigan C and its derivative were treated with the 2-nitroacetophenone derivatives, it resulted in formation of nitrochalcones, and when these nitrochalcones were in situ treated with the triethylamine DMSO in presence of sulfur, it resulted in formation of benzothiophenone derivatives. Overall, again, it can be generated in converted into one port approach. And this is the proposed mechanism. Reaction is found to based on control experiment. It has been found that reaction proceeds through a trisulfur radical mechanism. This trisulfur radical anion is formed. This trisulfur radical anion undergo this sp2ch insertion with the elimination of H radical, and this double bond is regenerated. Further, this triethylamine help in generation of this thiol type of anion, which undergo intramolecular nucleophilic substitution. This nitrite group is eliminated and it resulted in formation of benzothiophenone derivatives. The same methodology was further extended, where the nitrochalcone with different chemistry was developed. Here, 1-acetyl beta carboline, which is also another type of natural product, was reacted with 2-nitrobenzaldehyde derivatives. Reaction worked only in presence, this chalcone formation worked only in presence of cesium carbonate, other bases don't reacted. And when further it was treated with sulfur in presence of Ki, iodine as a activator produced the product in low yield. So Ki was used, and in one step here, 2-acyl benzothiophenone derivatives were obtained. And again, based on control experiment, it was found that reaction again followed a free radical mechanism and trisulfur radical anion is formed. Further, the sulfur insertion strategy was applied for the synthesis of beta carboline substituted amide conjugates, thiamide conjugates, where 1-formyl beta carboline, different aromatic as well as aliphatic amines were coupled in presence of sulfur to generate the beta carboline C1 substituted thiamide derivatives. The strategy was further extended for the synthesis of sulfur analog of LNGO-bucine. This has been previously synthesized by Dr. Ajay Srivastav, but we have synthesized this sulfur analog in just one step by coupling this natural product of N-methyl Cumigan C, tryptamine and sulfur under catalyst free condition in DMF at 70 degree. However, when the just base is added in this reaction, tryptamine does not undergo condensation, just it acts as a source of ammonia and bis-carboline derivatives were obtained. In another study, we explored this Cumigan C for A3 coupling or a Poirot type of reaction with the aromatic amines, electron-rich amines, and with alkyne. The reaction proceeded well in presence of iodine under transition metal free condition, and the products were obtained in 40 to 70% yield. And these beta carboline C1 substituted quinoline and fused quinoline derivatives were obtained that were analogs of nitramarine. Further, this aromatic amine was replaced with 2-aminopyridine. Actually, this study was very interesting because in this molecule also there is pyridine moiety. Here is a pyridine moiety. So there may be chemoselectivity and regioselectivity issue. However, we found that when reaction was performed in presence of copper sulfate, only then this reaction is regioselective. Otherwise, a mixture of product is obtained. Further, PTSA also play a very important role, and it was used as an additive, and reaction works only in toluene at 100 degree. The products were obtained in 60 to 90% yield. And the reaction proceeds through the formation of this properxylamine intermediate. Finally, I would like to summarize by using a single precursor, that is Cumisian C. We have synthesized diverse chemical libraries of compound. Almost every library involved more than 25 compounds. By using decarboxylative amination, sulfur insertion strategy, or barbier type reaction, or oxidative amination. Finally, I would like to acknowledge our worthy Vice Chancellor, Head of the Department, my PhD students who have worked day and night, CSIR and New Delhi, CSIR New Delhi and SERB New Delhi for research funding. Thanks to all of you for kind attention.
Thank you, Dr. Virender, for a nice talk on so many molecules you have prepared. We can have two quick questions. Anyone? Students? If not, let's thank again Dr. Virender for a nice talk.
Thank you very much, Professor Virender and Professor Anil Kumar. May I now request Professor Anil Kumar to kindly present him a token of reverence. Thank you, Professor Kumar and Professor Singh. With this, I now invite Professor T. Punyamurthy to kindly come over the stage and hand over a token of appreciation to Professor Anil Kumar for his valuable time and kind cooperation during this session. Thank you, Professor Kumar and Professor Punyamurthy. Ladies and gentlemen, now this is the time for tea break. It is going to be for 10 minutes, so see you all at 4:45 p.m. I request the speakers of the next session to please come and submit your PPTs. Once again, I request the speakers of the next session, Professor Ramani Guru Brahman,
डॉक्टर प्रभात के कोई जाओ ना लेकर आओ। क्या क्या लेकर आने में? उनका ये पीपीटी गायब हो गया है इधर से। डॉक्टर कोई लगता है पेन ड्राइव से यहीं पे वायर आप बोलना नहीं है ट्राई में गए हैं। डॉक्टर प्रभात थर्ड है उनका वैसे भी। सुनो उसको फोन करना राकेश का नंबर है क्या? गुड इवनिंग।
Ladies and gentlemen, I request you all who can hear me outside this auditorium, please come inside and take their seat. We are beginning with the next session.
So I don't know, this is first time.
Please come back and take your seat.
Is it been here? Okay, speaker.
Yes.
Okay, perfect. Is this one or? The speaker?
No, this is speaker. This one. Here. So will you begin?
Fine, right?
Okay. You can
So why, what is?
Ladies and gentlemen, we have with us R.D. Kaushik from Gurukul Kangadi University to chair this session. We all have heard him earlier, so he's no more introduced. I directly hand over the session to him. So may we have you here?
No.
Hmm.
Yeah, no? Good afternoon, everybody. In this last session of today, we are having 2 invited lectures and 4 special short invited lectures. Invited lectures will be of 20 minutes each, and the short invited lectures will be of 10 minutes each. So before we commence with these lectures, and I invite the orators, I would request all the orators to limit their talk to the time allotted. And our first speaker is Professor Ramani Guru Brahmam. Is he there? Okay. Professor Guru Brahmam, he is Assistant Professor in Department of Chemistry at Indian IIT Jammu. He got his PhD from University of Hyderabad and did his postdoctoral research at Stockholm University and National Taiwan Normal University. With these words, I invite Professor Brahmam to deliver his talk.
Yeah, thank you.
Yes.
Okay, first of all, I would like to thank Dr. Satpal for giving kind invitation and the organizing committee. Okay, so we established our lab around exactly a year ago, and I would like to give what we have done in the last 1 year. And so basically we are working in asymmetric synthesis, and it's like central core, and with the subdomains alkyne and alene chemistry and hydrogens and diazo compounds and some photoradars catalysis and hypervalent ion chemistry and organoboranes. Okay. So why diazo compounds are so important and what chemistry is going on? Actually, these diazo compounds are very old, but if you look at literature, what are the research progress on the diazo compounds in the last, I would say, decade, it has like a lot of boom. Okay, generally, let me categorize or narrate the diazo compounds. So Davis classified the diazo compound into 3 categories based on the relative reactivity. Generally, they are very reactive, but in relatively, we can classify them as more reactive and less reactive than again the moderate. So if you look at a diazo compound of this kind, and the diazo compounds having 1 electron withdrawing group here, and this one it is having 2 electron withdrawing groups, and these 2 are like acceptors. And these 2 diazo compounds are stabilized by electron withdrawing groups. If you look at these things and you have here partially negative charge, this will be stabilized by the electron withdrawing groups. And these are called like acceptors. And in the other way, and if you look at this has 1 electron withdrawing group and 1 electron donating group. And these are like donor and acceptor type groups. And the other way, the last category is donor. So the diazo compounds having either 1 or 2 electron donating groups. And relative to the reactivity wise, these are more stable. And because they are stabilized by electron withdrawing groups, and they are even less reactive compared to the other things. And since they are stable, you can isolate them and store them, and you can use whenever you want. And the other third one, they are like since they are having electron donating groups and they are more reactive, and you can't isolate them.
you need to generate in situ and you need to subject for further transformations. And the middle one is more important, well, like now Professor Davis explored like anything. And these reactions are more reactive and relatively, I mean, moderately reactive, but more selective. So let me give the literature in between 2000 to 2020, I mean around 20 years span. So almost there are like 7,000 reports on an average 350 reports per year. So we have chosen like donor and acceptor type diazo compounds, and these three are the categories which falls under donor acceptor diazo compounds. So here, if you look at, this is phenyl and this is like sterile, and the other one is alkyne. And among these three categories, this compound was least explored. This one is least explored, and perhaps the reason could be due to its synthesis. Maybe if you look at the literature, this synthesis can be done from homopropagylic alcohol. This can be oxidized, then the corresponding acid can be methylated, and you will get 3 alkanoids. And after that, you can use a diazo transfer reagent, then you can get the corresponding diazo compound. And in this flow, you need a diazo transfer reagent. And this was reported by Professor Davis in 2000. And in 2004, the GC Fu group, they reported from here to here, and they minimized this step, and using the starting materials and the further 2 steps, they minimized to 1. And this is like now alkyne diazo compound coupling in presence of copper catalysis. And from there, now every research group, they are synthesizing these diazo compounds in this path. And this report was there in 2004. Still, since even 2004, people didn't explore this diazo compound. And there are 2 more approaches to synthesize the diazo compounds, especially with alkyne substitution. Professor Mikonin group from France, they utilized this alkynyl aluminum reagent and diazo compound, and they got this kind of hydrogen reagent, hydrogen substrate. And from there, they subject this one in presence of manganese dioxide oxidation, and they employed this one in large excess, and they synthesized this one in situ, and then after that, they utilized it for alkyne carbon transfer reactions. And if you look at the other report in the synthesis of the diazo compounds, B and Jo groups, they utilize the inones or like now inols, and they condense with substituted hydrogen derivative. And this substitution is much more important. If you do, for example, if you don't have this substitution, and you may get some other side products which were not desired. So if you look at from here to here, it's much easier to go. But in their procedure, they utilize the chiral Lewis acidic metal to condense it. Then after this, if you want to get from here to here, then you need to treat with the base to get the diazo compound. And let's suppose this diazo compound is having either a tosyl group, substitution, the protection, then they used to get only the pyrazole, and it is not giving a desired diazo compounds. And that is the reason they went for large steric protection like nosyl and trisyl groups, which can minimize the pyrazole formation. So during our work with non-organic catalysis, so my student, so he isolated this compound in less than 10%. From there, we visualize this is stable enough, and we can synthesize and we can design a project. Then we designed a project like synthesis of hydrogens from ionoids and hydrogen condensation. And after that, we visualize this hydrogen can be oxidized using metal-free conditions like equivalent iron 3 reagents and so, and we can get corresponding diazo compounds. From there, till now, if you look at the earlier reports, till now they explored rhodium and silver catalysis, and that too they are having bulky ligands around. And we try to develop like simple copper catalysis with not much ligands surrounded by, and we got the products like substituted propargyl derivatives and the silanes. And we try to optimize the first step, like from inoate to hydrogen condensation, and we went a vigorous optimization. The full optimization has around 50 entries, and which out of them I gave the best 10. And this reaction went smoothly, even in presence of, even in the absence of the catalyst, and it gave the corresponding product in like 14%, but the undesired pyrazole we got around 33%. Then we vigorously screened a lot of acids and bases and bifunctional catalysts like anything. And at the best scenario, we got the desired product in with acetic acid and DCM. We got around 72% yield. And when we tune the solvents from dichloromethane to alcohol, we got in a good amount, like 81% yield in isolated one. And then we went through substrate scope, and this particular reaction gave good yields of hydrogens, irrespective of the stericness on the ester component. For example, if we move from methyl to ethyl and ethyl to tetrahydrobutyl and adamantyl, it gives nice yields, and this steric factor didn't affect any yield. And we also got the product with menthol ester. And further, we changed the scope on this R component from aryl to alkynyl. Also, it gave good yields. And then we switched to the substrate scope and screening on aryl component of the hydrogen. I mean, inoate. Then here, more or less like electron donating and withdrawing groups, it gave the nice yields. But when it has like two substitution at this one, the 2-methoxy case, we got poor yields around 58%. Then we also synthesize the hydrogens, which is having substitutions with naphthyl and heteroaryl, and we got good yields. But the most important thing is when we have the substitution with like TMS and tapes and TBDMS, in the TBDMS case, it gave us around 98% yield. It means the steric effect on this one has a dramatic role on imparting the yields. That one I will describe when I emphasize on the mechanistic part. Then we also synthesize the unsubstituted here without having any electron withdrawing group, and we got this product with a simple condensation of propargyl aldehyde and hydrazine. Okay, these kind of products generally earlier here Mikoni reported, and the procedure involves a lot of sensitive reagents and steps. But here straightforwardly we got in good yields. And we have done some mechanical studies to understand the mechanism. First, we executed the gram scale reaction, and it gave almost a compatible yield. So in the control experiments, so first we took the E and we subject for the photo conditions, and E isomer got converted into G isomer in 48% yield. Similarly, whatever the isolated G isomer, we subject again for the same conditions, and it got converted into E isomer in 75% yield. When you look at the yields from E to G and G to E, and conversion from G to E is very feasible. It means from this data, we can say that E isomer is more stable than G. Next, so we took this E isomer and we subject the same isomer for the at the same optimized conditions. We want to check whether this isomer is stable enough or not, or the hydrogens can be storable or not. Then at the optimized conditions, we didn't get the side product pyrazole, and at the same way we recovered the isomer back in 94% yield. And it has also some meaning in mechanistic aspect. There are two products in the mechanism. One is the desired one, the other one is pyrazole. Okay, and here in this path, we didn't get any pyrazole. It means there is no scope for having the pyrazole from E under the same optimized conditions. Next, during our reactions, we never isolated or we never observed the G isomer. When we thought maybe this G isomer is maybe we are getting, and it is forming in the reaction, but it is not stable and it's converting very quickly to the E. Maybe that is the reason we are only seeing E. Then we thought of checking from G to E isomer. Then we subjected this G isomer and at the optimized conditions, and we analyzed the data, but the G isomer was not converted into E. It means there is no formation of G isomer in the mechanism. So actually there is a good data from the literature and the NMR aspects. If you look at the E isomer and the alkane in the 13C NMR, these alpha and beta carbons having characteristic peaks. So when this amine and alkyne are synced to each other, then the alpha peak resonates at 76 ppm, and whereas the beta carbon, it are peaked at 104, and the difference is around 28 ppm. And the computational data was reported earlier. It says whenever such kind of systems are there, this will be around the region of 20 to 30. This value exactly fits with the computational data. Similarly, we also analyzed the G isomer, and this difference is around 4 ppm. And as per the literature or as per the computational data, it should be less than 10. And also, it means it is also fitting into the data. This is because when we have this kind of system, one of the NH will interact with this carbon. And so the electron density from this triple bond will be polarized towards the alpha, and the partially negative charge will be developed at this alpha carbon. And so it will be in upfield. Relatively, the other carbon will be in the downfield. Whereas such kind of thing you don't see in the G isomer. So based on the control experiments, we propose the mechanism. First, in presence of the acidic system, the hydrogen will add across the carbonyl group of the inoate ester. Then you will get corresponding acetal. And once you get the acetal, the acid can be protonated to OH, and you will get the dehydration, and you will get corresponding E isomer, like a simple condensation. Maybe this path may be leading through intramolecular hydrogen bonding. And the same way, if you look at the system here, this alkyne and this hydrogen component should be transferred to each other, so that it will be ending with the E isomer. And alternatively, we also got the pyrazole in less yield. I mean, as a side product. It could be through Aza-Michael initiated reaction. Let's suppose the hydrogen adding at this position, and you will get first enamine structure. Then after that, it will condense across the keto, and you will get corresponding pyrazole. And when we look at this kind of TBDMS or TMS or triisopropyl systems, TBDMS, it is around 98%. This could be due to the steric hindrance of the cellular group. Let's suppose, as per the control experiments, as per the predicted mechanism, the pyrazole can only form by adding at this position because of the steric position on the cellular group, and it is not adding at this position, and it is exclusively adding at the carbon position. And so in this particular system, we are only getting exclusively one product at the quantitative yields. Then we turn our attention to utilize the hydrozones and convert them into diazoacetates. And my student, she optimized this reaction with a lot of reagents. Although I didn't mention here, he used like triethylamine iodine system, and also she tried with the manganese dioxide. But in all the system, and she got the good yields with the PEDA reagent, this oxidant, and whereas this traditional manganese dioxide gave less than 5% yield, and even that takes a lot of time. So we are giving we isolated so many, and I'm giving here a few among them. And in the worst scenario, we got 95% yield in the particular transformation, and the best is 99% yield. It means this particular reaction is quantitative, and also it will, if your reaction is quantitative, then you can go for further transformation. I mean, you don't need to isolate the intermediate, etc. It will give you a nice yield in the second step. So if you want to run a sequential reaction, perhaps we need to get the yields in the first reaction like from greater than 90% yield. So if you look at the mechanism, so this hydrogen will be having like nucleophilic amine. This will add to the iodine center, and one of the ligands like from the acetates will leave. Then you will get this kind of I3 intermediate, and this is called in I3 chemistry ligand exchange. Once we get this intermediate, the lone pair on the imine nitrogen, it will go inside, and this I3 species will disproportionate, and it will give you the corresponding diazo compound and isobenzene and another ligand in terms of acetic acid. Unfortunately, these are very stable, and given reaction conditions, we got in good yield as we mentioned here. Next, we try to attempt application for these diazo compounds, and immediately whenever we look at the diazo compound, the first thing will come into trapping the carbon with this alkenes, so that we can get the cyclopropanes. And if you look at the such kind of alkynyl cyclopropanation, you will get through this transformation like this kind of cyclopropyl substitute alkyne, and they are very good for post-functionalization. For example, this strained ring can be opened up, and this triple bond can be functionalized simultaneously. You will get like annulations. So earlier, there are some strategies to synthesize this. If we look at one can synthesize this kind of system from anions. So you can trap the selectively the double bond of the anion, and you can propanate it, and you will get this desired skeleton. Similarly, you can add like alkyne to the methylene propane or cyclopropane. So in both cases, it will give you corresponding cyclic alkyne system. And in addition to these methods, there is one more report where you can use this kind of system, and you can subject this one for the rhodium, and you will get a kind of rhodium carbonide from a non-diazo substrate, and that can be trapped with a double bond, and you will get the desired system. And all these are like laborious to generate and get the products. And if you look at the carbon trapping from the diazo compounds, so Davis reported around 2000, and he used rhodium, and she reported the similar kind of hydrogen and base, then that can generate the diazo. Then after that, he used the silver metal, but with a bulky ligand. And then he got the metal like silver carbon that he trapped with alkene. He got the corresponding cyclopropane system. And these methods is either it's having like precious involved with the precious metals or the bulky ligand. So we thought it could be better if you substitute and if you minimize this kind of complexity, then we thought of optimizing the reaction with the copper catalysis. And we screened for various catalysts and the solvents and the temperature and equivalence. And they had the best scenario. This copper one, it gave around 86% yield with good diastereous selectivity. And we have done some substrate scope with respect to aryl, with respect to ester, and also with respect to aryl and the stearins. But in this substrate scope, what we found, we had good yields and good diastereomer selectivity when the alkene is having electron donating groups on this position and this array, and we have poor diastereomer selectivity when we subject for kind of aliphatic like cyclohexene and all. Yeah, fine. So the rate of chemistry over there will be electron fixed from the XRD data. And we have also executed one part system, and without isolating the diazo compound, they are relatively unstable. And so we subject for one part, and we could manage with the one part reaction, we could get some derivatives. And we also employ the same reaction with the selenide insertion, and we got nice yields from 60 to 68. And when you look at the mechanism, this nucleophilic diazo compound will add to the metal, and you will get like now this kind of intermediate, and the d electrons on the metal go inside, it will expels the nitrogen, you will get the metal carbene, and this metal carbene will add to the alkane, and you will get a cyclic intermediate in the constructed path, and it delivers the cyclopropane, and with concomitant like now ejection of the metal. And we have done like now in the last this project, we have developed a good strategy to synthesize hydrogens and bypassing metals and also the diazo compounds. And we subject them for the cellular insertions and cyclopropanations. And this is the first copper catalyzed alkyl carbon transfer reaction. And currently we are working with other things, and we have like now fairly good results in metal free conditions as well. And I would like to thank my group and Akashdeep and Parujamal. These two students were involved in the this project, and he established the hydrogen synthesis, and she stabilized the diazo compounds and the carbon transfer reaction as well. And other students, they are having Himan is having like photochemistry applications. And she's a project student in my group. And I would like to thank IIT Jammu for funding and also the infrastructure, and CSR and SERB for startup grants. Thank you, and thank you very much.
Yeah, just one or two questions. Any question? Any query? If no, then thank you very much for the nice talk. And we'll have to announce something.
So I would like to request Professor Kaushik to present a memento to Professor Guru. Thank you, sir.
Well, our next speaker is Dr. Amit Patwa. Dr. Patwa is Associate Professor in Division of Chemistry, School of Science, Navrachna University, Vadodara. He had been postdoctoral research fellow at Boston University, USA, and in CERN, France. He obtained his PhD degree in chemistry from National Chemical Laboratory, Pune. Has 28 research papers with more than 720 citations to his credit, one book chapter, and one international patent, S index 16, I-10 index 19. With these words, I invite Dr. Patwa for his talk. Please be restricted to 20 minutes. That includes actually
Before 20 minutes,
Okay.
I guarantee. Hello. Yeah. Okay. Thank you. Okay. So, good afternoon, everyone. First of all, I would like to thank Dr. Satpal Barsara for giving me the invitation to present my work in this conference. And also, I would like to thank the entire organizing committee, FCASI 2023. So, from like yesterday, you are listening about the drug molecule. Definitely, my talk is not about the drug molecule, but it is about the diagnosis. We are using the tantalum oxide nanoparticles for diagnosis for quantitative contrast endoscopic tomography. So it is the diagnosis of the osteoarthritis. Osteoarthritis is a joint disease which results from the multiple factors. Any people around like age more than 60, they may develop osteoarthritis. People having the body mass index greater than 30, they also may have developed the osteoarthritis. There are multiple reasons. So here you can see the healthy joint in this one, and here it is a degraded joint. So currently, if you see the technique, diagnosis technique available in the clinic, that depending on the X-ray. Here, if you see the first X-ray, it is a normal joint where you can find the proper gap between the two bones. If it develop the osteoarthritis in a very early stage, it will still look like the same. And when you visit the doctor, doctor will prescribe you the medicine, maybe painkiller, or he will ask or she will ask you to change your habits. But at that time, it has started to degrade the cartilage. If you develop the mid osteoarthritis, it may find there is a narrow gap between the joints, and at the later stage, the end stage, the gap will be very, very narrow. And this particular disease is not reversible. Once you go to this, the only solution is the knee replacement. There is no such medicine is available in the market as of now. So our idea is that if we can identify this osteoarthritis at very early stage, then researcher may find the solution or may find the medicine for this disease. So this particular articular cartilage is a hairline cartilage, which composed of 65 to 80% of water, 10 to 20% of glycan, and one collagen, and 1 to 10% of glycosamine glycan. The strength of this cartilage is mainly depending on the glycosamine glycan. This glycosamine glycan absorb the water and give the mechanical strength to this joint. And where there is a development of the osteoarthritis, this glycosamine glycan, the amount will get decreases. It will get deployed. This glycosamine glycan is mainly the long unbranched polysaccharides which contain negatively charged sulfate and negatively charged carboxylate group. So we inspire from the histology that this technique, what they are doing, they are staining the cartilage with the safranin O, which is a positively charged, and our GAG is a negatively charged molecule. So there is an electrostatic attraction between these two, and it will dye the particular cartilage with the orange color. So if you see, if you compare the healthy cartilage and the cartilage, where you can find the color, you can say that there is more amount of the cartilage, your cartilage is healthy. If there is no orange color or less orange color, the amount of the GAG is degraded, means your cartilage is degraded. So, but there is a disadvantage of this particular histology, that histology itself is a very destructive technique, and you can analyze a very small part of the cartilage. You cannot take like a multiple samples from all over the cartilage. So you can analyze only the part of the cartilage. So then we thought that already histology is using the UV part of this electromagnetic spectrum. We have two more, that is MRI and X-ray computed tomography. So today's talk is mainly depending on the X-ray computed tomography, where we are using the contrast agent to get a better image from the X-ray. So X-ray image, the quality of the X-ray is mainly depending on the contrast you have produced by your body parts. Like if you at that time, the contrast will not be the proper one. At that time, it will be very difficult to analyze your cartilage, and at that time, we are providing the contrast agent which will generate the proper contrast and you can analyze. In the next slide, you can see that these are the contrast agent which are iodinated molecules, which are FDA approved molecules. These contrast agents are either neutral or negatively charged. You can see the difference between the simple X-ray and the contrast enhanced X-ray computed tomography. You can see here. You can simply see the bond. You cannot see any cartilage between these two bonds. But if you provide the contrast and then you image it, you can easily see and you can decide the health of this particular cartilage. So we propose, you can say that just before 5 years, I presented this molecule in the FCASI 2018. It was a contrast agent with a positively charged. In this particular conference, I am going to present, which is the nanoparticle based contrast agent. Here is a tantalum oxide nanoparticle based contrast agent. So our hypothesis is that if you use positively charged nanoparticles, which will attract towards a negatively charged DAG, and we have a better uptake of this positively charged nanoparticle, by that way it can produce a better contrast and we can view the cartilage properly. The advantage is, as it carries the multiple charges, there will be a greater uptake. So keeping this in mind, our prime target was the synthesis of this nanoparticle and then the quantification of the biochemical content. Okay. So design of the nanoparticle, I will not go much in detail, but I would like to ensure that nanoparticle has a large surface area, so it can accommodate more positive charge compared to the small molecule that is a contrast agent, iodinated contrast agent. And the second thing is that if you see the tantalum oxide, tantalum and iodine, iodine has a value 67.4 KH value, which attenuates the X-ray much more better than the iodine, so it will produce much better contrast than the iodine. And definitely this tantalum is also working in the similar range where clinical scanners are working, starting from the 40 to 120 kilovolt. So this is the design where we have a core of tantalum oxide nanoparticle and then biocompatible layer, and then there is a charge on the nanoparticle. So I will not go into the details of the synthesis, but I will just read out that we started with the tantalum ethoxide. We treated with water and isopropanol and isobutyric acid to get the tantalum oxide particle, and then we coated it with the positively charged, which is a commercially available molecule and which accumulate more positive charge. And here the advantage is the positive charge is fixed on the nitrogen, so it will not depend on the pH. Whether we change the pH, the positive charge remains positive charge. It is a tetraquaternary common salt. And the second thing, particularly I would like to say that the isobutyric acid, here the role of the isobutyric acid is controlling the size of the nanoparticles which we want less than 10 nanometer. If it is less than 8 nanometer, it will be fine. So then we characterized the nanoparticle. We carried out the dynamic line scattering and measured the diameter of nanoparticle, which turned out to be the 5.6 nanometer, and zeta potential, which is plus 13, which clearly says that our nanoparticles are positively charged. And these nanoparticles we have synthesized on a gram scale. Definitely, it will maybe you can say that 5 to 10, 1 to 2 gram or 5 to 6 gram I have synthesized. And these nanoparticles are very white, fluffy powder. We can store it. It is stable enough. We can store it in the refrigerator for a long time. These are the 10 images Actually, we have synthesized positively charged nanoparticle, and to compare, we also synthesize the neutral nanoparticle to compare the results. Here, we also carried out the NMR analysis, which also shows that the nanoparticle, because we just integrate the number 1 and number 2 carbon, which is in proportion, which says that it is already coated with the material. Okay, definitely I will not go in much detail of this analysis, but then we carried out the diffusion analysis, diffusion of this contrast agent into the cartilage. And we saw that over the period of 24 hours, it will diffuse in all 3 layers, like surface, middle, and the deep layer of the cartilage. Here you can see that this is the healthy joint. This is the degraded joint. If you go through the simple X-ray images, you cannot find anything. But if you go through the contrast enhanced computer tomography, you can see the blue layer, small blue particles. It is degraded one. It is the only green one, that is the cartilage, but the blue one, which is not the cartilage one. So here you can say that it is a degraded one. So if we can identify the osteoarthritis at a very early stage, researchers may find the solution for the cure of this kind of disease. And also, we have develop a scale where we can find out the amount of the gag present from the intensity of the color you obtain. It's kind of technique that after 6 months or 8 months, you have to go to the clinic, just go through, inject the contrast agent and just have an image. You can see the health of your cartilage. So it is not a drug molecule, it is a diabetes molecule. If you can find there is a development of early osteoarthritis, at least you can start changing your lifestyle or taking the painkillers from the beginning, not at the later stage, because at later stage there is no cure. And with this, I would like to conclude that we have successfully synthesized this nanoparticle, and which is covered with a positively charged tetra ammonium salt, and this is correlating with the GAG content in the cartilage. And we have definitely more work is required to apply this particular one for a human purpose. The molecule which I presented in like earlier, that molecule is biosynthesized in a kilogram level, and that in horses, in rabbits, in rats, and even in human. And I assure you that all are living right now. So there is no issue with that, and it is a biocompatible molecule. So, and we publish our results in SCS Nano. And finally, I would like to thank my collaborator, Professor Mark Rista from Boston University, and Professor Brian Sterner from Beth Israel Deccan Medical Center, Harvard Medical School. And finally, I would like to thank Rajasthan University for providing some infrastructure to carry out the work. Okay, thank you.
Okay.
Any questions?
Thank you, Dr. Patwa. Any questions? No.
Okay.
Okay. Thank you for the nice talk and keeping yourself in the time frame. That's it.
Thank you, Professor Patwa and Professor R.D. Kaushik. May I now invite the chairperson, R.D. Kaushik, to please hand over a token of appreciation to Professor Patwa.
Dr. Bhatt,
Dr.
Rupesh Bhatt, is he there? Yeah. Can you give me your short CV? Yeah, please. Okay. You have to change the document. Yeah.
We may have now next speaker.
Yes. Hello. Yes.
Our next speaker is Dr. Prabhat K. Barolia. He is assistant professor in Department of Chemistry, of course, from Mohanlal Sugadia University, Jaipur. Got his PhD from the same university. And he has published 25 research articles in national and international journals. The title of his today's talk is Design, Synthesis, Anti-Diabetic, Antioxidant and Anti-Inflammatory Activities of Hydroxytriazines.
Hello. Good evening to all, and thank you, Professor Kaushik, for nice introduction. So today I am presenting my research on design, synthesis, anti-diabetic.
Dr. Barolia, you should know that you have got a short invited lecture.
Yeah.
Of 10 minutes only.
Yes, definitely.
Including questions.
Yes.
Okay.
So my talk is on synthesis, design, anti-diabetic, antioxidant and anti-inflammatory activities of hydroxytriazines. So hydroxytryazine is a very simple molecule which have three nitrogen connected in a linear way, which is composed of hydroxylamine group and diazo group. So this exists in two tautomeric form. One is hydroxytryazine and second one is triazine 1 oxide. So this compound has been synthesized from last 30 years in my lab, in my senior colleagues. So they have explored their chelating nature. So this is a good chelating agent. It can form coordination bond with most of the transition metals via nitrogen and oxygen donor atom. So they explored hydroxytryazine as chelating agent, particularly for spectrophotometric reagent for determination of transition metals. And they also explored complexometric applications of hydroxytryazines. They also explored biological activities, particularly anti-diabetic, anti-inflammatory, antioxidant, antibacterial, antifungal, lipid lowering, and analgesic and wound healing activity. So synthesis of hydroxytryazine involve three simple steps. First step is reduction of nitro compounds to their hydroxylamine. Second step is diazotization of aromatic amine aniline to diazonium salt. And third step is coupling between them, the product of step first and step second. So I started my research work with simple molecules, nitrobenzene and aniline. So I have synthesized 6 compound which consist of sulfacetamide moiety, and we have synthesized 6 compound and explored their anti-inflammatory activity by kerosene induced PA edema method. And we found that our compound displayed good anti-inflammatory activity, and our compound 6 reversed the inflammation by 98% in 4 hours. And this we publish in BMCL. Next, we explore anti-diabetic activity by alpha glucosidase inhibition method and antioxidant activity by DPPH radical scavenging method. So here we analyze alpha glucosidase and antioxidant activity by DPPH method, and our compound showed the anti-glucosidase activity and antioxidant activity. So our compound showed good antioxidant activity by DPPH method. In addition to that, we have also synthesized a series of sulfur drugs based hydroxytryazine, which is started from sulfanilamide, sulfadiazine, sulfapyridine, and sulfamethazine. So total we have synthesized 26 compound, and we published in bioorganic chemistry. We studied for anti-diabetic activity by alpha glucosidase and enzyme inhibition method and alpha amylase inhibition method. So these are both digestive enzymes which digest oligosaccharides and polysaccharides to monosaccharides, or alpha amylase digest starch into monosaccharides. So if we hinder these digestive enzymes, we can manage the diabetes by hindering these two enzymes. We also did DPPH inhibition and ABTS inhibition method. Found that our compound 9, 8, 9, 10 showed the good alpha amylase activity, which is comparable to standard acarbol. So here 2 compounds are good active than standard acarbose. Here we also did the ABTS inhibition antioxidant activity. So here our compound number 11, 12, 13, 14 showed the good results, which is less better than standard drug ascorbic acid. In addition to that, we also did the computational study by molecular operating environment, and here we analyzed a docking study on human pancreatic alpha amylase enzymes, and here we found that binding energy of our compounds is comparable with standard. In addition, we also synthesized some more sulfur drug-based hydroxytryzine, taking the starting material, sulfa-thiazole, sulfisoxazole, and sulfa-methoxazole. And we have synthesized, I think, 18 compounds and published in Helion. So here we also studied alpha-glucosidase and alpha-amylase inhibition activity, and also did the DPPH and ABTS assay. Here we did the docking study with alpha-glucosidase enzymes, and we found that our binding score is comparable to standard. So here our compound is interacting with NH group and OH group of the hydroxytryzines. In addition to that, we also synthesize a few more compounds which are based on sulfa-nilamide and sulfa-acetamide, and recently we have published in Molecular Diversity. So here are the results of alpha-glucosidase and alpha-amylase activity, and also the results of DPPH and ABTS activity. So I'm not going in detail. For the same compounds, we did anti-inflammatory activity again by carrageenan-induced edema method. So here we found that our compound is reversing inflammation after 4 hours by 93 to 98%. Here we did the docking study with for the anti-inflammatory activity. So here we found that our compound is also showing binding comparable to standard Calax IP. So our compounds are good anti-diabetics. So here is the mechanism of alpha-glucosidase inhibitor enzymes. So our compound is act as alpha-glucosidase inhibitor. So alpha-glucosidase is a digestive enzyme which hydrolyze the polysaccharides and disaccharides to monosaccharides. So if we hinder the alpha-glucosidase enzyme, so it will delay the release of glucose from intestine to bloodstream. So this is the release of glucose in intestine when the voglibos, which is a standard drug which is used to inhibit the alpha-glucosidase enzyme. So how we can control the diabetes or manage the diabetes by hindering these enzymes? So by taking these type of inhibitors, we can reduce this peak height, and we can keep constant the glucose level in bloodstreams. So this is the probable mechanism for antioxidant effect. So our compound can capture this radical by donating hydrogen from this hydroxylamine moiety. So overall, the overproduction of free radicals. Here I have created the relation between anti-diabetic, antioxidant, and anti-inflammatory activity. So the overproduction of free radicals may diminish the antioxidative defense system. Actually, the excess presence of free radicals in blood streams also diminish the antioxidative defense mechanism, and it's caused the redox imbalance. And it also causes the autooxidation of glucose and metabolic abnormality, which leads to enhanced insulin resistance. So by managing the oxidative stress, we can also manage the diabetes. Hyperglycemia and oxidative stress also promote the overproduction of pro-inflammatory proteins with cytokines and chemokines, which leads to inflammation. So by reducing inflammation, we can also manage the diabetes. So in conclusion, diabetes type 2 can be effectively managed by defensive pancreatic beta cells from oxidative stress damage and inflammatory damage, by hindering alpha-glucosidase and alpha-amylase digestive enzymes. Thereby we can delay the glucose absorption in intestine. So such a combination of activity present in the sulfur drugs-based hydroxytrypsin derivatives could suppress the progress of type 2 diabetes mellitus. So overall, our study highlights multifunctional role of hydroxytrypsin as anti-diabetic, antioxidant, and anti-inflammatory agents. So these are our collaborators. Professor I.P. Pati is helping in doing anti-diabetic and antioxidant activity, and Dr. V. Murugashan is helping in docking study, and Dr. Amit Bhargav from Udaipur is helping in anti-inflammatory activity. Thank you. Thank you very much.
Okay, thank you, Dr. Baralia, for keeping everything in brief. It was a good talk. Any questions from audience? I think everyone is feeling tired after these 2 days of active activities.
Sure, sir. Thank you very much. May I now request the chairperson to kindly present him a token of appreciation to Dr. Barolia. Thank you so much, Professor Kaushik and Professor Barolia. We may have the next lecture.
Our next speaker this afternoon is Dr. Madhulata Shukla. She is Assistant Professor, Department of Chemistry, GV College, Ramgarh, Kaimur, VKS University, Bihar. She did her postdoctoral research at IIT BHU, and she has 23 international publications, 4 book chapters. 1 book has been edited by her. She is a coordinator or convener of CRI Bihar and Jharkhand chapter. And today she is delivering his short talk on green synthesis of silver and copper nanocatalysts using ionic liquid and their photocatalytic applications in reduction of paranitrophenol. Dr. Shukla. Yeah, you have got 10 minutes. You have got 10 minutes, okay? Okay.
First of all, I would like to thank R.D. Kaushik sir for the nice introduction. And I would like to thank the convener of CRSI 2023 and all the organizing members for giving me the chance to be here and present a lecture before you. So thank you all the teams of the FCASI. Today I will be discussing on the synthesis of silver and copper nanocatalyst using the ionic liquids and their photocatalytic activity. So first of all, I would like to explain why we are choosing the ionic liquid for the synthesis of nanoparticle. Generally, the people used to synthesize the nanoparticles in normal organic conventional solvent. Here we are choosing the ionic liquid. So what is the importance of ionic liquid? As we know that when we synthesize the ionic liquid, then we need some when we synthesize nanoparticles, then it has a tendency to aggregate, and the smaller particles get aggregated to a bigger particle. And for that, we require this stabilizing agent. And for that, we have to add extra stabilizer, extra chemicals we have to add. But when we are using the ionic liquid, then it can itself act as the solvent and also as the stabilizer and also as the reducing agent also. But sometimes it may not work as the reducing agent. So the importance of ionic liquid is that it acts as a solvent and stabilizer. Also, it can, due to the presence of cations and anions together, it works as the electronic as well as the steric stabilizer. Also, the percentage yield of nanoparticles is quite high in ionic liquids. Also, the reaction can be carried out at higher temperatures, because the boiling point of ionic liquids are very high, of the range of 150, above more than 150, means we can carry out the reaction at 300 degrees Celsius also for synthesizing the nanoparticles. This is the process for the synthesis of silver nanoparticles in ionic liquid. Here we have used glucose as the reducing agent, because initially we have synthesized without glucose, but the nanoparticles which were synthesized found to be unstable. After 1 month, it seems to be showing the different UV spectra. For that, we can say that that is not stabilized. This is the TEM image of the silver nanoparticle synthesized, and it is of the range of 55 to 135 nanometer range. And all the process has been, all the synthesized nanoparticles has been characterized using the 1S, 13C, IR, TEM, and all those images. This peak at the 423 nanometer represents confirms the synthesis of silver nanoparticles. And also we have found that from the NMR spectra, that when we compare the NMR spectra of pure ionic liquid and the ionic liquid containing the nanoparticles, it has been found that there is no peak shift, indicating that this, due to the presence of silver ion in ionic liquid, there is no changes in the cation part of the imidazolium ionic liquid. So this is the photocatalytic activity of silver nanoparticles. Degradation of paranitrophenol has been compared here. We see that in absence of light, there is less degradation, whereas in presence of light, here the degradation is quite high. And we can also clearly see here that from the graph, in absence of light, it follows the first order reaction, whereas in case of presence of light, it follows the zero order reactions. Now moving to the next synthesis in ionic liquid, cuprous oxide. We have synthesized the cuprous oxide also in the same ionic liquid using the same reducing agent, and we have found a powder form of spectra here, exciting spectra of the powder which we have obtained for the cuprous oxide. And here we see that there is huge difference when we are using the ionic liquid. The nanoparticle synthesized, this one is for the sample S1 without ionic liquid, and this is for the S2. Here the size is quite large and the shape is also very different. Here rod shape is obtained, and in this case of the range of 20 to 50 nanometer particle is being obtained here. So this is the UV visible spectra of these two ionic liquids, synthesis of this silver cuprous oxide nanoparticles, and this is cuprous oxide in ionic liquid. Next, here also we have seen that this is the this is the degradation of methyl orange with the S1, and this is in presence of, this is for the S1. This corresponds to the S1 and this corresponds to S2. S2 means CO2 synthesis in presence of ionic liquid. Here also we see that the CO2 in ionic liquid rate has been increased here. Degradation of MO has been increased when we are using the ionic liquid. So next we have characterized how these ionic liquids are performing, that what effect are being present in this CO2. For that we have carried out the DFT calculation, because the molecular structure studies is very important to analyze the properties of any moiety. So we have carried out the DFT calculation for that one, and these are the details we have written here. This is the density of a state plot. This is for the BMN BF4. Here a huge band gap is observed, but when it combines with the silver nanoparticles, then the band gap decreases to a lot. Next, for the CO2 also, the band gap is quite high, but when it comes in contact with ionic liquid, then the band gap decreases and it increases the photocatalytic activity also. Here we have represented the IR spectra of silver nanoparticles, indicating that only the BF stretching frequency and the C2H of the imidazole ring is being affected, and all the stretching frequency of the cation part remains unaffected. This explains that, means silver nanoparticles or cuprous oxide nanoparticle is surrounded mainly by the anions rather than cations. Here we have carried out the IR spectra calculation also. This clearly indicates that the first, second, this one, asymmetric stretching of B2A and C2S out of plane bending. These peaks indicates that only those peaks are being affected in ionic liquid which are associated with the silver nanoparticles or copper nanoparticles. So finally, I would like to explain that ionic liquid, a green solvent, is a superior medium for the synthesis of silver or copper nanoparticles, and it is mainly surrounded by the anion part rather than the cation parts. It is clear from the DFT as well as the IR and NMR spectra. And next, intermolecular interactions can be analyzed very clearly using the DFT calculations. Thank you. And first, I would like to thank UGC for providing the UGC startup grant and the computer facility from the IIT BHU, which we usually did the computational works. Thank you. And
well, thank you, Dr. Shukla. Any questions from audience? No.
Sir, can I ask one question? Here.
Yeah, I'm also writing. But in this session, no one is asking questions. Everyone is waiting for validatory function.
It's too much. One question. You said that band gap is directly related to photocatalytic activity. Yeah. Here I am Dr. Pragati. Yes. Okay. I'm asking. Okay. Yes. How the band gap is related to the photocatalytic activity? Can you please explain your
just a minute, ma'am. I more slide, but due to the lack of time, I have skipped all those things.
Okay.
Here, see here. Here, silver cluster is there, and silver and PVP like that. PVP, and this is for the another one. PVP is acting, degrading the band gap here. 1.33, 1.22, and here silver cluster and means in case of ionic liquid acts as a medium, then it helps in degrading the lower band gap between these two. So it activates the photocatalytic reactions.
Okay, one more thing I want to ask you. Ionic liquid, right?
Okay.
Reducing agent as well as stabilizer, you are saying. I didn't understand if I use ascorbic acid and water as a medium, it will not be same.
If you want to synthesize the nanoparticles of these various components, because using ionic liquid, we can synthesize nanoparticles of the range of 2 to 10. So that you cannot do with that salt.
And it will be any morphology, specific morphology, it will be there, or spherical shape of nanoparticle only?
No, not just a spherical one, ma'am. It can be of any size.
Okay.
Okay, then you can discuss it later on.
Okay.
Also, she will be there till validatory. No problem.
Thank you so much.
Thank you. Yeah.
Thank you so much, sir. May I now request Professor R.D. Kaushik to kindly hand over a token of reverence to Madhulata Shukla. Professor Madhulata Shukla. May we have the next speaker?
Good. Dr. Bhupesh S. Bhatt. Dr. Bhatt got his PhD from Sardar Patel University, Vallabh Vidyanagar, Gujarat, and now he is working as assistant professor in Department of Chemistry in the same university. 62 papers to his credit, and he is going to deliver his short lecture on spiroquinoxylin beta-lactam base heterocyclic spirins, synthesis, characterization, molecular docking, and biological studies.
Okay.
Dr. Bhatt.
Okay, thank you.
Please. And You should know that it's only
Yes,
10 minutes.
yes.
Not more than that.
I will try to finish
It can be less than that.
Yes, thank you. Okay. I'm very much thankful to organizers of FKC and especially Dr. Satpal Badsara sir for inviting me over here. Today's I'm going to give some brief about quinaxolone beta lactam cyclic spirans. Okay, so why I have chosen this spirocyclic molecule? They have covered almost all the branches of science, and you can see the applications includes. You can see many applications of spirocyclic compound. Over here, we can see some spirocyclic compound based drug molecules which are used as anti-cancer agent, anti-inflammatory agent, and quinaxolone moiety based drug are also markedly available. We can have some of the drug over here which are used as antiseptic agent, resi dogs, and beta lactam ring is a very privileged scaffold that we know. And there are mainly these 4 classes of beta-lactam antibacterial drug, that is cephalosporin, penicillin, carbapenemins, and monobactam. So we have combined all these 3 scaffold in a single molecule to synthesize this molecule with a very simple and scalable method in which the ninhydrin reacts with orthophenylene diamine to gives us this indinoquinaxolone on. And then this reacts with substituted phenyl hydrazine to get this hydrazone molecule, and then beta lactamization by this acetyl chloride. Then we have performed many biological activities like biomolecular interaction studies, antibacterial molecular docking studies, anti-cell proliferative studies. First, we have started with DNA interaction studies because we know that most of the drug exert their activity by interacting with DNA. This is UV visible spectra of constant concentration of our spirocyclic compound with varying the concentration of DNA. And when we have evaluated some of the parameter, that is epsilon A, that is apparent extinction coefficient, epsilon F, that is extinction coefficient of DNA in a free form and in a bound form. And when we draw a spectra between this and this 1 upon epsilon B minus epsilon F, we have got a straight line, and the slope and slope to intercept ratio will give us this Kb value, and Kb is the measure of strength of DNA on the strength of interaction of between this spirocyclic compound and DNA. Okay, in a similar way, we have performed this emission competitive binding assay by fluorescence spectroscopy, and over here DNA does not give any emission spectra, and that's why DNA intercalated ethidium bromide intercalated DNA we have used over here, and we can see that our molecule can successfully displace the ethidium bromide from over here, and it intercalates in between the of DNA base pair. In a similar way, we have performed this viscosity measurement, and over here again, due to a chain lengthening due to intercalation of compound, we have increase in the relative viscosity. All the data are comparable to the standard drug like ciprofloxacin and doxorubicin. Then in a similar way, we have performed this BSA interaction studies because in our blood, the major proportion is a plasma protein. And over here, we have also got this moderate Kb value, sorry. Okay. And over here in the case of fluorescence fluorescence study, we don't require ethidium bromide because the presence of amino acid gives the emission spectrum. And over here, we have also got some good result in the molecular docking study. We can see that this is the docked picture of our molecule with a DNA over here, and we can see that major interaction are van der Waals interaction and pi-pi interaction, and in some case we have hydrogen bonding also with this cytosine and guanine. This is the dog picture of spirocyclic molecule with protein molecule, and we can see that again the major interactions are pi-pi interactions and van der Waals interaction with all this amino acid residue like leucine, lysine, arginine. And then we have performed the same docking study with a topoisomerase 2 enzyme, because topoisomerase 2 enzyme links with the
DNA replication processes, and most of the antibacterial drug exert their action by interacting with this topoisomerase 2, and the binding energy are also in good accordance with the standard drug which we have chosen. Okay, so up to here we have got many encouraging result, but as we have jumped into the biological activity, the first, that is antibacterial activity in terms of minimum inhibited concentration against 5 different gram-positive and gram-negative bacteria, we have not got any encouraging result. The compounds are not active at all, but they have moderate activity as far as the anti-cancer activity is concerned and cytotoxic activity is concerned. MTTSA was performed on MG63 cell line, and this is on brine shrimp. Okay. Then some brief information about structure activity relation. Then the substituent fluorine gives us overall good result in biological activity. Electron withdrawing gives us good DNA binding activity, but low LC50 and IC50 value. And electron releasing group, the molecules having electron releasing group does not have good biological activity at all. Okay, these are some of the pharmacokinetic pharmacokinetic profile of drug. You can see that good thing is that our molecules follow Lipinski's rule. They does not violate Lipinski rule of 5, and they have quite good pharmacokinetic parameter. You can see that absorption, human intestinal absorption is good. This CACO2 and human oral bio availability is less. Over here, there is a problem because it is having a very higher binding with the plasma protein. That can be a problem. Over here, this CYP3A4 inhibitor, this is a major class of cytochrome P450, and they does not this enzyme links with the metabolism of any drug. But over here we have got good activity. Excretion parameters are also good, and it is having moderate toxicity over here. Okay, we have evaluated all this hepatotoxicity, acute oral toxicity, eye irritation. Okay. So conclusion is that this heterocyclic spirins are very good DNA intercalator. Pi interactions and van der Waals forces stabilizes the complex with spirins. They are good cytotoxic agent, follow Lipinski rule, having good ATMET profile, but they are not a good antibacterial agent. Okay. Last month, our department has organized a conference where we 4 generation were assembled over there. Me, along with my research student Ravi, which this is my research student Ravi, which has performed this work. Along with me and my student, this is my research guide. This is the research guide of my research guide, and by the God grace, his research guide is also alive. Okay. And Gujarat is famous for tourism places. This is Rani Ni Vav, which is printed on our 100 rupees note. This is the renovated Pawagadh temple and Statue of Unity. And I welcome you all whenever you want to come over there. I will be happy to host you all.
Wow.
I am very thankful to Atrocity team and Sarpal sir for giving me opportunity to be here. I'm thankful to Department of Chemistry and UGC Dr. Vegopar. Only MTT essay was performed by Dr. Vegopar. All the other biological activities are performed more or less by my student doctor. Thank you all.
Okay, okay. Thank you, Dr. Bhatt. Okay. Just one question. No? Okay then.
Thank you, Dr. Bhupesh. May I request the chairperson to kindly present a memento as a token of reverence to Dr. Bhupesh.
He is already there. You are taking behind.
Yes, yes.
Next speaker is Dr. Richa Saini. She is assistant professor in Department of Physics, Gurukul Kangri University, Haridwar. She is working in theoretical condensed matter physics in low dimensional systems of material sciences, published over 24 research papers. She has got over 12 years experience. Today's title of talk is Phenomenon of Heat Conduction in SI Nanowires.
Thank you, sir. Now, good evening, everyone. And thanks for organization and Dr. Satpal sir for giving me opportunity to present my theoretical work on this platform. And I'm the last presenter of this conference. So now let me start my work, Phenomena of Heat Conduction in Silicon Nanowires. Introduction in the brief, thermal properties of thin films and microstructures, especially thermal conductivity, have played an important role in understanding the performance, functionality, and reliability. And the phonon transport is drastically influenced due to the various effects, namely size effect and increased boundary scan and quantization of phonons as well as electrons. Now, the thermal conductivity in nanostructure is mainly a function of both boundary and internal phonon scattering mechanism. As the diameter of nanowires compress, its results in higher surface into volume ratio, which offers significant reduction in thermal conductivity. Some theoretical investigations reveal that as the diameter of crystal decreased by 20 nanometer, this lead to modification in phonon dispersion relation and reduction of phonon group velocities as well as due to phonon confinement. Now the thermal conductivity of nanoribbons has been observed by the effect of age, clarity for both zigzag as well as armature edges. And the simulation by the Monte Carlo methods also used to demonstrate thermal conductivity of silicon nanowires, which suggests that the confinement effects are significant when the nanowire diameter approaches tens of nanometers and reduction reaches up to the 40% for 10 nanometer thick through phase nanowire at room temperature. Now the lack in the investigation of heat thermal conduction in the form of in-plane and cross-plane scenario, simultaneously along with the inclusion of boundary effect due to the phonon confinement in nanowires and phonon relaxation times. Phonon relaxation times that is calculated by tau. The effects of these scattering phenomena on thermal conduction into in-plane as well as cross-plane directions are analyzed, numerically estimated, and excellent agreement with experimental data is achieved. And the formulation of the problem. Now the interpretation of the present work is based on the fact that the consequent of confinement of phonon and electron motions are greatly limited in such regions, due to which phonon transport activity is subsequently affected in the restrained directions. And the formula by thermal conductivity of nanostructure as by the K is K perpendicular and as well as K parallel in the direction of Ki, where I is represented by perpendicular and parallel directions. And this formula is calculated by the Callaway modified model, and in the view of this Callaway modified model is Ki Kb upon 2 pi square Vi and beta h cross square, and omega d, and d is the defect phonon for, and gamma k omega E exponential beta h cross omega I minus 1, and this. But using many body quantum dynamics of electrons and phonons, the line width for included numerous scattering mechanisms have been obtained with the help of green function via the modeled Hamiltonian, and that is the equation. We do not go from for the deep calculation, and the phonon frequency line width, gamma k, various scattering process, and Cb is the, and combined boundary scattering, and D represented by defect, A anharmonic, ADN anharmonic defect. For both calculation, parallel and perpendicular. Phenomena of scattering, we calculate boundary scattering by this formula. Tau inverse CB is equal to summation of I, tau inverse CB I, and this is equivalent to Li inverse Vi. And in case of nanowires, the Kashmir length can be expressed by perpendicular in 1.12 and 1/2, and L parallel is equivalent to 1.1. For L1, L2, 1/2, with D is the diameter of nanowires. Point defect scattering as well as calculated for the nanowires. Imperfections, defects of substitutional isotopic points impurities present in the solid play a deterministic role to limit the thermal conductivity both in bulk and heterostructure systems. And the new form for the line width into the plane and cross plane direction is the gamma D defect omega. Omega is the wave vector, and this is equivalent to A1, A2, A3. These are the constants. Now the phonon-phonon scattering for nanowires at subsequently higher temperature at the lattice vibration models, dominantly pursued and phonon-phonon collisions in the anharmonic phonon, and this is the formula for gamma 3 anharmonic phones, and the modified formula for the lifetime or phonon-phonon scattering for nanowires in 3 anharmonic function is equivalent to summation of I gamma k 3I omega I. Interference scattering. Interference scattering is by the described form of gamma 3 defect formula. This is equivalent to 144 pi epsilon omega, and this is the V3C and S plus alpha and omega plus alpha and minus alpha and omega minus alpha are the parameters for this calculation. Considering the reshaped phonon dispersion relation and adopting some mathematical simplification, the line width into in plane and cross plane direction takes the form for the 3 defect functions. Analysis of thermal conductivity for silicon nanowires, the phonon confinement in a justified manner. The theoretical study of the thermal conductivity of silicon quantum wire has been taken up use of 2 reasons. Firstly, silicon and germanium material systems exhibited a great promise for high temperature thermoelectric applications. And secondly, for the problem of lattice thermal conductivity in the semiconductor solid is imperative in a view of continuous downscaling of the future size. And to justify and analyze the investigations of the proposed model and to figure out the thermal conductivity of silicon nanowire with diameter of 37 nanometer, 56 nanometer and 115 nanometer into the in plane and cross plane direction. And for the experimental data, these are the theoretical calculation, but for analysis the with match the experimental data we have taken from the work of Lai, and these are the due to the some lack of information of experimental data, we take some parameters of and constants to use the analysis of thermal conductivity of silicon nanowire. This is the table of parameter and constants, and these are the results for 37 nanometer silicon wire, and this is the 56 nanometer silicon nanowire. And the conclusion of this work is the influence of various phonon scattering events responsible to offer thermal resistance has been investigating, considering modified phonon dispersion relation along with phonon confinement in nanowires. And the analysis of thermal conductivity for the 3 sets of nanowires with diameter of 37 nanometer, 56 and 115 nanometer has been carried out successfully and exhibit excellent between the theory and experimental result. And this model, that means modified Callaway model, applied to other semiconductor nanowires also to the analysis of thermal conductivity. And this is my paper. Thank you.
Well, thank you, Dr. Richa. As usual, I don't expect any questions from audience. immediately after this, everyone is expecting the valedictory function. Thank you very much for this brief talk.
Thank you so much, sir. We have a very cooperative audience, no doubt. I request once again to you, please hand over a token of gratitude to Dr. Richa Shaini. I now request Professor Meenakshi Jain, Head of the Chemistry Department, University of Rajasthan, and Dr. Satpal Singh Badsara to kindly come over the stage and hand over a token of reverence to Dr. R.D. Kaushik. Thank you so much, sir. Thank you so much, ma'am. And now we are moving towards the end of this conference. Ladies and gentlemen, we are very shortly about to begin our valedictory function. Please have patience and be seated. If you are outside this auditorium, please come inside and take your seat. We are about to begin with our valedictory function. At the very beginning of the inaugural function, I said that this is going to be a marathon. And I believe it has been a marathon for all of you, a marathon of many sessions altogether. Was it? Well, as I said, it has been a hectic schedule for all of you, ladies and gentlemen. Now, it is the time when we draw curtains to this two-day international conference, Frontiers at Chemistry, Allied Sciences Interface for FCASI 2023. I extend my warm welcome to each and everyone present here and hope that you have thoroughly enjoyed all these sessions. May I take this opportunity to invite may I request all of you to please be seated in the front rows. Thank you for being a cooperative audience. I now take this opportunity to invite our dignitaries, Professor T. Punyamurthy, Professor P.M.S. Chauhan, Professor R.D. Kaushik, Professor Rodney Fernandez, Professor Anil Kumar, Professor Minakshi Jain, Dr. Satpal Singh Batsara, Organizing Secretaries Dr. Asha Gurjar, Dr. Pragati Fageria, and Dr. Ria Salani to please take seats on the dais. Ladies and gentlemen, may we have a big round of applause for them. Please show me your energy. Please take this. Please take this. See, we have so many eminent speakers and experts here that even the chairs are less. I extend my warm welcome once again to all the dignitaries. So instead of wasting much of the time and taking your time, I now invite Professor Minakshi Jain, Head of the Chemistry Department, University of Rajasthan, to give a formal felicitation to Professor T. Punyamurthy, who have joined us here from IIT Guwahati. In this queue. Next, I would like to invite Dr. Mamta Raka to kindly felicitate Professor P.M.S. Chauhan. Coming up to the next, I would like to invite Dr. Ganesh Jangir to felicitate Professor R.D. Kaushik, who have joined us here from Gurukul Kangdi University. I would like to invite Dr. Ria Salani to felicitate Professor Rodney Fernandez, who have joined us from IIT Bombay. Next, I would like to invite Dr. Pragati Pagaria to felicitate Professor Anil Kumar. I also invite Dr. Satpal Singh Batsara to felicitate Head of the Department, Professor Meenakshi Jain. Thank you all. Now, after this formal welcome ceremony, I further proceed. And at this point, I would like to invite Organizing Secretary, Dr. Asha Gurjar, to present the report of first day. Dr. Asha Gurjar. Good evening, one and all. It is indeed my privilege to present the report of the 2 days international conference FCASI 2023. The Department of Chemistry, University of Rajasthan, along with the Indian Society of Chemists and Biologists, Rajasthan chapter, and Chemical Research Society of India, Rajasthan chapter, organized the 2 days conference of Frontiers at the Chemistry Allied Sciences Interface, FCASI 2023, on 20th and 21st April 2023. We are very happy to share with you all that conference received overwhelming response from all the quarters. The proceedings of all the sessions were live on YouTube. Day first, that is 20th April 2023, started with the grand inaugural session. The conference was inaugurated by पूरा starting था Day first, that is 20th April 2023, started with the grand inaugural session. The conference was inaugurated by Padma Shri Professor V.K. Singh and Professor Ganesh Pandey as the distinguished guest. Professor P.M.S. Chauhan graced the occasion as the guest of honor. The inaugural session began with Kulgeet of the University of Rajasthan, Saraswati Vandana, and lamp lighting. The chairperson, Professor Meenakshi Jain, presented the welcome speech. The conference convener, Dr. S.S. Batsara, briefed the audience about the conference. This was followed by the felicitation of esteemed guests. Our distinguished guest, shared their ideas and views about the conference. Thereafter, the abstract book of the FCASI 2023 conference was released, followed by the presidential address of Professor Rajiv Jain, Vice Chancellor, University of Rajasthan. The inaugural session was followed by 16 technical sessions spread over 2 days. The very first keynote lecture was delivered by Professor V.K. Singh. The second keynote lecture was delivered by Professor K.R. Prasad. The second session, that is Royal Society of Chemistry session, was chaired by Professor P.M.S. Chauhan. In this session, Professor Ganesh Pandey delivered keynote lecture, and Professor Mark Telfer, Professor A.K. Sahu, Professor B.L.V. Prasad delivered their plenary lectures, and Professor Alakesh Bisai delivered invited lecture. Second session was followed by lunch. During lunch session, third, that is ACS poster session, was organized parallelly. Session fourth, that is New Journal of Chemistry session. Session fourth, that is New Journal of Chemistry session, was chaired by Professor R.T. Pardasani. In this session, one keynote lecture was delivered by Professor L. Ackerman. 3 plenary lectures were delivered by Professor V. Gandon, Professor C. V. Ramana, Professor M. S. Singh. 3 invited lectures were delivered by Professor C. M. Reddy, Professor Amit Kumar, and Professor D. Banerjee. Session fifth, that is Elsewhere session, was chaired by Professor D. Kumar. 2 plenary lectures were given by Professor T. Punyamurthy and Professor S. Hota in this session. Session 6th and session 7th were organized parallelly, and the sessions were chaired by Professor Rakhi Gupta and Professor Asha Jain. 4 invited lectures were delivered in these sessions by Professor S. Pandey, Professor M. Bhanuchandra, Professor N. K. Sharma, and Professor P. Chauhan. The day ended with the dinner gathering. Thank you. Thank you very much, Dr. Gujjar. I now invite Organizing Secretary, Dr. Ria Sailani, to present the conference report of the second day.
Good evening to everyone. In the continuation of Day 1 report of International Conference FCASI 2023, Day 2 started with Session 8, that means RSC poster presentation. It was chaired by Professor Alkesh Bisai, Professor Ajay Kumar Srivastava, and Professor Siddharth Sharma. Around 60 delegates presented their research work and new ideas through beautiful posters. In Session 9, approximated 25 young researchers and faculties presented their research through oral presentations, and this session was chaired by Professor Mark Telfer and Professor R.K. Gunsaria. And also in parallel Session 10, approximate 21 delegates presented their research through oral presentations, which was held in Hall 1 in HRDC, and session was chaired by Professor Debasis Banerjee and Professor Lakshmidhar Rao. Session 11, that means CRSI session, was chaired by Professor A.K. Sahu. In this session, 2 plenary lectures were given by Professor Chin Fali and Professor K.N. Singh. Professor Anil Kumar and Professor R. Janapur Reddy were delivered invited lectures. Session 12 was ISCB session and chaired by Professor T. Punyamurthy. And in this session, 4 invited lectures were given by Professor R. A. Fernandez, Professor Lakshmidhar Rao, Professor Ajay Kumar Srivastava, and Professor D.K. Tiwari. Session 13 was organized as a ISCB poster session. Around 80 posters presented by young researchers, and these were evaluated by Professor Alakesh Bisai, Professor Ajay Kumar Srivastava, and Professor Siddharth Sharma. After the lunch, Professor Anil Kumar chaired the session 14, and 5 invited lectures were delivered by Professor Jiban Jyoti Panda, Professor Sandeep Murarka, Professor Ajay K. Saha, Professor Siddharth Sharma, and Professor Virendra Singh. The parallel session 15 was held in
Hall 1 at HRDC and chaired by Professor R.A. Fernandez. 4 invited lectures were given by Professor Ritesh Singh, Professor Sanjay Pratihar, Professor Suresh Kumar, and Professor K.C. Bhardwaj. The last session, session 16, chaired by Professor R.D. Kaushik. In this session, 2 invited lectures were delivered by Professor R. Guru Brahmam and Professor Amit Patwa. And 4 short invited lectures were given by Dr. Prabhat K. Barolia, Dr. Madhulata Shukla, Dr. Bhupesh S. Bhatt, and Dr. Richa Saini. In short, approximately 400 delegates from all over India and abroad attended the conference. This 2-day scientific program included 4 keynote lectures, 11 plenary lectures, 25 invited lectures, and 4 short invited lectures. A total of 40 oral presentations and around 200 poster presentations were held by young scientists and students in different technical sessions. To encourage scientific research, best oral and best poster presentations will be awarded to young researchers. Presentations were evaluated by a panel of esteemed judges. We thank them for their help and support. I hope this conference provided an excellent academic forum to the researchers, scientists, and industry personnel in understanding the interface, phenomena in chemistry, and other related science. Thank you so much.
Thank you so much, Dr. Selani. Ladies and gentlemen, now it is the time when we hear out the insights, thoughts, and remarks of our esteemed professors. May I now request Professor T. Punyamurthy to kindly address the gathering.
So thank you very much, Madam. Very good evening to all of you. It was a wonderful conference. We have gathered to conclude this event. As all of you know, conference is the place, forum, where in short period we'll be able to make new friends. This is very important in present situation to continue our excel, the research. And always whenever we meet, we will tell we met in Jaipur. So in this way, I am sure all of you got new friends from this conference. This is very, very important. In addition to that, you are carrying a lot of chemistry. Definitely, I am sure you will use it. This is also very important. As research, these two are very important to excel, and both you have got. Maybe that quantity will vary. Maybe some have got 2 friends, some maybe more friends, and we have got. This is very, very important. And also, as you know, we have excellent talks from India, from abroad. Can you get it by reading books? No. So we got all the things, and the talks were very excellent. Whatever the speakers, they have picked very nicely. Some of the areas, and what do you know, now we talk about sustainable chemistry, where the electrocatalysis, photocatalysis, very important, and the pioneer in the world, he gave talk. So what you can expect, right? So we have learned so many things we got from this in 1.5-2 days time, and we also should always remember, and we have to apply. This is what we give respect for this. Whatever we spent the last 2 days time. And otherwise, all the talks were very excellent. In addition to that, and the nicely arranged oral presentation, they have put pioneer Mark Tellifer. He is the world well known in cross-coupling reactions. He was chairing the session. I was very impressed. The students were very competitive with the faculty members when the presentation. They are our future. I think nicely they have arranged. And some of the students even more, and the presentation was very clear and more confident comparing the teachers. They put some associate as professor, together they were competing. So that is very nice and very, very important observation. And the chairman, neutral person abroad, is well-balanced person. And he asked some of the questions. They were very useful for the students. Don't worry, no need of answering all the questions. But if you remember the question, that itself learning for us. In addition to that, I have gone through the poster display. The students were very enthusiastic and seriously drawn the posters. And earlier time we were not much. Now that means, and we are moving in the right direction. More conferences needed. I am sure in this particular conference we have got a lot of things. Me particularly, I learned a lot of chemistry, and I am sure all of you also. I also met some friends after a long time, after COVID, 2.5 years. This is very important. It gave opportunity to meet and also made new friends. Overall, I feel this conference was very nicely arranged. I would like to end congratulating Satpal for the excellent arrangement, hard work, effort, and the energy. And he spent his team. I congratulate once again and thank you very much.
Thank you very much, sir. Your words means a lot to us. May I now request Professor P.M.S. Chauhan for the next speaker.
Good evening. On behalf of ISCV, I congratulate Dr. Satpal Vatsara for excellent organizing excellent conference. As you know, when conference organized, it required the 4-month work and team of 50 people. Just like when building erect, the foundation stone you cannot imagine. But when foundation stone is strong, the building is strong. So I congratulate the all team of this organizing committee. Please stand up and give the big applause, please. Congratulate all members of the organizing committee who work day and night and make your stay comfortable. Thank you to all. Organizing the conference, not the simple work. Sometimes the people cannot imagine. I remember 2008, Professor Anil Kumar organized a big international conference, the BITS planning, and at that time 850 members participated at that conference. The age of Anil Kumar is 35 at that time. And he is very much worried about how it is possible to handle the 850 people BITS planning. The most of these guests, they are coming either in Jaipur or in Delhi. I invited one of my friends, Stephen Brice, now the director, Karlsruhe Institute of Technology. I think his group is around 200 people right now, presently. And he came around 3 a.m. He arrived and he is so afraid, Prem, how it is possible you organize this big conference in remote place. And these wonderful people, now the professor and very senior professor. And now I am looking the Satpal. He is also around the same age group and he also organized a wonderful conference. So I don't want to say a lot of things, but one thing is excellent. And the present of this valedictory function, the 90% the student is present, this again wonderful thing, and only 10% the senior people I'm looking. So it's good because the young generation is so much interested and stay here the last function validity. So I again congratulate all our participants and please to our ICV is organizing the international conference January 7, 8, 9 Rajkot. That conference is again very big and we already received the acceptance of 75 speakers from outside country and we are expecting 1,200 participants in Rajkot and the venue of that conference is Marwari University. So we again invite you all for participating conference. Thank you to all. That's
Thank you so much, sir, for your appreciative word. You have filled us with joys and energies at the same time. May I now request Professor R.D. Kaushik to bless us with his kind words.
Dignitaries on the dais, of the dais, delegates, students, research scholars. Professor Chauhan, he has thrown much light on the on particularly on this conference. And really when we see persons like Satpal Basra, the young man here, I always remember my earlier days when I was young and I started doing this type of ventures. And today really I am happy that Dr. Basra has excelled in this field also, in addition to the excellent research work he is carrying on in this university. As far as this conference is concerned in general, we are at Haridwar. We are having Science Congress Haridwar chapter also, and around 60 conferences we have organized so far. And in Haridwar chapter, 13 conferences we have organized, including many international conferences. So I know the labor that one has to put in while planning and organizing a conference. Now, here this particular conference has been very meticulously planned and very nicely organized. There are many teams working for the success of any conference. For example, accommodation. It is being taken care of by so many people. You will not find those students or those volunteers here at the time of inauguration and at the time of rather valediction. Similarly, the transport committee. Those members you will not find in this hall. They are always working on the roads, carrying the delegates from one point to other, taking care of everything. Then food committee. Then, of course, technical session committee. And here, I congratulate the organizers in general and Dr. Satpal Basra in particular for, in my view, he's getting 10 out of 10 marks for every committee that he has founded. This is, sometimes they say that organizing a conference is only a mela. No. This is much more. If you just see only technical sessions are going on and that's all. Not only that thing. You need every committee that I have just mentioned, transport, accommodation, food, technical session committee, every committee, we expect every committee to perform an excellent work. Otherwise, the conference is not success. If you are not satisfied with your accommodation, you are not satisfied with the food, not satisfied with the transport, then naturally, when you are sitting in the sessions, technical sessions, you are not satisfied there also. This is my personal view. And because here the volunteers, and particularly we must congratulate the volunteers here, because they are behind everything there. They are having every delegates, every VIPs, every scientist phone numbers in their pocket. They are frequently calling us, sir, when will you be ready? We will pick you, this and that. In that way, I must congratulate Dr. Basra for this. And I must congratulate this university also. Very well supported by the chemistry department. The HOD is there here. I hope such type of conferences will be organized in future also, particularly by Dr. Satpal Basra. Thank you very much.
Thank you so much, sir. You have not only inspired us, but also motivated us to take up such kind of academic fest time and again in the near future time. And now, ladies and gentlemen, this is the time when we are supposed to hear few remarks and feedbacks from our beloved students or the budding scientists. I request anyone who has something to speak, please come and say it out. Thank you very much. May I know your name? Okay, sure. Please introduce and let's speak.
Okay. Thank you, ma'am, for giving me this opportunity. This is Anuvasita Parekh from BITS Pilani campus. And good evening, everyone. I would like to thank all organizing committee of FCASI 2023 on behalf of research scholars from BITS Pilani. It was a pleasure to be a part of this conference, as we are aware that the heat wave has started to hit the Rajasthan. Even after this conference was very comfortable for us, and this comfortness has given us opportunity to absorb the work more from various streams, and all lectures and keynotes are arranged in single hall, which is, as for me, it has helped me to not to get confused that where is my lecture, where I need to go and where I need to attend. So when it is in single place, I don't need to go here and there. This is also best part of this conference. And finally, it will help me to find that what kind of research has been going around in this world and in India as well. And finally, I would like to thank for the nice food which is given to us. It was really satisfactory. Thank you so much.
Thank you so much, Anuvasita, for your lovely feedback. May we have someone else? Is there anyone? Please come, please come.
Very good evening, all. Honorable dignitaries and all the guests. Very good evening, all of you. First of all, sorry, I'm not a PhD student. I'm a student at IIT Madras pursuing BTech. So I just came with my guardian here, and I observed it was very fabulous presentation given by all the researchers. I was literally flattered when I saw some of research based on magnet, and some was giving a fabulous research work, had done very fabulous research work on Monte Carlo. I heard, ma'am, your work also. And apart of that, I also observed how to represent the different kind of presentation. So thank you all to teach me how to do research work in my future. I will pursue such kind of things. Thank you and have a good day.
Thank you so much, Chetan. We are actually thankful to you. Not only we are blessed, but I believe the nation is blessed to have such youngsters who have such kind of mindset. Thank you very much for being here.
And now, taking this ceremony further, I would like to invite the convener of this conference, Dr. Satpal Singh Batsira, to take over the session.
Thank you, Dr. Priti. Hey, I will rather say warm good evening. Yeah. Dignitary of the dais, Professor Punyamurthy, Professor Kosik, Professor Chauhan, Professor Panadis, my team, Dr. Asa Guja, Dr. Pragati Sagedia, Dr. Riya Salani, head of the department, my good friend, I would rather say Professor Anil, all the dignitary and all the delegates, my dear speakers, and my dear friends. Before I move further for the official proceedings, definitely I'll take this opportunity to speak something. And no doubt I'll be stealing this part from Conference Organizing Secretary Pragati Fagedia's things, because he'll be going to perform a lot of things. I'm sorry, Pragati, I'll be stealing few things. No doubt stealing me from a lot of things. Thank you everyone for your nice comment. I was nothing, I was just a face. There are people behind the scene. They work very hard, day and night. I must take this opportunity to take their name. Abstract things was done by my secretary, Pragati Fageria, and no doubt Poonam Kumari from my department. I request if students are there, just rise up for a while so everybody can see Poonam and Asa Kumawat. So the beautiful abstract book let appear due to their hard work. Thank you. Then the next comes the abstracts. Registration. Registration was taken care by fully by Pooja. She is my student. Akanksha. Thank you. Well done. Yeah, thank you, Pooja. Then local hospitality, Dr. Lalit. And I must inform all of you, there are my good young friends from MNIT, Lalit, Deepak, and Nitin. If they are here, they are from MNIT Chemistry Department. Akanksha, yeah, if I miss some name, please forgive me for that. Coming to the next things, my MSc students, they are here from last 2 days, no doubt. Whatever we ask, they are always ready to help us. My colleagues, Dinesh Jangir, Navneet Chaudhary ji, Mamta Ranka ji, Ake Gulsara ji, my good friend from Amanat Sudhir Kasab ji, Rajkumar Joshi ji is here. And yeah, there are many more. My friends from colleges, Dr. Mudit Gupta, Anil Bansal, Kumar ji, Hemlata ji, and many more. From my department, pattern of this conference, Professor Sharma ji and Professor Nirmal helped me a lot in the organization of this conference. What I learned from them in the last couple of years, which I could present in front of all of you over here. Head of the department, no doubt, and Honorable Vice Chancellor. So I can say that what you have seen, that everything has been done by these people. Again, I'm saying if I miss someone's name, please forgive me. So whatever good happened, that credit goes to them. Please give a huge round of applause for them. And if there are any shortcomings, I take full responsibility. Please my deepest apology. If there was any inconvenience, my deepest apology. See, all the information were shared by website. And I'm happy to share the website was designed by my brother-in-law. My younger sister husband, he has a startup here, and he got done everything for me. Night 9, I asked him, 10, I asked him, they have a small kid, still they helped me a lot. So this was all about the Team AppCast 2023. Yeah, without delegates, there is no conference. We need money, no doubt about that. Someone has to speak, someone has to listen. Thank you everyone for being over here and spending your valuable time with Team AppCast 2023. Now coming to the proceedings. So basically I'm here for to announce the best presentation awards. So basically, as I told in the inaugural ceremony, that AppCast 2023 conference was well supported by many sponsors. At University of Rajasthan, we got funding from Royal Society of Chemistry, SAB, ACS, RFC, then Thermal Officer, TCI, and yeah, I think I may miss one more name. REACC, yeah, Elsevier, everyone supported us. So SCS and RSC has supported us in form of some best presentation awards. So basically, the organizing committee have divided them in oral presentation and poster presentation. So in oral presentation from RSC and JC, yeah, one more thing. So when conference are going on, meanwhile, and JC associate editor, my good friends and my teacher, I would like to say, Professor was here. And he said that I want to add NJC and I want to, because the presentations are nice, and he announced this thing after today's lunch session that he will be sponsoring some prizes from the NJC also. So we named it RJC NJC Best Poster Awards, RJC NJC Best Oral Presentation. Similarly, there are SCS Best Presentations. Then our own society, Indian Society of Chemists and Biologists. Professor Chauhan is here. He is the secretary of ISCB. He called me and he asked, कुछ भी चाहिए बताओ. मैं बोला सर हमको ISCB की तरफ से अपन अवार्ड देंगे. Can you believe? He himself get everything done, printed the certificate, get the certificate from Lucknow to here. Thank you very much, sir. So we will be having some oral presentation from ISCB as well as best poster presentation and the best oral presentation from ISCB. No doubt, in AppCast 2023 also believes that there must be some sort of what you call appreciation from our side also. So there will be some sort of best oral as well as poster prizes from the Team AppCast 2023. So now I will be announcing the results. I must thank the experts who have evaluated all the presentation, and like Professor Ajay, Professor Siddharth, Professor Sandeep have evaluated the posters, and no doubt oral presentation were evaluated by the chairmans of the particular sessions. Yes, the poster presentation, about the poster presentation, the experts suggested there are wonderful posters. All are perfectly well designed, but there are some limit. So please, there will be some names, but it doesn't mean that others are not better. All are equally good, but no doubt we have some number. So I will be reading those names. So I will start with the AppCast 2023 Best Oral Presentations. And no doubt we have decided not to make it 1, 2, 3. It's a collective. So everyone is equal. We do not give any numbering to them. That is the first prize, second prize, third prize. All are equal. So the AppCast 2023 Best Oral Presentation. Before I start, I may request the dignitaries, Professor Fernandez, Professor Anil, Professor Munimurthy, and everyone to kindly hand over the certificates to the awardees. Before that, I just forgot to inform you that you will be wondering that what you will get in form of award. So from ACS poster, ACS will be giving a certificate plus in the 2,000 rupees, and this money will be credited through RTGS to your accounts. So Team APCAS will contact to the awardees, will receive all the details, bank account details, and it will be sent through RTGS transaction. So it will take at least 10 to 15 days. I hope you will bear with us. Same for RJC, NJC awards. Again, there will be a certificate and rupees 2,000 cash. I will say money, I can rather say. And they also promise us that they will be sharing some vouchers. And again, the same thing, they ask us to share the email address of the particular awardees. And I believe they will be directly sharing those voucher details with the awardees. From ISCB and FKC, there will be a certificate and memento. Okay. So with this, I start with the FKC 2023 Best Oral Presentation. Oral Presentation 2. Surbhi Bhatt from Central Institute of Rajasthan. Please have a big round of applause. If she is there, please come. And if she is not there, any one of his or her friend can receive that the things on behalf of Surbhi. So one sir, please. Oh. I think it will be continuous. So I request please come forward. So it will be easy to give handover from here. Prof. Everyone, please, yeah, yes, and make it fast because we are different days. Yes. No problem. Okay, so ठीक है. Then Professor Anil and Professor R.D. Kosik can start. Then stop. Yeah, so Professor Munimurthy can join. Please join. I'm sorry. Yes. Up. Then. Okay, then please. Next, Sanjita Maharana, oral OP10 from IIT Bombay. And next is oral presentation 5, Anuvasita Parik Bisplani. Akanksha Yadav, OP8 from University of Rajasthan. Yeah. Congratulations. Then for so there are many. So I think the oral presentation. आप लोग oral का आप लोग कर लीजिएगा ना. पुण्यमूर्ति आप तीन लोग oral कर लीजिए. Then poster. Yes, that is better. Yeah. Coming to ACS Best Oral Presentation OP 2025, Arakdeep goes from University of Hyderabad. If he is there, yeah, okay. OP 16, Sonia Rani, NCL Pune. OP 28, Dr. Rekha Bai, National Chung Sing University, Taichung, Taiwan. Coming to RSC NJC, best oral presentation, OP4, Biplab Dera from CU, Gujarat. If he's not there, any of his friend can join. OP6, Deepak Sharma, MNIT Jaipur. Coming to the best poster presentation, now I may request Professor R.D. Kaushik, Professor Pranadhisan, Head of the Department, Professor Oxygen, kindly hand out the certificates and mementos. Yeah, so from FKC, it is Satya P. Panda, IIT Jaipur, P132. Yeah. P40, next is P42, Jyotirangana Misra, CSM, CRI, Bhavnagar. Coming to the ISCB best poster presentation, Nidhi Jangir, University of Rajasthan, P116. Next is Narsimha Verma, BIDS planning. Narsimha is here. I think any of his friends can come and collect the certificate. Priyanka Kumari from Department of Botany, University of Rajasthan. Some of the presentation work came from the different because it's allied sciences. So they are working in biochemistry. So we included them, and so Priyanka Kumari from Department of Botany, University of Rajasthan. Kusum Uchania 109, University of Rajasthan. Coming to the RSC NJC best poster presentation, P69 Lalit Negi, MNIT Jaipur. Pushwinder Kumar, Punjab University, Punjab. If he is there, otherwise anyone from Punjab University can come and collect the certificate. Nobody is there? Okay. Dhananjay S. Nipte, BIDS planning. I think my conference organizing secretary also can join. Please come. Please. You be there. You. Please come. Come. Dr. Asa, Dr. Riya and Dr. Pragati, please join. Next is Poonam Kumari, University of Rajasthan. Monica Yadav, 152 MNIT Jaipur. Next is ACS, best person presentation. नहीं सर आप लोग रहिए सर ठीक ओके 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 फाइन so yeah now so ACS best poster what's your name Monica okay okay Anchal from Vanvastali Vidyapith Anchal if she is there Anchal sorry Prahlad Meher from IIT Jodhpur P130. Number Riya Nagar from MLSU Udaipur. Aman Chauhan, P102, University of Rajasthan. Ashish K. Tiwari, NIT Trichy, P18. Ashish is here. Any of his friend can collect one. Asa Rolania, University of Rajasthan. She's not there. Okay. Namrata Kotwal, IIT Jammu. So this is all about from my side. Thank you very much. Looking forward to that for the next version of AppCast. Will update soon. Thank you very much for joining us. Looking forward to seeing you, all of you at different platforms, different academic platforms across the country and globe. Thank you very much. Jai Hind.
Thank you very much, Dr. Batsira. Ladies and gentlemen, we are now moving to the end of the ceremony, which is vote of thanks, but it will be followed by national anthem. So be seated there. May I now request Dr. Pragati Fageria for the formal vote of thanks. May I request you to please be seated.
So, a very good evening, one and all. As all things come to an end, so is our conference AppCast 2023. I, Dr. Pragati Fageria, take this opportunity to propose a formal vote of thanks to those who have directly and indirectly contributed to organizing this conference. At the outset, I thank our guest of honor, Professor PMS Chauhan, Chief Guest Professor VK Singh, and Professor Ganesh Pandey. We are really enlightened with your knowledge and presence. I wish to express my gratitude to Chief Patron, Honorable Vice Chancellor, Professor Rajiv Jain, and patrons for their guidance and encouragement at every point of time in the organizing this conference. With utmost appreciation, I extend my sincere thanks to HOD, Department of Chemistry, Professor Meenakshi Jain, for her unconditional support. I want to express my heartfelt thanks to all the dignitaries present on dais and off the dais. Our team is highly grateful to the esteemed delegates and speakers of the conference for their presence and efforts in making the conference a grand success. I also express my sincere thanks to all the session chairs for conducting all the sessions on time. We had a great help from Professor Alakis Bishai, Professor A.K. Srivastav, and Professor Siddharth Sharma for evaluating the posters, and Professor Mark Talifer, Professor R.K. Gunsaria, Professor Debashish Banerjee, and Professor Lakshmidhar Rath for evaluating the oral presentations. I am really thankful to Dr. Preeti Sharma, Dr. Rohit, Dr. Mamta Raka for the smooth conduction of the sessions. A special thanks to the organizing committee, teaching and non-teaching staff for their unflinching support and coordination, specifically our students, Dr. Lalit, Dr. Rekha, Dr. Pratibha, Ms. Poonam, Ms. Pooja, Ms. Asa Kumawat, Ms. Asa Rolania, Ms. Amreen, Ms. Kusum, Mr. Ashutosh, Ms. Ekta, Mr. Rajesh, Ms. Akansha, Sobna, and technical committee, Rajesh ji, Ajit ji, and photographer Vinod ji. I express my sincere thanks to all the delegates and budding scientists who came here and presented their research work. I hope you all have enjoyed the great chemistry here. I extend my congratulations to all the winners and participants for their active involvement. I personally am thankful as I met with my supervisor, Professor Pandey, my examiner, Professor B.L.V. Prasad, faculty members from my alma mater, BITS Pilani, Anil sir, Ajay sir, Dalip sir, and many amazing juniors from BITS Pilani. We want to thank our heartfelt gratitude to our sponsors, RSC, ACS Publications, Elsewhere, Serb India, Thermo Fisher Scientific, Reaxis, etc., for their generous funding. Without that, the conference would not have to be organized in such a beautiful manner. We are really thankful to the social media and media persons for covering the conference news. Last but not the least, I wish to acknowledge all the amazing scientists and budding scientists who came here and presented their research work. I hope you all have enjoyed. I wish you have enjoyed your stay at Gulabi Nagri. I might have forget to mention some names, please forgive me, but thank you very much, each and every person involved to organizing this event. So, हमारे कन्वीनर के अंदाज में मैं लास्ट लाइन कहना चाहूंगी कि हमारे जीने का तरीका थोड़ा अलग है, हम उम्मीद पर नहीं जीत पर जीते हैं. थैंक यू वेरी मच.
I'm sorry, I forgot. Please forgive me. So, since the communicator was beside, I forgot to acknowledge. See, Doctor, thank you very much. I acknowledge the great effort put by Dr. Preeti Sharma. She is from physical education. Yeah, see, please come out. Come. She is from physical education, and you can understand how difficult to compare and do the anchoring for chemistry. Being from physical education, thank you very much, Preeti, joining us. And one more, my friend Rohit, he is from sociology. They did all this communication thing for chemistry. Thank you very much. Yes, sociology, yeah. And I take 1 to 2 minutes to just felicitate some of our team members. So my request, Professor Punyamurthy sir, just to kindly hand over the memento. Dr. D.K. Jangir, please be on the stage. D.K. Jangir, Professor. Anil Bansal, Dr. Anil Bansal. He is from Agarwal PG College, Jaipur. Dr. Sudhir Kashyap, I hope he is here. Okay, no problem. I will give him. Dr. Mamta Raka, Department of Chemistry. Dr. Mudit Gupta, Principal, LBS College. I think you can collect. आप ले लीजिए उनका नाम. On behalf of Mudit ji, you can collect, no problem. Mudit Gupta, Mudit. Dr. Anita Kumari, Department of Chemistry. I hope she's here. She's not there. Professor R.K. Gunsaria, Department of Chemistry. I think that, okay. My good friend from Department of Botany, Dr. Navneet Chaudhary ji. No doubt, our beloved head, chairperson of this conference, Professor Meenakshi Jain. And this is all from my side. Thank you.
थैंक यू सो मच सर. आई नो अभी जैसा प्रगति मैम ने बोला कि हम रूल्स एंड रेगुलेशन नहीं जिद पे जीते हैं, तो थोड़ी सी जिद हमने कर ली, यहाँ लाइव आपको डेमोन्स्ट्रेशन भी दे दिया. So with this, now may I request you all to please rise for the national anthem. Thank you once again for being lovely audience. And now you may leave. So goodbye. Hope to see you soon.
Thank you so much. Blessings.
Very well planned. आपकी पूरी टीम आपके बैक पे बहुत लंबी थी.